Good morning, good afternoon, good evening, everybody. Thank you so much for joining us on this ROV SuBastian Dive 679. We're here off the coast of Chile and off the coast of the Atacama Desert, the driest desert in the world, in fact. And today we're at the same site as we were yesterday. We're just a little bit shallower, and we're going to be exploring the ridge. And I'll pop up the bathymetry in just a second, just to give you some spatial awareness and some context. And today we're going to do something a little bit different than yesterday. We're going to be hunting for methane seeps. Good morning, good morning. Nice to see all the viewers popping in. Really nice to have you all join us. We're going to be looking at this area of sediment deposition and then moving our way up along a potential fault line in order to find some methane seeps. And then we're going to be doing a lot of multidisciplinary work to characterize the microorganisms and the geology and the macrofauna. So we've got a little bit of everything here today. As always, if you have any questions, ooh, nice rat tail. As always, if you have any questions, feel free to put them in the chat. We've got a very amazing and very eager team of scientists here in the mission control room waiting to take your questions. And without further ado, I'm going to pass it on over to our watch lead for today, Dr. Lauren, who's going to introduce herself and tell us a little bit about what we're seeing here this morning. All right, over to you, Lauren.
Thanks, Sophia. I'm Dr. Lauren Seiler from Stockton University in New Jersey, and we are here off the coast of Chile, off the coast of the Atacama Desert, and as Sophia said, we are hunting for methane seeps today. So we took a look at the bathymetry here, found some potential fault lines, and we're just going to follow along those fault lines today, looking for places where methane may be seeping from underneath the sea surface. And if we find these methane seeps, there's a good chance that they will be colonized by all sorts of interesting animals and bacterial nets. And so we're really hopeful today that we'll find something really cool to show you guys. Good morning, everybody. It's nice to see all of you again. Yeah, we're going to be looking for bubbles. That's right. So we did do some bubble searching using sonar on the ship, but sometimes methane seeps don't produce enough methane gas to be visible from the surface, especially if they're super deep. So we're just going to be going along the bottom now, looking for bubbles, looking for mats of bacteria and other signs that a methane seep is present. So we're just slowly making our way up this ridge right now. We'll stop and focus in on interesting things as we find them. Right now, I think the first thing that I noticed the moment we got down to the bottom was how much marine snow is down here. It's just a ton of marine snow. So as we mentioned yesterday, marine snow is caused by productivity at the surface. So we are in an incredibly productive part of the ocean here off the coast of Chile. There's a lot of upwelling in this particular area, which means that cold, nutrient-rich bottom water is being delivered to the surface, where it's feeding all of the phytoplankton, algae, and so all of the stuff that lives up at the surface is feeding off of these nutrients and is super, super productive. And all that productivity means lots and lots of biological activity that eventually sinks to the bottom as this marine snow. So we could be looking at clumps of excrement, clumps of algae kind of held together by exopolymeric substances, the goo that they secrete and helps them clump together. And it all kind of sinks down to the bottom slowly in this marine snow, which really lives up to its name. It really looks like a gentle little fall of snow here. And each one of these particles is actually colonized by millions of bacteria that are slowly breaking these particles down, using it for food. And particles that are not broken down will eventually be buried. And this is a really important way that carbon is stored on geological timescales. So as this carbon sinks to the bottom, if it's buried in the sediment and eventually becomes rock, it gets stored for long, long periods of time. If it's broken down, it eventually becomes carbon dioxide gas and gets re-released to the atmosphere. So marine snow is a super important part of the global carbon cycle. Good morning and good afternoon to everyone in the chat. How y'all doing? It's nice to see you all. Yeah, I'm seeing Turkey. I saw Colombia. Fantastic. Oh yeah, can we zoom in on that there? Yeah. What do we got here? Seeing a lot of these beautiful acorn worms that we saw yesterday. Not as much of the, what are they called? Xenophyophores that we saw? The xenophyophores. There were lots more at the deeper site. I'm not seeing as many of those today. Seeing a couple, I think, but not nearly as many as we saw yesterday. We're like everywhere. There that worm is. Hey, buddy. Woo! This bottom, the bottom here is super, super fluffy because there's so much marine snow. So every time we set down or get close to the bottom, the thrusters kick up a whole bunch of that particulate material. Oh, they're asking about the big sea cucumber. Yeah, we did get it up. It clogged the hose yesterday, but we did manage to get it. It's a big fella. So we're going to be doing less biological sampling for the first like 3/4 of this dive. And then we'll save all of our biological sampling for the end after we've had a chance to really explore this ridge. But as we go along, if we see anything super interesting, of course we will stop and take a look at it and potentially take a sample. So still lots of opportunities to see cool things as we make our way up this little mountain here. Hannah described it really well yesterday to me. She said it's like taking a hike up a mountain at night with one flashlight. Someone checking in from Sweden. Hello, hello. Got viewers all around the world today. Fantastic. All right. What's that down on the bottom there? Do we see a sea anemone? There's a... Our SciCam keeps giving us some issues today. How about this guy? Can we zoom in on him? This long boy. Pretty.
It's a nice eel.
Yeah. A silvery color. Very pretty. Full disclosure, I am a microbiologist, so I'll talk a lot about microbiology whenever I'm leading these watches, but when it comes to animals, I'm going to be relying a lot on the other experts in the room to tell us what we're looking at. Good morning, Ian. It's nice to see you again. Ian's a good friend of mine. I love it when my friends jump into the chat. My husband should be joining us soonish, and maybe my dad as well. They're watching from home. On the way down, we saw a really, really pretty worm, maybe? I think it was some kind of polychaete worm. It looked like it was made out of glass.
I think it's a floater.
A floater?
Yeah.
Super pretty. It's really something to get to see these animals in their normal surroundings, in their native habitat, just living their lives. It's completely different from seeing it in like an aquarium or in a book. You really get to see them. Oh, someone from Iquique. Hello. Iquique is not far from us, a couple hours north. Good morning. Thanks for joining us before you go to work. Okay, is that another acorn worm there? Mm-hmm. Yeah, some brittle stars. Some more of those xenophytophores, but not nearly as many as we were seeing yesterday. Lots of brittle stars here and buried in the sediment. Pretty. I love that color. Purple's my favorite color. It's just very exciting to see so many beautiful things. What's that over there, that purple? Is that another worm or something different? Mm. Big sea cucumber? Buried boy. Yeah. Looks that way. There's a very cool shrimp as well, with extremely long legs.
He doesn't seem so sure about that.
Every time we tried to catch a shrimp yesterday, they were just like, nope, not today. Starting to make our way up this hill here.
Yep.
Definitely looks different from the other site we were at yesterday, even though we haven't really gone super far.
Ooh, there's like a stalked something in the distance.
Oh, crinoid maybe?
Yeah. Get a pointer. Wow.
This eel earlier might have been a sorcerer eel, maybe? I don't know my fish, so I'll take your word for it. Yeah, I think that's a crinoid. I love those.
Annie, I can't see.
Yeah, that's pretty. Look at that.
That looks like an umbrella. It's a really bright.
It's not coming back. Long time. I'm concerned about this.
So, if you're just coming in now, we are off the coast of the Atacama Desert in Chile, searching for methane seeps along what we suspect are some little fault lines along this ridge. We haven't quite reached where the faults are yet, so we're going to be checking for bubbles, we're going to be looking for microbial mats. Our SciCam is having some issues, so we're gonna see if we can.
Yeah, the viewers are looking at our sit cam right now.
Yeah.
Our SciCam is not Our Psycam needs a bit of coffee today.
Yeah. Don't we all?
Yeah.
Whom amongst us? Do you want to show them the bathymetry? Oh,
I definitely do.
yeah.
Let's do that. I just want to get it set up real quick.
Yeah. So we can show you kind of the path that we're hoping to take today as we swim along up this ridge. Jess, you have a shout out in the chat from another ROV pilot on a different boat. You have a shout out in the chat. Yeah. Another ROV pilot?
That's Ben.
Oh, that's Jess's husband, Ben. Hi, Ben. I've heard a lot about you. All good things. Just wanted me to make sure you knew that. Got a sea anemone down there and another sea cucumber. The fish going by. Just gradually making our way uphill here till we reach this ridge.
Oh, I also see our colleagues over at Universidad Católica del Norte is tuning in. Good morning, buenos dias.
Good morning.
From Chile.
Yeah. Buenos dias.
I do want to know who is chatting as Sala de Colecciones Biológicas, though. Is it Maxi? Is it Javier? Who is it?
Yeah.
Show yourself.
That one looks like a pickle.
Yeah.
Giant sea pickle. Oh yeah, Ben's working on the new ROV at the Florida Institute of Oceanography. Very, very exciting stuff. All right, let's take a look. Show us the bathymetry. Cool. So here's this ridge we're looking at here. The long white line is where we landed, and we're just making our way up towards this ridge. This little, I don't really want to call it a mount, but yeah, little ridge. So we're gonna make our way to the base and then just start climbing up along some of these fault lines that we think are here. It looks like there's like 2 or 3 fault lines kind of converging at this one spot, so we chose a path that would allow us to cover as much of that as we can today. Oh, little sea cucumber leaving a trail along the bottom there. So hopefully we will find some bubbles or some cool microbial mats as we ascend this very interesting feature here. Oh, someone in the chat wants to be an ROV pilot. That's amazing. That's awesome. Beautiful. Look at that. Sea anemone? Anemone. They're so hard to pronounce. And the green, is that a urchin?
Roger.
Yeah. Oh, sea urchin there. Pretty. Very pretty.
Okay.
All right, we are
Stand right there.
making our way up to our first dive waypoint.
We're going to steal your fan for our converter so our camera might not go out as often.
SciCam keeps giving us grief. Just doesn't want to cooperate today. Seeing a few more sea cucumbers here. Still quite a bit of marine snow, though I don't think it's falling as heavily as when we landed. I don't know. It's kind of hard to tell. This is such an incredibly productive area. Ian, it's your birthday? Happy birthday, Ian. I didn't know it was your birthday today. We have a birthday on the ship, too, so. Yeah. Check out the MATE program if you want to become an ROV pilot. Oh, wow, that's awesome. That you want to be an ROV pilot because of Schmidt Ocean. That's amazing. It's so great that an institution like this exists to inspire people and bring these images to the public. There's no point in doing science if we don't share it with everyone. Then who are we doing it for? Oh, okay. Well, now I'll remember. Jax is September 1st and you're June 1st. Another purple sea cucumber on the bottom.
Yeah, our SciCam is still flickering
On the way up. I don't know.
We're still having issues with our SciCam. I think that's another one of those big pink sea anemones that we saw yesterday. We saw a huge one yesterday. Hopefully we'll be able to get the SciCam in working order so we can get some nice close-ups. Some of these. But that's the trouble with doing science, is that sometimes your equipment doesn't work, especially when you're at sea. It is a cool expedition. So we did find a few living fossils yesterday. We found some brachiopods. We found, what was the other one called? Now I'm blanking, of course.
is it a Titan?
The chitin, yeah, the leptochitin. Yeah. The leptochitin and a couple of brachiopods. And of course, we'll be on the lookout for more of those when we come up to some rock outcroppings, because they're brachiopods, so of course they want to have something to attach to. So if we come up upon any rock outcrops, we will stop and sweep.
Ooh, hello to Jeremy Horowitz in the chat. Just saw your new publication about a new family of black corals discovered. Very, very
Ooh,
cool.
that's
From
awesome.
our expedition last year. Nice to see you in the chat.
This is a big pink thing. It's a little pants to the side of it.
Ooh, pink koosh ball. I'm just kidding, of course. Do not touch the forbidden koosh ball.
Let's see if I can.
No? Oh.
Sorry.
Who's saying sorry?
I've seen a lot of these little mounds here. Not sure. Not sure what's causing those. Little mounds and little pock marks. No microbial mats yet, though, which is what we would ideally like to find. If we find an undersea dinosaur, I promise, I will let everyone know. Oh, what's that down the bottom there? Another sea cucumber or nudibranch, maybe? That looks like an... Oh, nope, that's got legs. A worm?
I got it.
It's like all of a sudden, legs! Lots of legs. I think that's the same species that we saw coming down in the water column, maybe? Looks pretty similar.
Potentially.
It's hard to say. Yeah, got a nice floating 360 shot of the first one. I would also not be sad if we found a cephalopod, frankly. We saw a Dumbo octopus yesterday in the water column, but we haven't really seen any on the bottom. Octopi or squid. But I'd be down for it. All right. Gonna keep making our way up here. We're almost to our first waypoint, which means we're almost to where the ridge starts to really get steep. Seeing some more of those sea cucumbers we saw yesterday. No shortage of sea cucumbers on this expedition. So if you like sea cucumbers, you are in luck. And lots more of these little mounds and pock marks, which could be promising. I think we're coming up on another anemone. Yeah, we got some good samples of rock yesterday. They did crumble pretty easily, which is kind of strange. But we did manage to get some nice big pieces of rock. Got some very good cores. I don't think any of the cores failed when we went to subsection them, which was really nice.
How does xenophyophores come up? Can you bring them?
Here's our anemone. More of these xenophyophores.
Do the xenos make it okay?
Hmm?
The xenophyophores, did they make it on deck all right?
Yeah, Yeah, the xenophyophores also made it on deck okay, I believe. Sampled a couple of those. They look like Boston lettuce. Right, little shrimp there. Ooh, okay. I think this is where things are gonna start to get a little bit steeper. We are almost to our first waypoint here. Oh, kicking up a bunch of dust here, so some particulate matter. Ooh, big pink sea cuke. Spiky boy. Yeah, Sebastian's playing in the mud. Well, as a microbiologist who loves all things muddy and slimy, I am in my element right now. Couldn't be happier. Happy as a little clam at high tide, as they say. Seeing lots and lots of particles here in the water. Wow, yeah. So that could be stuff that we're kicking up, but there's also plenty of marine snow coming down from the surface. So if you missed that comment earlier, marine snow is particulate matter that is produced at the surface of the ocean. Most of the life in the ocean is concentrated in the top part where there's plenty of light. And so all that light feeds phytoplankton to produce tons of food for fish and other critters. And then all of that productivity eventually sinks down to the bottom as deadfall and excrement and dead cells and clumps together as this marine snow, which each little particle is sort of like its own little micro community. Whatever doesn't get eaten up by bacteria gets buried along geologic timescales. Very important part of the global carbon cycle. The burial of carbon in the deep ocean. Yes, we have been finding microplastics down here, unfortunately. Which, yeah. Beautiful acorn worm. I love how it's got a little frill up by its neck. I don't know if you call it the neck, but sort of looks like a little cravat. It's very fancy.
And it's been any better or no?
Yeah, acorn worm.
We zip tied a bunch of fans up. Took that SOP out, it was smoking hot.
Looks like the cycam's behaving better. Yeah, it is disappointing that we're finding microplastics, but it is also not surprising. We're finding plastics in places where people have technically never been before. It just makes its way down. We are in the Anthropocene, the geologic age of humanity. Some shrimp here, vibing. All right, we more or less reach our first waypoint. Now we're going to start climbing up this ridge. Pretty little urchin here. Looks like a lemon. Or like one of those lemon cucumbers. So pretty. So pretty. Yeah. I've got Drew behind me logging all of our biological observations?
It is.
Yeah. And up we go. Keel is telling me that the identification of shrimps is complicated. And you can't always tell what shrimp you're looking at just by taking a glimpse at it. Oh, just a sec. Seeing more of those xenophyophores. Ooh, big green sea cucumber here. Spiky pickle. Hi, husband. Oh, I see. Okay, so it sounds like my oldest child is claiming rights to the TV.
We have not seen this species before. And we just warm up the people to get a sample, and we were interested.
Which species? This one?
This one, we have not seen the species before.
Okay. All right. Okay.
It's gonna get dusty, but then we're gonna figure it out.
As we have not seen this particular species of sea cucumber before, I have received a request to sample him, so we're just gonna take a brief pause here.
We're on seep.
Yeah, we're gonna sample this one. I agree. The octonauts have a lot more going on, but
Hello, ROV.
Yeah, can we pause the move, please?
in fairness, they have a script writer. We're just kind of going off the fly here.
Thank you.
How's the baby?
What would you like to keep? Put it. Would you like to put it? I want to collect this back.
Where would we like to put it? Bio box or bio box? Okay.
Look at it first.
I agree. We should put some googly eyes on this guy. The question is, which end do the googly eyes go on? All right, we're just gonna let let some of this dust settle so we can get a nice visual on him before we take him off the bottom. Oh, hi, Dad. We
Whatever
found
one
a
you
sea
like.
cucumber that we have not observed
Yeah.
yet.
Maybe
So
kind of start with the first one.
we're going to let the dust settle a little bit, get a nice shot of it, and then take it for one of our bio boxes. And then continue to make our way up.
What job are you doing?
Alright, so we're gonna take her little pictures. He's big. Another big sea cucumber. Now we're just gonna very gently lift him up off the bottom. Yeah. Oh. 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 Maybe we're gonna have to try a re-grab there. Okay. There we go. And just put him in one of our bio boxes.
Alright, poor child.
As you can imagine, it is tricky to sample these organisms delicately and without causing any damage to them. Yes, we have different colored grips than we did yesterday. We had the yellow ones yesterday. Today they're red. I think we're trying something slightly different. So this one's going in Bio Box 1A. And that was requested by Nick. Whoop. There we go. Floppy boy. Yes. All right. And let's continue to make our way forward. Nice job, everybody. So, if you're just joining us, we are making our way up this ridge off the coast of the Atacama Desert in Chile, hunting for methane seeps. There's a couple of potential fault lines along here based on our bathymetry data. So we scanned the bottom with our sonar last night and the night before, and this spot looked like we may be able to find some methane seeping from below the seafloor here, where these fault lines meet. So the samples will be studied by our biologists on board, and I believe some of them will be going to the local museum in Chile. After they've been collected and studied. We're doing our very best not to take anything that we cannot use for scientific purposes.
Hydraulic prop bars.
Alright, now we're going to start to make our ascent up this ridge towards our next waypoint.
Should not think it's 70. No, you're good. What the 75?
Making very good time here. We are, of course, tethered to the ship above us, so we can only go so fast. But we also don't want to miss anything cool on the bottom as we go along. So we're just taking our time here. My dad and my husband have tuned in, and also my friend Ian is here.
I'll have a fan on and then get in their blankets just because.
Oh, there's another one of those green sea cucumbers. They're so bright. It's really remarkable. Seeing another sea anemone, another crinoid. Oh, there's quite a few of those green sea cucumbers here. We did not see those yesterday, so it's interesting. We're not super far from where we were yesterday. We're a little bit to the northeast and a bit shallower than our dive yesterday. We're not seeing as many of those xenophyophores that we did. Yesterday there were so many all over the bottom. Today we're just kind of seeing a scattering of them. These sea cucumbers are so pretty. Looks like a velvet couch. Like a really fancy velvet sofa with a little ruffle at the bottom. Yeah. Very stylish. I love it. Alright, things are going to start to get a bit steeper pretty soon as we climb up this hill. Keeping an eye out for bubbles, for microbial mats. Seeing a lot of these little bumps.
What kind of temperatures are you expecting to see? What kind of temperatures are you expecting to see on methane, just the bacterial mat in particular?
If there is methane coming out of the bottom, we will likely see a temperature rise as well. Not as hot as a hydrothermal vent by any means. So a lot of times methane seeps are called cold seeps, not because they're cold, but because they're colder than a hydrothermal vent. They're still much warmer than the surrounding seawater. So probably see a temperature rise. We would see bubbles, we would see pock marks in the surface, and hopefully some microbial mats to sample. And here we have a shrimp. We've been trying to sample the shrimps, or we did yesterday, but every single one was elusive. We were just not having it. Look at those legs. Oh, fish. Is that a rat tail again? So, yeah. I'm learning my fish. I cannot identify fish for the life of me, but I'm learning. Super pretty. He does not want to be photographed today. He's like, please, no paparazzi, no autographs. Okay, and up we go.
That's awesome.
This is a very exploratory dive for us. This is a part of the ocean that is not particularly well mapped, and methane seeps have been found to the south of where we are and also to the north of where we are. And since the whole margin is pretty similar along the coast of South America, I would assume that there would be methane seeps here as well. But up until now, folks have not had too many opportunities to look for them, so we're gonna see what we can find. What's that over on the right there? Another polychaete worm? Nope, anemone. Never mind. Looked like a coiled up worm, but that's an anemone. And a shrimp. Very pretty. Very, very nice. All right.
Let me see some more xenophilus.
More xenophyophores. More of these big pink shrimp. There is a maximum number of collections that we can make per dive just because we only have so much space on the ROV. So we decide before the ROV dives how many bio boxes we want to put on it. And sometimes we can put more than one animal in the same bio box if they're the same animal. Other times we have to isolate them. We can also put specimens in the coral quivers. But there's a limit to how many we can reasonably take. And also, we don't want to be greedy. These are living animals that we're taking from their habitat, so we don't want to take an animal without good reason. So we're trying to be as judicious as we can in our specimen selection, both for practical reasons and also because we want to respect this environment. Ooh, what is that? Is that a oh my god. It's a trash bag. Wow.
Plastic bag.
Unbelievable. We're at 2,777 meters, and there is a trash bag. Well, we're just gonna get that out of here. That does not belong here.
But there's stuff on it.
We may need to recommend
I Canada is gonna have stuff on it.
Yeah, wow.
Yeah.
It's a big trash bag, too. It's like a black garbage bag. Wow. All right, well,
If we put this.
where should we put that?
We got a lot of space. It would fit in one of the small bio boxes.
We might be able to stick into a quiver.
Hmm?
We might be able to stick something into a quiver.
Stuff inside a quiver.
Okay.
Yes.
We're gonna stuff that inside of one of our quivers. We don't want to leave that here.
We don't really have a trash collection button, so
Yeah, we don't generally put a trash collection container on the ROV, but maybe we should. Yeah, that is a real bummer.
I'll note it as a biology collector.
We're all the way out.
All right, so
Oh, yeah. Sorry, dude.
oh yeah, yep. There may be some microorganisms, of course, that have evolved to degrade plastic. So we may find some of those down here. You find some in the sediment or maybe even on this bag.
We can grab a look at it.
There are microbes that have evolved ways to do just about anything you can imagine. There are microbes that can eat dynamite. Yes. There are microbes that can break down plastics, oil from oil spills. It's a really incredible thing to me about microorganisms is that if there's a way to do it chemically, then they've figured out a way. They've evolved a pathway for it. A lot of times there will be some theoretical metabolic pathway, and we'll say, well, that could exist in an organism, but we've never seen it yet. And then eventually we find a microorganism that has it. It's just a matter of finding it. A lot of times we find them in wastewater treatment plants, actually. We found so many new metabolic pathways in microbes in wastewater.
Really?
Yeah, I guess it's because they just there's a very high turnover rate and they get exposed to so many different compounds. And every wastewater treatment plant has a microbial community of its own that's completely unique and hasn't really been fully characterized. So a sewage plant will have just microbes that have been seeded there to start breaking down waste. And then it just sort of evolves on its own, and the folks who run the plant just sort of encourage the breakdown of the waste and certain metabolic pathways and oxygen conditions and things like that. But other than that, those communities are not very well understood, and every single one is probably quite different. So we're always finding new metabolic pathways in microbes, and a lot of times they are in these wastewater treatment plants. Ideonella sakiensis is a plastic-eating bacteria. Well, I'd believe it. I'm studying Ideonella back home, but as a potential iron oxidizing bacteria. Yeah, so I've
They can
been.
do several things.
Yeah, oh yeah, yeah. Lots of bacteria are multitaskers.
We actually are looking at trying to identify plastic degrading bacteria in our lagoons where we work at too.
Oh yeah, Drew says he's trying to identify plastic degraders as well at home in Florida.
Hello.
I didn't see him there earlier.
I've been here, logging everything.
I've been here the whole time.
Logging most everything. And Tony is my backup logger.
Yeah, Drew had to step out of the room for a water pump issue, but he's back. All right, so let's safely stow this plastic bag, because there is no way we're leaving this here where it does not belong.
Armando's already said he wants to look at for plastic-eating bacteria.
Yeah, we could definitely look for some plastic-eating bacteria on that.
I think we're all gonna
Absolutely.
take a cut.
Yeah, why not?
There's always a possibility.
Yeah, you just never know what you're going to find. I have to hand it to the ROV pilots. This is the trickiest crane game ever.
Yeah.
I don't think my hands would be steady enough. I'd have to back off of the caffeine, I think, probably. We're just gonna try to twist that in there and put the lid back in. And then continue up this ridge. Hello, Australia. What time is it in Australia? It's gotta be like the opposite, like nighttime?
It is late at night.
Yeah.
Very late.
All right, yeah, we're getting that down in there, I think.
Yeah, we are stationary at the moment.
Nearly midnight in Melbourne. Wow, yeah. Well, thank you for joining us so late at night. Oh, this is tricky. You can see there's also a lot of particulate matter down here. If you're just now joining us, we are in an extremely productive area off the coast of Chile, the coast of the Atacama Desert. Lots and lots of productivity in the surface, which creates all of this falling particulate matter all around us. Even down here at 2,500 meters or so. And we're also kicking up a lot of material from the bottom, which has settled. Interestingly, when we took the cores, though, they seem to be pretty solid. Maybe held together by a silicious ooze from diatoms. Maybe some clays. We've got our geologists looking at subsections of each of our cores after they come up. Alright, I think we're going to put that bag in a bio box instead, because it's just not going to fit in a quiver. Rather, it would fit, but it's too difficult to get in there, I think. Maybe when it comes up, we'll split it up amongst the microbiologists so we can look for microplastic degraders. Right. Nudge that in there.
Box 2A.
Biobox 2A, big plastic garbage bag.
I'm just gonna mark that down as other organism.
Hmm. Did you also make a note of there's like an anthropogenic? There's definitely a data button for garbage.
Oh, I made the anthropogenic observation.
Okay, anthropogenic observation and then
We didn't
other.
have a trash bag graph. So
I wonder why we wouldn't have trash bag listed under biological samples. Okay. Put the lid back on the coral quiver, and then we can move along up this hill. Once we get to the top of this ridge, we're gonna decide what direction to take. There's two possible ways we could go to follow along the fault lines. So we're gonna see what we see. Oh, there's a tiny something. Shrimp? Swimming there? A little something with legs. That's what I look like when I'm swimming. All right. Get that closed there. And we can head on out. Trashiest plastic bag is. It's unfortunate, but it's the reality of things. Alright, we are lifting up a little bit here. Wow, there's just a ton of particles in this water. Yeah, it does look soupy. Let the dust settle a little bit. Get through it, yeah. Things should clear up in just a moment here. There we go. Much better. Oh, we got a little fish on the bottom. I think another rat tail. Couple of fish.
Bridge, ROV.
ROV bridge?
Yeah, we can resume the move now, please.
Copy that. Resuming the move.
All right. Falcor's following along behind us. Falcor 2. So we are climbing up this ridge. He attempts to follow along a fault line. Oh, another beautiful anemone. We got a huge one yesterday.
Want one of these?
No, we already got one. I don't think we need another. Just gonna appreciate it in all of its beauty.
yeah, we'll go.
Oh, and to the left?
Yep. We to come back to it.
Yes.
It's gonna be pretty messy, but I see it.
We're expecting to see
I'm gonna sit down right in front of it and then it'll clear itself. Yeah, that's not an issue too. Break the laser. Oh.
Another pretty anemone here. So much. So much stuff in this water. 'Cause in addition to all of the marine snow, every time we land, we kick up a bunch
You want to
of
plus
this
this?
fluffy stuff.
Okay.
This is standing. I thought it was something else.
Nope. We don't need it.
Nope. Okay.
Let's keep going.
This is important. We're showing the beginning.
Where's past this?
Yeah, get out of this dust here. Fantastic. All right, gonna continue to climb up this ridge. Keeping an eye out for bubbles, pock marks, temperature variations. You've already climbed up quite a ways. We're at 2,568 meters now. So we're just making our way up this hill. Haven't really seen much rock yet. It's been mostly a very fluffy sandy bottom here. But we may see more as we get to steeper places where all of this organic material can't really settle and slides down instead.
Yeah.
That cam keeps giving us issues. I've seen another one of those nice clear sea cucumbers that we've been seeing quite a few of. Very pretty. Some fish, brittle stars, seen a fire forest, lots of that, although not as much as we saw yesterday. It was everywhere yesterday. I suspect because we're on kind of a steep incline, we may see less stuff until we get up to the top. But I'm not sure. A little sea spider down there, maybe? that?
zoomed in on,
Looks like another
and that's why you
sea cucumber. Sort of covered in some sand there. Some dust.
been going on and off.
Yeah.
Just for you, right?
And we're taking a pause to swap out pilots. All right. And up we go. And as you can see, we are climbing up this ridge here.
Carry my momentum.
Sorry, I'll move them waypoint.
So we're making our way up this hill.
Updated waypoint, please.
Keeping an eye out for
Great, thank you.
methane seeps, which would be indicated by bubbles, microbial mats, temperature anomalies. I'm going to continue to stop and look at cool animals and other interesting features as we go, but today's dive is primarily for the purpose of looking for these seeps. We're going up along a ridge where we think there are a couple of different fault lines running underneath. So these are good places to look for methane seeping from underneath the seafloor. Methane is a great source of energy for deep sea biological activity. So we would expect to see mats of microbes, maybe tube worms or mussels, maybe some yeti crabs. That would be great. Shrimp. Oh, scared him. The shrimp have been pretty shy. And there's another one of those great big pink anemones that we sampled yesterday. So we've been seeing quite a few of those. They are so big.
You can go get people in another room. That's something. Okay, thanks.
I keep seeing these like little hills of sediment. Little divots. I keep an eye out for outcrops of rock, because we are also looking for living fossil brachiopods, other mollusks. So if we see rocks, we'll stop to take a look. But so far, it's been pretty sandy on the bottom. Lots of this fluffy organic material, collected marine snow from the surface.
I only saw him in the logs. I was not here.
No seeps, I take it.
No seeps yet, but the day is young.
How would you know if you see one here? You would see bubbles would be the main thing you're looking for? Or
Yeah, we would be looking for bubbles. We'd be looking for mats of bacterial growth, temperature anomalies. We often see like little pock marks in the sediment.
what causes these?
So,
Material or something?
yeah, so these methane seeps are pretty common along margins like the one that we're on right now, where you have one tectonic plate sliding underneath of another. So here the ocean plate, the Nazca plate, is sliding underneath the South American plate, and that's what forms the Atacama Trench. And the sliding of one plate under another causes the one on top to buckle and creates what's called an anticline. So these are like ridges in the plate around which sediment collects, and methane gets trapped underneath and then bubbles up to the surface. And methane is a great source of energy for all kinds of microscopic life. There are bacteria that eat methane. And then they serve as a food source to other microbes. There are microbes that can use methane as a source of energy for fixing carbon. So we are keeping an eye out for these critters. If we see a big mat of microbial growth, and they could be all kinds of different colors, so they should stand out against the background. They might be white, they might be pink, they might be orange. That would be a good indicator that we have methane seeping up from under the seafloor and feeding all of that microbial life. And then, oh, there's a little brittle star kind of climbing along the bottom there. And then other animals will feed on those bacterial mats, so we may see tube worms, mussels, crabs, like a little oasis on the bottom. We may also spot what's called a false seep. So these are seeps where there's methane being produced, but it's not from geologic activity. It's caused by a deadfall. So something large dies and falls to the bottom, and it's broken down by microorganisms, and some of those microorganisms produce methane gas as a byproduct. So if something dies and is buried, methane can be produced by the decay of that dead animal, and it looks like a methane seep, but it is not a methane seep. So a true methane seep is produced by geologic activity, and a false methane seep would be produced by deadfall. Honestly, it'd be interesting if we find either one. So I think we're coming up on a great big sea urchin up here. These are very pretty. We're continuing our climb up this hill, ridge. You can see that marine snow, it often looks like little bits of snow, hence the name, but sometimes you'll see these like longer wispy bits, and those are held together by what's called an exopolymeric substance or exopolysaccharide, EPS. These are produced by bacteria to help them stick together to make biofilms, and so that's what really helps like marine snow clump together. It's this like sort of snotty mucousy type substance that bacteria produce. It produces these like long wispy bits that fall down to the bottom of the ocean. And if they get buried in the sediment, they can eventually turn to rock. So this is an important sink for carbon. It's a way for carbon to be stored on geologic timescales. But the whole time it's falling, it's also being broken down by bacteria that are turning it back into carbon dioxide, which can be released to the atmosphere. So the production and the decomposition of marine snow is a really important part of the marine carbon cycle and the global carbon cycle. We will definitely get excited if we see bubbles. I will try to back away from the microphone because I know I'm going to scream, and you don't need to hear that. I'll do my best. But yeah, if we see bubbles, it's going to be a party in here. But we're fairly confident that seeps are here because this is a very likely margin for them, and they have already been detected much further south, well, not way further south, but further south than we are now, and also to the north of where we are. So we're kind of right in between where a whole bunch of methane seeps have been found along this margin, and we just happen to be in an area that the bottom has not been very thoroughly explored. Thank you for the permission to scream, but I will try to at least back away from the mic a little bit. I don't want to make anybody go deaf. We found a lot of sea life yesterday. Lots and lots of animals. Seeing fewer things here along this ridge. It's not necessarily a bad thing. But it is different from where we were before. We were kind of on this flat little plateau where a bunch of marine snow could collect and probably feed all of the life down here. So, in addition to being an important way to store carbon, marine snow is a really important source of food for life on the bottom of the ocean. There's no light down here. So most of the productivity in the ocean takes place in the first 200 meters or so, where light can penetrate. So phytoplankton and algae and seaweed can capture that light and make food, the same way that plants do on land. And deeper down, there's no light as a source of energy, so energy has to either come from above in the form of sinking particles like this, or from below in the form of chemical energy, like at methane seeps and hydrothermal vents. So some of you, as you were reading the description of this particular expedition, might have come across the term astrobiology. Many of us on board the ship are astrobiologists. If you're not familiar with the term, astrobiology covers a whole lot of different disciplines. It's extremely interdisciplinary, but the main crux of it is that astrobiology involves trying to explain how life originated on Earth, what conditions were required for the origin of life and how it developed, and how life might be found on other worlds and under what conditions, what places we might be likely to find alien life. This can also include space biology, which is distinct from astrobiology. Space biology involves how do we take life from Earth, like human beings and plants and animals, and move them to another location, say if we decide to colonize Mars. So primarily, the scientists on board the Falkor 2 right now are interested in finding ancient life from Earth's history, but also considering the possibility of exploring the deep sea as an analog for life on other ocean worlds. There are numerous ocean worlds within our own solar system. Moons of Jupiter and Saturn that we know have oceans on them. They're covered with ice. So they're fairly cold, but at the bottom of all of that cold seawater, there may be hydrothermal activity or other types of geochemical reactions that produce energy and warmth for living things to survive and thrive. Ian mentioned the panspermia hypothesis. So that idea is that life originated somewhere in the universe or in our solar system and then was transported to Earth, potentially by a meteorite or something like that. I think it's an interesting idea. What I don't like about it is that it's basically kicking the can further down the street in terms of solving the origin of life on Earth, because it just moves the question to another level. But it's certainly a possibility that Earth was seeded with prebiotic compounds from some other location. We've found amino acids on the Murchison meteorite, for example. When was the last time we found bubbles? Well, we haven't found bubbles yet on this expedition. So this is only dive number 2, though, so still got plenty of time. But we do have folks on board who are experienced in this sort of thing, so fortunately we are not going in totally blind. So we've chosen this location to look for bubbles based on the bathymetry data that we have from the mapping that we've been doing every night in between stations. It looks like there's 2 or 3 fault lines kind of converging here, which is a really good place for methane seeps to be found. They're usually found along fault lines and changes in the topography. So we're just keeping an eye out for those, for mats of microbial growth. Right now we're almost halfway to our next waypoint, which is on the top of this ridge. That's a really gorgeous purple sea cucumber. Sea fan. Another rat tail fish.
Connect the coral to the left.
Huh?
I suggest to connect the coral to the left.
The sea fan.
Which is the rest of the well, it's behind us now. I think it passed it.
Yeah.
Yeah, okay.
If we see another one, maybe. If we see another one, maybe.
Yes.
We're starting to see some outcroppings of rock. There's a sea fan over here that Gil would like to take a look at. Really big purple anemone. Oh, that fish is checking us out. It's giving us the side eye. And I was like, I don't want anything to do with you.
You want to collect?
Yes.
Do we really need the sea fan? Or can we continue to go? Okay, let's stop and take this sea fan really quickly.
Lauren, we did this?
Yeah, we're gonna take this one.
Bring it, Geology.
Are we bridge?
We're gonna take a sample here. Can you pause and move?
Yes, you can.
All right, where would you like this? Quiver?
Yeah, if it'll fit in a quiver, I think we should put it in a quiver. Rather than use a bio box.
Could you take a zoom in picture first?
Can we zoom in and get a picture of it before we sample it, please? Thank you. Oh, the polychaete worm that was on that urchin yesterday, or rather sea anemone yesterday, I think that escaped. We took his house. He was none too pleased. It's very pretty. Yeah, it looks like there's an isopod on the sea fan. So we may get both of those.
The museum asked him to take more coral.
Gotcha. Okay. So we'll be taking this specimen for the local museum in Chile.
If we want to connect the isopod, it's going to be better with a suction, but the coral needs the grab, just as I said.
Okay. Can we potentially suck the isopod off first and then grab the sea fan and put that in the coral quiver? That way we can get both. Can put that in coral quiver 1. But I think we're gonna the sea fan. The isopod, we're gonna use the suction and put it in one of the suction sampler jars.
Sir?
Yeah, there was a comment that panspermia raises the question of what's considered a basic unit of life. If comets brought water to early Earth, which led to life, is that panspermia? My argument for that would be no, because there's lots of places that have water that may not have life. Comet meteorite may also have brought organic compounds, but those don't necessarily count as a living thing. So, but there's a lot of debate as to the definition of life among scientists and certainly among astrobiologists.
Can you come back
So
with some?
Sure.
that could be a very long discussion regarding what is a living thing, what is life, and what is not life. The NASA definition of life, incidentally, is a self-perpetuating chemical system capable of Darwinian evolution. But that's only one possible way to describe it. All right, so we're going to try to get that little isopod with our suction sampler. And then go in with the pinchers to get this coral for the museum. Oh. Ah, I think we got him. There's also a sponge or something at the base of the coral. Looks like a sponge.
Went in?
That was
One.
that went into which section?
That went into suction sampler 1. Okay. And now we're going to take the coral and put it in coral quiver 1, please. Good morning, folks on the Hess Deep Rift. That's exciting.
Nothing's in any quivers yet, right?
No, the quivers are empty. We tried to put a plastic bag in there, but it didn't go so well. All right. Let's get that coral. Eel. Just got photobombed by an eel.
Is that enough for you?
How much do you want, Gil?
Try to get from the base.
Try to get it from the base? I guess as much as you can. Okay. It's pretty. All right.
That's what you did, coral quiver 1.
Yep, coral quiver 1. Yes, we changed the tips on the manipulator arm since yesterday. Good observation. They were yellow yesterday, they're red today. I think these are working a little bit better. Some of you may know we have a 3D printer available to us on Falkor, so whenever we need new parts, we can make them right on the ship. When you're at sea, you can't go to the Home Depot. Sometimes you gotta make your own stuff. Alright, so we're just gonna put the lid on that. Yes, we are currently at a subduction trench in active margin, where one plate is sliding under another plate, and that's what forms the Atacama Trench. And then hopefully what will form the methane seeps that we're looking for. We know that they are further to the south and further to the north of us. We're just in an area where no one has found any yet because nobody's really had the opportunity to look, so. All right. Nice job. Let us continue our ascent. Someone's asking about eel pouts associated with methane seeps. Does anybody know about that?
What's it?
Eel pouts at methane seeps? Eel pouts. Someone is asking for Jenny. Right. Can continue climbing upwards. Yeah, let's keep going. And up we go. Hi, Pete. I've been thinking about MST3K for several days now. Just yes.
White, bright, similar like the one. Can we just pass?
What things?
Time to time I see this white, bright scenes in the bottom. We got some
On the rocks?
They're very noticeable. I would show it to you once I see more.
Okay, I'm trying to zoom in if we can.
Not many of those, but I will tell you.
Okay, yeah. If you see one, just shout it out. Oh yeah, yeah. Well, we may see a lot of eels if we find a seep, potentially. Actually, every time I've seen the ROV use its arm, I think of that scene in 2001: A Space Odyssey, where Dave is blasting himself back into the ship using the manual airlock. It's one of my favorite scenes in a movie because it was filmed in 1968 and it was done using all practical effects. And it still looks amazing every time I see it. It's one of my favorite films. But yeah, also big MST3K vibes.
Yeah, Lauren, I'm definitely feeling that too. I actually wanted to really give a quick shout out to the SOI engineers. We had some pumping issues in the back with some of the stuff that my group is working on, and they fixed our pump.
Thank you, SOI engineers.
So much.
The SOI engineers are truly amazing people. Everybody who works for SOI is amazing. Seeing more of those sea fans that we sampled a moment ago. So it's very cool. A little bit of deep sea coral here for you. And we are slowly making our way up this hill. We are now at 2,515 meters, 2,515 meters. So we started out about 200 meters deeper than this, give or take. So we're making our way up this little hill, and then when we get to the top, we're going to decide whether we want to keep going up and over, or actually first I think we're going to check out the left, then we'll decide if we want to go up and over or if we want to go to the right. So we're just having a look around here. But as Hannah put it to me, it's like going on a night hike up a mountain with one flashlight. So fortunately, we have great bathymetry data to help guide us here. So can we zoom in on one of those little white things on the ground here? What do we see? It just looks like some debris, I think. Yeah. Okay. Keep going. Yeah, I don't think that looks like a broken bit of shell or something, maybe. Okay.
That's it.
I would keep going.
This hike is killing me.
It's very different from yesterday. We're
Yeah.
actually going up an incline rather than going around down like a flat bit of the bottom. This looks very different.
You think that's really fast? Really good, very fast.
This next one.
I am not evil. I use my powers for good. Mostly.
I took my headphones off for 1 second, and that's what I came back into, is I'm not evil.
I'm not! To quote Dr. Forrester, why? Because science, that's why. Ooh, look at that jellyfish. Can we get a quick zoom in on that? Over on the right? If possible. I don't think we've seen anything like that yet. Ah, there we go, yeah. Yeah, there's lots of brittle stars here, and a lot of them are just kind of like hiding under the surface. Ooh, look at that color. Very pretty. And now we get to see underneath. Doing a little turn, little spin, a little 360 spin, so we can appreciate how beautiful you are. Thank you. All right. Thanks. We keep moving. Yeah, just wanted to get a good shot of that because it's pretty. Doing it for the gram. Bathymetry is poetry. I really like that. That is so true. Get up a little bit past this dust. Schmutz. Okay. We're almost up to the point where this ridge is gonna flatten out, I think. And then we can start going along some of these fault lines we see. less marine snow here at this particular spot. Although that also could be the angle that the camera is pointed. Maybe some outcroppings here, a little bit of rock. This over here. An octopus or something? Or is it- No, I think that's another acorn worm. Never mind. Let's keep going. You can see we're starting to see some rock here sticking out from underneath the sediment, I think. So the rock here has been pretty brittle so far. Broke apart the last time we tried to sample some. There's another one of those green sea cucumbers that we got earlier in this dive. Crinoid, maybe. Oh, can we stop to get that crinoid, please? It's down here. Right there.
So when you see the thing, right? So they're
So you can pause the vessel?
Yeah, can we sample that? We could put it in the coral quiver.
Bridge, ROV, can you pause and move?
can we get a close up on it first?
Yeah, close up first,
Thank you.
if you please. We're collecting coral samples for the museum. So they have some samples. That one's very pretty.
Do you have an image? We're going to collect that. It's a crinoid. So I'm going to describe this as pre-collection image.
Alright, so just get a nice close-up shot of that beauty.
So that's
Thank you. Oh, wow. Look at that. It's like peacock feathers.
Does it burn? Oh, sure. Yeah. Okay. Uh-huh. Can you show me? Oh, yeah. Yeah. Okay. Can we quickly zoom in on this gray patch of sediment here? We just want to show that the sediment underneath of this, this sort of tan material on top appears to be different in color. It's like it's been turned over by something burrowing.
making a job.
This little mound there. That's all. Okay. Can we use the suction sampler for this one, actually, instead of the coral quiver?
Sure, which one did you use already?
One.
So you want to use 8 and 1?
Okay, so that's going in suction sampler 8, please.
I would find a few things.
That's just true.
Yes, sir. We use the sections of the try to do something. That's why. Then we move out, so the trash bag is this one, black trash bag.
Alright, so we're gonna take up this crinoid, this beautiful feathery crinoid, using our suction sampler. I love that we basically just have like a little vacuum hose on the ROV. I know it's more sophisticated than that, but all right. Yep.
Zip up just a little bit, in case it flies out like start losing. These things will fly pretty quick.
It may react.
Yeah.
It's not touching the
Start to suck it up. It might try to swim away. He looks very stationary. These can sucking up for science. Yeah. All right. And ooh. There we go. Okay. Nicely done. Thank you.
Can I get a confirmation on the suction sampler?
That was sampler 8.
Sampler 8.
Thank you.
So far back.
Okay, and we're going to continue forward. We can see more outcroppings of rock here as we reach closer to the top. Yeah, I like to imagine it makes the bank tube noise. The pneumatic tube. Boom. So some of you were not here when we tried to suck up a very large sea cucumber yesterday and it got stuck. It was a big chungus.
Lauren,
Yeah.
I think I saw some loose rocks over here. Do you think that we can grab it and put it in the rock?
Our geologist has requested that we
Yeah.
grab some rocks. Can you point out which ones you'd like? Come up here with the pointer. Yeah. Show us what you want.
Yeah. I see some of them that are really loose.
Down there by the section. Okay. Yeah. We could put those in a rock box. Can do.
It might be stuck in our hose, that guy. We'll leave it running out.
In fairness, I love to stop and pick up a good rock, and I'm not a geologist. You know, I'm just a rock enthusiast. A rock fan, if you will. Do you want it in the rock box?
Maybe
Or
it's
the
better
coral
to
quiver.
Put it in the coral quiver.
Coral quiver.
If it fits.
If it's small, it might not stay in the rock box. All right, let's put it in a coral quiver then. We're definitely going to see a lot more rock, I think, as we get up here, because the marine snow from the surface. We've used one, so let's use number 2.
Yeah,
Coral quiver 1 is full.
Yeah,
we have
coral quiver.
used the first one, the number 1.
Yes, we
So I
used
think
one for that fan
The
coral.
rest is
So the rest are empty. Just tell us which one you want to put it in. Number 3? There are absolutely geologists with stickers that say Geology rocks. Yeah, that's a thing. That's a thing that exists. We're definitely seeing a lot of marine snow today.
I think this mic is showing like mud. It might kind of fall apart when we're trying to grab it.
Oh yeah.
That's the one, right?
Yeah, that's the
Yes,
one.
that one,
Let's
yeah.
see if we can pick it up. These rocks have been pretty fragile and loose, so this might fall apart when we try to
Yeah.
grab it. We could always get the scoop.
It may be fragile, but
Yeah. Oh, there we go. Pretty good. So yeah, there's a ton of marine snow at this spot. I'm guessing it's because we are closer to the coastline.
that's
And there was a deeper oxygen minimum zone here as well.
you can make some
So there's a lot of productivity here judging by how deep the oxygen minimum zone went. It's really, really big. Yes. So that oxygen minimum zone is caused because all of that productivity is caused by upwelling. So upwelling is where a lot of cold, nutrient-rich seawater is transported from the bottom up to the surface, and that feeds phytoplankton and other primary producers in the surface ocean. And all of that productivity results in lots and lots of eating. And when things eat, they breathe oxygen and turn it into carbon dioxide. So they suck up a lot of oxygen out of the water, and that creates the huge oxygen minimum zone. So the presence of this really huge oxygen minimum zone would indicate that there's lots of productivity at the surface, and that's what's resulting in all of this marine snow, all this creation of organic matter that's sinking down to the bottom and creating these fluffy layers that we're seeing here. We're in an area of the world where there is a lot of upwelling. And so it's very, very productive. In some places, the oxygen minimum goes all the way to the bottom. Geology team, do you want any other rocks? Where? Oh, can we look at the bottom? Down by the where? Yeah, point it out to me, please.
And I got confirmed information that was in.
That was Coral Quiver 3.
Can you look a little bit ahead? So we're here.
Oh, okay. Yeah, let's head over to that rock and take a look at it.
And when everything settles,
Yep.
we may have a look, a closer look to this.
Yep.
But
Okay, yeah, we can take a look at those.
I don't expect to do many of those rocks.
Yes.
They keep going up.
Yeah. I expect we'll be seeing more and more rock the higher we get up this ridge, so.
Which one? The one that we
Pete's making puns in the chat, as I would expect him to. Who invited the English professor to the chat? It was me, I invited him. All right, let's take a look at this rock here.
Okay.
And also,
Can we zoom in on it? This little hole there of some kind. Limpet?
Let's collect that limpet.
Can we collect
Keyhole limpet.
keyhole limpet? Okay. All right. Keyhole limpet for Nick. What is the best way to collect that?
piece of it.
It's in a crevice.
Yeah. Can we maybe just like break off that whole rock and? Because I think otherwise it's going to be difficult to get. Yeah. Yeah, if we can get this in a rock box, as long as it won't fall apart. Oh, okay. Actually, can we put that in a bio box? Yeah. I don't know if there's a way to get... looks like it's going to break apart pretty easily. One of the rocks here seem to do that. Maybe. We have a pretty big bio box that would probably fit in. That's a terrible joke. I'm going to remember that one. If you don't have eyes on the chat, the joke is, where does a geologist keep their peridotite clock? On the mantle. But oh, yeah, that. Okay.
not sure that honestly this is going to be fruitful. I think it's just going to fall apart.
After this sample, I think we should just try to get up to the top of the ridge here. Unless we see something truly shocking.
I don't even know if it's becoming too difficult, we can abort.
Okay.
Okay, maybe one more try and that's it.
Okay. We're gonna try one more time, and if this is proving too challenging, we're just gonna drop this one because we're not sure if this is a live limpet or not. But if we can get it, we'll get it. And if not, we'll continue on our way. Yeah, this is... Yeah, these rocks are not... Yeah, like a clay thing. Right. Yeah. Okay. Yeah, let's keep going. Maybe we'll see more of them on something easier to sample.
Before we go, Lauren, can we get a closer look to where the rock were and the upper?
You want to zoom in on that rock again? Or further up?
Yeah. We get a closer look over here where so
Yep.
we can see the.
Zooming in here on some
Okay.
rocks of interest.
Perfect. Thank you.
Do you want to make that
Do you want to make an observational picture here?
Can you make like a
What do you want to call them?
yes, outcrop?
Okay.
Outcrop?
Yeah.
All right. I'm just going to make an observation here. Let me know when you're ready to go.
You can see that it was where the white rock were.
You ready?
Yeah,
Okay.
yeah, thank you.
Let's keep going. Thank you. And up the hill we go. Just a little bit further and we will reach the top of this ridge here. Okay. Oh, that's messed up, Pete. I'm not gonna repeat that. Okay, coming up on some more outcrops here. This little shelf of rock. And we are almost to our next waypoint.
Continuing move. Thank you.
Can we zoom in on those, oh, sorry, this area here? It's like a sea star, maybe. Or an anemone. Yeah, sea star.
The stuff is just, yeah.
Okay. Neat.
Starfish, yeah.
Yeah.
Four arms. Maybe one stuck inside.
Oh, yeah, maybe the other arm is where we can't see it.
Yeah.
Cool.
Aquí lo demás.
Okay.
A little bamboo coral.
Yep.
Starfish.
Okay.
Sitio.
Got this little ridge here. Seeing some more of these corals. Yes.
I would like a push core here anywhere.
A picture?
Push core.
A push core.
Yes.
Okay.
So carbonate is interesting.
Okay. Can we take a push core for microbiology, please?
Can we, I believe we wanted to reserve push cores 1 and 2 though, so.
Yes. The first two will be used for Aparna's work. So we will do push core 3, please. That?
Yes.
Oh, okay. Right on top of this urchin. Yes.
Is that number 3?
That would be number 3. Just gonna center it right over this urchin, I think.
Right next to this yellow thing?
Right on top of it.
Or you want to get the other thing?
Yes.
Well, it's pretty hard.
Okay. Well, we'll take as much as we can manage. Armando, are you,
Maybe a little.
is that good? I think we might have trouble getting a full core here.
Okay.
All right. We'll just take what we can. Yes? Good? Good. All right. Push core 3 for Armando, including Spiky Boy. Looks like a sea urchin of some kind. All right, and let's continue. It's Jax. You are the bonus spiky boy, Jax.
That's it for you.
Yep, let's keep going. It's swimmy boy. All right. Another one of those really outstanding purple sea cucumbers down there. It's so bright. If you're just tuning in, we are off the coast of the Atacama Desert, the driest and oldest desert in the world, in Chile. And we are climbing up this ridge in search of methane seeps. We're at the type of margin where methane seeps are relatively common. We have a marine plate sliding underneath of a continental plate, which causes the continental plate to buckle. Sediment piles around these buckled areas called anticlines, and methane from under the seafloor seeps up to the surface. So we're following along the bathymetry here that we have mapped. Several locations where it looks like there may be fault lines in search of bubbles, microbial mats, temperature changes, pockmarks, things that would indicate the presence of a methane seep. Methane seeps are not as hot as hydrothermal vents, so they're sometimes referred to as a type of cold seep, but they're not cold. They're still warmer than the surrounding seawater. They're just not as hot as a hydrothermal vent. And we may find mats of bacteria growing on the surface, using the methane as a source of energy and a source of carbon, and feeding little mini ecosystems. So we're keeping an eye out for bubbles and other telltale signs. We're almost up at the top of this little hill here. We've already sampled some coral. We took a push core. Hang on. Yes? What? Can we zoom in on this rock, please? Just below, down here? This one. Yeah. Definitely some cool rock here. Some tunicates, fan corals, maybe a sea pen down at the bottom there. Take it as an observation?
Yeah.
Okay. We're just gonna mark this as an observation here in our notes. This?
No, this is going to collapse.
What?
Maybe the suction will be the best.
What do you need this for?
Excuse me?
What do you need this for?
It is for the museum,
For the museum?
for the Chileans. Yes.
Okay.
I'm collecting for them. I'm not collecting for me.
Okay.
Yep.
Suction?
Yes.
Okay. Can we take a suction sample of this?
Sure. If I would think that last one we took got stuck in the hose.
Have you received that tube?
You hear me, Lauren?
Sorry?
We think that last one we took got stuck in the hose, FYI, so it might be clogged up, we'll give her a go.
Okay, cool. We may have a hose clog, but we will try. Also, can we take a sample of the rock as well? The rock itself? If it's possible. Yes, you are just in time for a collection. We're collecting one of these tunicates using our suction sampler. And then I think we're going to try to grab a piece of that rock that it's sitting on.
Trying to straighten myself out a little. So, I'm ready for it.
Okay.
Oh, the big guy came in. Shit.
You get in? Okay, good. Yeah, good. Perfect. All right, we're not clogged then. Cool. All right. I'm ready when you're ready.
Okay.
Yeah, I'm ready.
This one we're taking for the Chilean Museum. If we can get it in the sucker. Yeah, there is some really beautiful banding on this rock.
How we are trying.
Oh. It's going. Might be stuck.
You know?
Big chungus here. It's clogging up the...
If it gets stuck, can we spit it out in a bio box?
Can we spit it into a bio box? Or...
We could try to knock it in.
Okay.
Let's see if I can get it out.
Okay.
Kind of jammed in there now. Is that-
Okay.
Yeah, it's all
We're gonna try to put that in a bio box.
Yeah.
There it goes. There. Yep.
Alright.
All right. We're gonna put that in a bio box.
I'm still not sure it's gonna come out, but we'll try.
Well, we'll give it a shot.
I'm ready for the report.
Yeah.
It's fully Augustus glooped itself. Yeah, it's a very good way of putting it. Yes. I'm gonna get that tunicate into a bio box. Which one is it going into?
Alright, might take it on suction.
Yeah.
I've just sampled a tunicate, and we are going to dispense it into bio box 2A if it will come out of the vacuum, which we're not entirely confident that it will.
Do we need to 2A?
2A. Yeah, if we can get it out. I think it's stuck. Yeah, I don't know. Oh, maybe. It's falling out. It's coming. Nice. I was worried there for a sec. He turned his smile upside down. Okay, can we grab...
Rory, what is that thing that is stuck in bio box number 2?
I think it's an
That was
arm.
the trash can.
trash bag.
That metal arm there?
Oh,
That's
our
part of
temp-
the...
That's our temp probe.
Yeah. All right. Can we grab a piece of that rock, please?
I think it's actually in too. We can close that. That's just this fellow who fell. He's okay.
Oh.
Four chokes on it.
Yeah, he's on that.
Looks like we lost a lid to one of our push cores. It got knocked off, so we're gonna put that back on. Where's that at?
Does he go like that?
Yeah. Silver part.
Oh no, that's a separate thing. Okay,
Silver nozzle.
that's our temperature probe.
Alright, so that should be okay there?
See, you can come back a little. Yeah, right. Press it down.
Press it down?
Yeah. You see where the U clamp is? You need to come behind. Right
You
the U.
can back up a little bit.
Yeah. Yeah, it's on the right there.
Yeah, I'm not a big fan of this.
All right, we're just trying to get our temperature probe back in place. Okay,
Alright, so what are we doing then?
let's keep going. Or no, rock sample. Sorry,
Rock sample?
rock sample. Then we'll keep going.
Which suction sample was that we just used?
Oh, we put it in bio box 2A.
Oh, sure, that's right.
Let's have it. This.
Alright, you just want to try and break a piece of this off?
Yeah, let's take a chunk of it, put it in a rock box, please. If it's really fragile, we can put it in a coral quiver, but this looks a little bit more solid than the other stuff we were looking at.
Let me see if I can break it. So it's pretty tough.
Yeah. Just try to break off a hunk of this for the geo folks. Oh, that is deceptively crumbly underneath. Strange. Maybe we can break off a bunch of chunks of it and then get it with the scoop into a coral quiver.
Keep breaking it?
Yeah, get a little bit more maybe.
Alright, then left for a scoop, you reckon?
I think so.
Okay.
Thank you.
And where's that scoop gonna go?
Can we
Back
put-
rock box?
Put it in a coral quiver, I think.
Coral quiver?
Yes.
open a quiver real quick first. So,
2?
Coral quiver 2.
Lots of fluffy stuff in there. I think we should have a good amount of rock. Alright, so that's going in coral quiver 2. Coral quiver 2. Nice.
okay? Happy with that?
Yep. Looks good to me. Yes.
Hold on, let me see if I can zoom in. I saw two things that look like brachiopods, but right now I lost it. Yeah, but I cannot see them.
Zoom in on the rock
Yeah,
we were just on.
I lost them.
Okay. Can we zoom back in on that rock, please?
They were right there on the upper, just behind the arm.
Here.
Yes, those two.
Okay, can we zoom in right there?
Yeah, those two.
Yeah. Over here. This area. Are those brachiopods? No. Those look like sponges.
That's right. You're correct.
Yeah.
Yep.
Okay. That's a sponge. Okay. Let's keep moving then. Onward and upward. We're gonna swap out dive leads. So I'm gonna take a quick break and I'll be back in the room in a little bit. Thanks so much.
When you come back, present the banner.
Present a what?
When you come back, introduce the banner.
All right. Thank you so much to Dr. Lauren for the narration. She'll be back soon. Thank you.
So hello everyone online and the people in the room. I will be doing the next shift of narrating this dive, a very different dive from tomorrow. We are going to do more exploratory dive, looking for some seeps, you know, some fluids that come from underground and are usually hot, although we are also looking for cold seeps that even form some kind of ice. That is interesting because it has lots of methane and have unique microbial and other organisms habitats. So for the people online and also the people in the room, we are looking for structures, linear structures, any kind of faults or other, you know, geological structure that may have these leaks of fluids or other kind of, you know, gases even. So even when we see any bubbles or this kind of stuff, we will try to stop there and to see what's going on. So like yesterday, I will try to change in between English and Spanish. So if someone watching Spanish speakers, they can follow. So hola a todo el mundo, soy Antonio Molina, soy científico planetario y estoy haciendo ahora el cambio de turno para narrar esta travesía submarina. Esta inmersión va a ser un poco diferente de la que tuvimos ayer. Estamos buscando seeps, básicamente son emisiones de fluidos que vienen del subsuelo, tienen un origen volcánico y ya que estamos en la zona de subducción es posible que encontremos esos fluidos que están asociados a tanto a diferente vida microbiana como también otro tipo de organismos. Así que tanto a la gente que está conectada online como a la gente que está en la habitación y bueno, el resto del equipo que estamos por todo el Falcor 2, vamos a buscar por esta pequeña inmersión, unas 2 horas creo que estaré con vosotros, tanto fracturas, afloramientos de rocas con alineamientos que den sensación de que por ahí puedan emerger esos fluidos o también burbujas o otro tipo de cambios. Entonces, si cualquiera, tanto en el chat como dentro de nuestro equipo ve cualquiera de estas señales, pues iremos allá a ver si podemos encontrar esos ecosistemas tan particulares. So now we are going through a fault system that is just to the south of the Mejillones Peninsula, and it's like a huge structure with different types of faults conjugated. That means that they are in perpendicular fractures, and they were formed because of the subduction of 2 plates. As you may know, the Earth's surface is formed by many different plates, and some of them are oceanic plates, other are continental plates. So we are now when one oceanic plate is meeting a continental plate and is going below it, and that forms different winkles and other kind of tectonic features, and that's why we are expecting to find these seeps. Como estaba diciendo, nos encontramos en donde se encuentra la unión entre la corteza oceánica y la corteza continental, la placa de Nazca y la placa subamericana, sí. Y en este caso, pues lo que vemos es como una de las placas, la placa oceánica, pasa por debajo de esta placa continental, formando una serie de formaciones tectónicas interesantes, que son este sistema de fallas que estamos recorriendo, y debido al cual esperamos encontrar estas surgencias de fluidos de origen subterráneo. Así que estamos subiendo por este sistema de crestas. Bueno, no sé si llamarlo crestas, básicamente son unas elevaciones en la batimetría, es decir, en las elevaciones que encontramos bajo del océano. So we're looking for, through this elevation, it's like a very narrow mountain that it's right in the bottom of the ocean here. And when going to try those fractures, bubbles, rocky outcrops. Me alegra mucho que parte de nuestra audiencia pueda disfrutar de esta experiencia. Bueno,
Try to get the stuck crinoid for the museum.
Okay, so maybe we can stop a little bit now. Do you mean, Nick? Do you mean this one?
Yes.
Okay.
Is that a Yeti crab way up there? Oop, I see the white.
These are
So I eyed up in the, where's your,
Here?
the
Yeah.
starfish?
Okay. Thank you.
You wanna go left? You wanna go left?
Estamos parando ahora mismo porque hemos encontrado este organismo que puede tener interés para nuestros compañeros estudiando la biología de esta zona del fondo marino. So we're just stopping by just to see. We are going to sample some of the organisms that we just found for the people interested in this organism living in this
So you're gonna wanna sample this or?
bottom of this ocean here. Estamos a unos 2,5 kilómetros de profundidad. We are about 2.5 kilometers deep, a little bit shallower than yesterday's dive.
You can go to the small biobox because the stack is got bent.
Okay, so which
What did we have before?
we just had the cucumber, so
It's not gonna fly
Are we
anywhere.
gonna cut him or are we gonna try and get the whole thing?
If you get a good grip, it's easy to take it out.
Try and get the whole thing?
Yeah.
and we may try to store it in the bio box number 1 in one of the 3 remaining boxes.
1B.
Okay, 1B, sounds good.
I think this guy will not fly away, right?
No,
He's
it's stuck to the bottom. Yeah.
Firebox 1B?
Yeah. What's the name of the organism? Echinodermata?
Crinoid.
Crinoid? Okay, so this is a crinoid. And we are retrieving that for sampling.
Down to your sill whittle. Watch out if you need to, rack in. Say 1B?
Yeah,
Yes.
bio box 1B.
You don't wanna use one of the big ones? I don't know how big this guy's gonna fit in there.
Do you rather to have it in a large?
The stock will bend. Will bend completely. The stock will bend.
So it's
It's fine.
flexible enough
It's
to
very flexible.
to fit in there, I think. Maybe it needs a little bit of a push, but
Yes.
Okay, thank you.
Okay. Thank you.
Perfect, thank you.
Okay, so we have this specimen collected. We are going to keep moving forward. So I have here next to me Aparna.
Hi.
It's a college that is working, studying the microorganisms that live in these areas. And I don't know, Aparna, if you want to talk a little bit about this.
Okay, so to the audience, I was yesterday also describing a little to the 3-kilometer deep site that we had yesterday, and we have collected some nice samples of biological organisms. Some nice rock samples and microbial sediments. So here we have colleagues from CSIC in Spain. We have colleagues from USA, Uruguay, and we are from Chile. And our goal of this project is to have the trace of the ancient life forms in the depth of Atacama Trench. So as my colleague Dr. Antonio told, our goal is kind of understanding how during the subduction of Nazca Plate and South American Plate this area millions of years ago formed, and if we can get some biosignatures of, and here we can see some nice.
You can do a glance. Looks like a lobster. Sorry, Aparna.
Yes. So we are seeing a nice lobster here. If we can,
I mean, we are not taking a sample, but
if we can have a zoom.
yeah, just a closer look. But just if it goes away, it's okay. I think
Okay.
it's good. It's swimming.
It wants to hide.
Okay, maybe now we can just take a closer look at it. About it. So, Aparna, I was reading. I mean, a colleague of mine told me that she found that usually when you are looking for these seeps, a signal that we are close by are that you find how you say that, crabs, bivalvi, and star seas. So this could be a good, a good news.
Yes, that we can find it nearby somewhere.
So Aparna, also, even though you are from India, right?
Yes.
You speak Spanish, so you can give us some Spanish little talk about what you just said.
Okay, so para la audiencia, ya ayer estaba también discutiendo sobre un poquito cómo nos muestreamos en los sitios de casi 3 kilómetros de profundidad del fosa de Atacama. Bueno, nosotros somos un grupo desde España, de Chile, de Estados Unidos, Uruguay, de distintos continentes, y aquí estamos buscando la vida antigua de fosa de Atacama. Y somos un grupo de microbiólogos, geólogos, biólogos, y aquí podemos ver un crustacean, y como mi colega mencionó que cuando podemos ver algún lobster o tipo echinoderm, hay posibilidad que podemos encontrar al lado algún punto de metanos o burbuja de metanos.
So Aparna, we are going to try to look around in this area
Yes.
because as we just found this lobster
Yes.
is made an indicative
Yes.
because also we are looking not just for these seeps but also microbial mats, right?
Yes, exactly.
Para la gente en español vamos a dar una pequeña vuelta por los alrededores porque al encontrar esta langosta es un indicativo de que puede que haya tanto coal seeps como coal seeps, no, perdonad, seeps de metano o otro tipo de fluidos, así que vamos a estar recorriendo los alrededores un poco porque no solamente buscamos estos efluvios de material, sino que también estamos buscando tapetes microbianos, ¿no es así?
Sí,
Aparna?
sí. Queremos buscar los tapetes microbianos porque probablemente es una indicación también que durante todo este millones de años cómo los microorganismos están sobreviviendo y adaptando en este tipo de clima extremo. Maybe we can go and look.
We can continue, but not maybe straight ahead, but maybe looking to the whole side, so we just in case we see something with the instruments. So same direction, but not just straight ahead, right? Okay, so
Is that what you want there? Is that what you want?
yeah, perfect. Thank you. So, I just been told that maybe we can go back to where the lobster were, so I don't know exactly we have any indication on where we were, but that was closer.
Bridge.
And just to
Copy that. Really good pause.
look around. Disculpad la interrupción, vamos a volver a la zona en la que estaban esas langostas. So we're going back with the lobster were, just to trying to look if they are actually a signal of those different conditions. So which kind of condition we are talking about, Aparna?
We are basically trying to find some gas bubbles that can be some possible exploration of methane or some geothermal fluids. Queremos buscar algún seep de metano o algún fluido geothermal que normalmente podemos encontrar en zonas de subducción.
¿Y por qué estos fluidos son tan interesantes? ¿Es porque contienen esos químicos que sirven a los organismos para alimentarse, ¿no? It's
Sí.
because they have all the chemicals and components and also physical conditions that are good for the microorganisms and other organisms, right?
And also the floating particles, we can see some marine snow. So that's also an indication that how much organic decomposition and organic life we can have in this kind of depth of ocean.
So let's see, fingers crossed.
Yes.
Cruzaremos los dedos para ver si encontramos algo por la zona. Aquí podemos ver un pez tranquilamente cerca del fondo. We are seeing this fish swimming by. We are losing signal, I think. Sorry for the fleeting image. Perdonad por que de vez en cuando perdamos la señal de vídeo. De nuevo estamos explorando la costa de Chile. We are exploring the Chilean coast, close to the subduction area, cerca de la zona de subducción. Looking for living fossils, buscando estos fósiles vivientes, esos organismos que creíamos extintos, pero que puede que sigan vivos en estas zonas que han sido prácticamente inexploradas. try. Even I think it's
Maybe we can look into the floor
That's
more.
where it's at, if you get the stock Crinoid.
yeah. That's you're right.
So just to the left would have been where we saw the spot.
Mm-hmm. So maybe let's keep going, but instead of the same direction that we were, little bit maybe to the right, and keep going up. So again, if anyone see any changes, sudden changes in the parameters, the temperature, the salinity, or do you see any white bubble? I mean, some bubbles or some changes in the material, some fractures, let us know. Si cualquiera ve un indicador como pueden ser esas burbujas, pero también cambios en el material, fracturas, y en los parámetros físicos que estamos monitorizando, la temperatura, la salinidad, por favor, hacérnoslo saber para que paremos. Y si buscamos por esas tapetes microbianos, Aparna, ¿qué es lo que estamos buscando exactamente en las cámaras?
Bueno, desde las cámaras podemos ver algunos tapetes de distintos colores, puede ser verde, amarillo, naranja, marrón, y es una indicación de tapete microbiano, puede ser de multiespecies o multigenus, que alguno encontremos ayer. Normalmente para formar tapete microbiano necesitamos encontrar algunas rocas porque el superficie duro es importante.
Sorry, Aparna.
So basically what we are trying to see here also, we are kind of searching for some colored microbial mats that can be, if they are of cyanobacteria, can be of green, or if that is multi-genus or multi-species, can be some orangish yellow or maroon that we have found yesterday. But for formation of microbial mats, it's quite important that the microbes get a surface to get attached. Here we are not seeing that much amount of rocks, so probably a rock surface is a good indication that the microbial mats remain there unchanged for years. So we are yet to get those looks today. We have not found it till now today. But if we get some methane seeps nearby, it's very possible that we can find that kind of structures also.
Let's keep looking. Yeah.
There is a sea cucumber lying there.
Yeah, the pinkies, oranges, squeezy thing. A lo largo del día de hoy hemos encontrado muchas anémonas, pepinos de mar. Por ejemplo, ahora mismo estamos viendo una especie de gusano marino, todos estos organismos como con forma de cruz morados. Hemos visto erizos de mar, como el que estamos viendo ahí, esos, ¿cómo se llaman, Aparna? Son estrellas de mar, pero como más flexibles, ¿no?
Echinoderms.
Eso es, equinodermos. So, today we look to some... also, y también muchas... ¿Cómo se llaman en español? Anguilas. Varias anguilas. So, today we were able to see many cucumbers, sea cucumbers, anemone, sea urchins, and also this kind of worms that are those cross-shaped purple critters that we see almost everywhere. And the thing is, as we get closer to rocks, outcrops, they may have their more diversity, right, Aparna?
Yes, even the rock outcrops are, or the fault rocks can have possible exposition of methane gas also. So if we can find some rocks, maybe we can have a closer look to that. Ah, okay.
Okay, we will try to speak louder. Vamos a intentar hablar un poco más alto para que nos podáis escuchar mejor. También en la habitación a veces ocurren discusiones y comentarios porque todos estamos atentos de encontrar lo que nos interese. Entonces, estamos intentando mantener el tono relajado para esto. Al final es una inmersión muy larga, son muchas horas. So we are going to talk louder, so even if some other people is speaking in the room, you can hear us. You know, some people that's just looking for different things. So we may have little conversation around. And that's something on there. I don't know if it's, what is that? Probably.
Can we have some?
Maybe trash, garbage. Yeah. Anthropogenic. So hemos visto una bolsa de basura. Y continuamos. Adelante. Un pequeño pez.
Can we go and see the trash and we can make an anthropogenic entry probably?
No, it's okay. You guys can put that later and
Okay.
So sometimes these garbage, for example, before we took a plastic bag, it's not just because also we want to clean the bottom of the ocean because of all those trash that we found, but also you can usually find in there some organisms associated to those, for example, plastic eating bacteria. Isn't that right, Aparna? So I think hemos visto otros restos de basura, como por ejemplo antes, hoy, yo no sé si estabas tú aquí, Aparna, y lo has podido ver, pero
No.
hemos tomado una bolsa de plástico y no solo también para limpiar el fondo marino, sino porque podemos encontrar a veces en estas bolsas bacterias, por ejemplo, que comen plástico, otros tipos de microorganismos, ¿no es así?
Sí.
So we keep looking for something like fractures, bubbles, all these kind of different geological manifestations, let's say. Estamos buscando por esas burbujas, esos materiales, esas manifestaciones geológicas que nos puedan indicar la presencia de estos seeps.
bridge. Yes, we are. Sure. Doing 0.3 at the moment. What would you like to do?
Let's try 0.4.
0.4. Thank you.
So not just these geological features, but also we are looking for crabs or lobsters or starfish or even I don't remember which was the other indicators of different conditions that may help us to find these seepage, these seeps. Estamos no solamente buscando estos cambios geológicos, sino también organismos que pueden indicar que han cambiado las condiciones ambientales cerca, y como pueden ser pues cangrejos, una langosta como la que hemos visto antes, estrellas de mar. Esta zona está completamente cubierta por sedimentos. Podéis ver como pequeños montículos y algunos agujeros. Probablemente todos sean creados por organismos biológicos, como pueden ser estos pepinos de mar, que lo que hacen es comer parte de estos sedimentos y filtrarlos. Se quedan con la parte orgánica y van dejando un hilito de esos sedimentos. So we are looking at all those mounds and holes that are mostly generated by organisms like the sea cucumbers that we just passed one orangey pinky one, and they filter the sediments, they dig into the sediments.
We
and
can see some rocks nearby.
Yeah. We're starting to see some rocks. Estamos empezando a ver algunos afloramientos de roca, pero poco.
There is a Crinoid.
Sea urchin.
Sea urchin.
Yes.
So we are diving helped by bathymetry that is a little bit coarse, but we are hoping to find across these faults some seepage. We are around 2.5 kilometers deep. Estamos como a 2,5 kilómetros de profundidad, y siguiendo la batimetría, es decir, la topografía del fondo que hemos podido obtener estos días, estamos intentando buscar esas fallas que pueden ser por donde se escapen los fluidos.
Aquí podemos ver algunas rocas también. No sé si podemos dar una vista más cerca.
I think it's okay unless we see something, because it's similar. I think apparently you didn't, you wasn't able to look to previous outcrops similar to these ones that we just seen. Hemos encontrado unos afloramientos parecidos antes, incluso hemos tomado algunas muestras de roca, y estos, por desgracia, no parecen muy distintos. So these are not quite different from the ones that we just passed by some minutes ago. Let's keep going. Este pepino de mar tiene, no sé si lo viste, pero tomamos una muestra de uno muy similar antes a la no sound. Aquí vemos otro pepino de mar, another sea cucumber. So, what we are diving, this is not very different from where we are exploring another planets. Esto no es muy distinto de cuando exploramos otros planetas. The technology of the SuBastian is top notch. We have a ROV quite impressive from the technological point of view, and we're able to go through this bottom of the ocean, observe, take sample. Creo que no me escucha. Sí. I think there is no sound.
We're pretty far ahead of the boat.
Oh. So we're still diving close to the coast of Chile, and I was saying that this is not very different from exploring other planets. Estaba comentando como esto no es diferente de explorar otros planetas. Oh, así. Sea star, yeah, over there. Una estrella de mar. Justo hemos pasado por ahí.
You're going downhill. Roger.
Okay. Who is this?
So we just have a change of seats on the people working, the engineers working, piloting the SuBastian, the ROV. Acabamos de hacer el cambio del personal que maneja el ROV SuBastian. Y como comentaba, esto es una tecnología muy avanzada que nos permite llegar a estas profundidades, observar, tomar muestras, y no deja de ser bastante similar a lo que hacemos en exploración espacial cuando llevamos un vehículo como puede ser el rover Curiosity o el Perseverance, o por ejemplo los que esperamos poder hacer en las lunas heladas de Europa, o Encelado, Ganímedes, Titán. La mayor diferencia es que al final aquí, si we just saw some garbage, o un poco de basura en el fondo.
You collect it?
Do you want to collect the garbage? No, I think we need to keep going. Okay, maybe we can have a closer look to it.
Roger.
An observation. So for
Go ahead. Call Tyler. Go ahead and zoom in.
So it's incredible how we can find trash everywhere on Earth, even here, and how life still use it to shelter sometimes. Or es increíble cómo podemos encontrar esta basura por todas partes del planeta y la vida las utiliza como hogar, ¿no? ¿No es así, Aparna?
Sí, podemos ver que también estamos utilizando tipo un substrato para adherir a una superficie y es impresionante. En realidad somos humanos, a veces somos poco cuidadosos para la planeta.
Sí. So for the people that maybe just join us in the stream, and also for the crew in here, now we are going up this fault system. Trying to find some faults, linear features, rock, outcrops, or whatever that may indicate the presence of seeps or fluids coming from underground. So if we see any bubble, any change on the color of the material, even there are some specific species that are related to those seepages. So crabs, lobsters, star seeds, sea stars, and bivalves. Estaba comentando que para la gente que se haya unido recientemente, estamos atravesando el sistema de fallas que hay al sur de la península de Mejillones, en la costa de Chile, buscando fracturas o otros burbujas, otros elementos geológicos que nos indiquen que puede que haya emanaciones de fluidos subterráneos con un gran interés biológico y que también a veces están asociados a la presencia de otros organismos. Así que también buscamos cangrejos, bivalvos, incluso estrellas de mar. Ahora mismo hemos dejado de subir por este relieve tectónico y estamos siguiendo uno de los alineamientos que hemos podido ver en la batimetría del fondo. So we just stop going up these faults. These tectonic structures. And we're just following one of those faults. So over there, I think we may have a tsunami, maybe not just stop and just a tsunami. So this is one of the, I don't know if it's a crab or a lobster, no estoy seguro si es un cangrejo o una langosta, pero es uno de estos indicadores.
No, squat lobster. Other organism. Squat lobster.
It seems like a lobster, right? Guillermo, ¿es una langosta, sí?
Squat lobster.
Lobster.
Squat lobster.
Squat lobster. Squat lobster.
It's possible to take it.
I don't think so. I will try to keep going, but maybe another time.
Go.
He just went, yeah. So we're going to continue, but this is a good signal that we may find some changes. And looking all the time at the temperature, the salinity, all those parameters that may tell us that there are changes around. Estamos buscando en los parámetros que puede detectar el ROV, el SuBastian, tanto la temperatura como la salinidad, y que pueden ser indicadores de cambios en el ambiente, esos cambios que estamos buscando. Y efectivamente hoy estamos dedicando esta inmersión a la búsqueda de estas emanaciones de fluidos, pero en los próximos días intentaremos ir a lugares más profundos. Actualmente estamos en 2,5 kilómetros, casi exactos, pero SuBastian tiene la capacidad de llegar hasta los 4,5 kilómetros. So today we're just exploring around these fault systems, looking for seepage, seeps, these fluids. But on the next days we will go deeper, as we are today just at 2.5 kilometers deep. It's not just, it's quite a depth altitude, but we will be able to reach even 4.5 kilometers deep. I'm looking here to the bathymetry, and the line that we are following. We are just in a depressed area, following a fault. But as you can see, mostly we can find our sediments. So estamos siguiendo la batimetría, y ahora mismo estamos descendiendo un poco, siguiendo una de las fallas paralelas a la falla que hay al sur de la península de Mejillones. A ver si encontramos alguno de estos emanaciones. Follow the same direction, but the little bit more to the right. To the right?
All right, or right or left?
The one that we are facing.
Yeah, right that way.
Do you mind if I say your names? So we have here Benjamin and Taylor as pilots. Benjamin y Taylor son nuestros pilotos, los que están manejando SuBastian, tanto la navegación como el uso de los brazos mecánicos y todos los elementos para muestrear. So they are maneuvering the SuBastian, both piloting but also using all these two amazing mechanical arms.
Try to see more rocky material instead of this
You want to take a closer look? Yeah. So we are going to zoom in to see a closer look to this nice creature. Vamos a tomar una observación más cercana de esta criatura.
Sí.
So this is another sea cucumber, but we are see it just swimming around. Es otro pepino de mar, pero podemos verle en lugar de posado en la superficie nadando. So for the people online, can you hear me clearly if I speak in this volume, or do you rather me to speak louder? Para la gente que está conectada online, ¿me podéis escuchar bien? ¿Lo suficientemente alto? Okay, perfect. So somos un grupo de científicos de diferentes instituciones. Estamos gente desde España, Chile, Estados Unidos, incluso Aparna, ¿verdad?
De Chile.
Vienes desde la Universidad de Chile, exacto.
Bueno, somos parte de 3 universidades de Chile, Universidad Autónoma de Chile, Universidad de Tarapacá y Universidad de Arturo Prat. Tenemos grupo de investigadores quien trabaja en microbiología, de museo, así que tenemos intereses variados para entender este tipo de ecosistemas desde distinto punto de vista, o más un punto de vista interdisciplinar.
Eso es, exacto. Desde España también
Sí.
tenemos gente, mayor parte desde el Centro de Astrobiología, aunque trabajando en diferentes intereses, pero también de otras organizaciones. Y desde Estados Unidos tenemos gente desde Florida Tech hasta, no recuerdo cuál es la organización de Loren, disculpadme, pero de varios institutos y muchos con interés de los estudios astrobiológicos.
There we can see another sea cucumber.
Yeah, they are swimming around.
Yes.
Un montón de pepinos de mar. Lots of them. So we were saying that we came from many different institutions, many different interests, but most of us works in the field of astrobiology. So we are trying to look for, not just looking for light outside the Earth, but also to know which are the limit of light on Earth itself, and what are the conditions that the leaf needs and how they adapt to different conditions, in this case to the bottom of the sea, or eating, for example, the the material that this seepage has, also extreme temperatures. Como decíamos, lo que tenemos en común todos los investigadores implicados en esta campaña es que en mayor o menor medida estamos implicados en estudios astrobiológicos, que no se trata únicamente de estudiar la vida en otros planetas, sino también entender la vida en la Tierra, cuáles son sus límites, su distribución y cómo se adaptan a las condiciones extremas de temperatura, o cómo pueden alimentarse en zonas hidrotermales como la que puede que encontremos hoy, siguiendo estos sistemas de fallas. I think we can really right, yes, going the line, but same Okay, can we see a closer look of those? So that's a nice crustacean there. I don't know if it counts as the crabs that are maybe indicative of this seepage, but it's good to see something different. Hemos visto esta especie de, supongo que cangrejo, que creo que tiene una anémona. I don't know. Y quizá es otro de los indicadores de esa posible cambios en las condiciones que encontramos al buscar estas zonas hidrotermales.
Es impresionante.
Y gracias a todos los que confirmaron que se me escucha bien. Thanks to everyone checking in on sound. That's a great specimen. So, can we take this sample? Vamos a intentar obtener esta. Vamos a intentar aspirar este espécimen. Que parece especialmente interesante.
Antonio, at where we are going to put it? In biobox, coral quiver, or suction sampler?
I think it's coral quiver 7. No. Suction sucker 7, sorry.
Okay, suction sampler 7.
Yes.
Okay.
Sí. Siempre el obtener estas muestras es un proceso delicado. Estamos trabajando de nuevo a 2,5 kilómetros de profundidad, maniobrando estos brazos mecánicos. Es un proceso complicado, además de tratar con criaturas que también son bastante delicadas. So we are always this sampling. It's a little bit delicate, not just from the technological. Okay. So we're trying to take it from the backside just for to get another. Yeah. Thank you. this process is always delicate because not just it's difficult to maneuver with all this. Run away.
Very fast.
Yeah.
Is pretty intelligent crab.
Se escapó.
Se escapó.
He ran away.
They came down in time to see that crab. No, 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 no.
Okay, we're fine. No worries. Can we keep going? Okay, so we are going to try to take it. As I said, it's a very, very, very delicate matter because, of course, they don't want to get into the tube. Por supuesto, va a hacer lo posible por evitarlo, pero podemos obtener mucha información de un ejemplar de estas características. Super difficult to move and take it. Let's see. Es muy complicado moverse de forma delicada con SuBastian. Es un vehículo bastante grande, más grande que un auto, que un coche particular. Podéis mirar las especificaciones del SuBastian en la página de Schmidt Ocean Institute, so you can look at the specification of this ROV SuBastian in the Schmidt Ocean Institute webpage. How amazing it is. So let's see if we are lucky. And if not, we may need to let it go. Okay. Okay, it got in there.
Suction sampler 7.
Thank you so much. So, Benjamin and Taylor, great work. Thank you. And let's keep going. Después de esto, vamos a continuar la búsqueda, un poco de exploración después de esta pequeña parada. Let's continue exploring after this short stop. Successfully retrieving this nice specimen. again, we are looking for fractures, we are looking for linear structures, different kind of materials outcropping, and of course this kind of specimens that indicates possible hydrothermal activity nearby. Estamos buscando tanto esos lineamientos tectónicos, otras estructuras geológicas que puedan indicar esa posibilidad de que encontremos estas emanaciones hidrotermales, pero también organismos como esa langosta que pueden ser indicadores de esta presencia. So now the waypoint goes there and then goes right right? Yes. So whatever is easier for you, you can either go directly to the next waypoint or we do like a curve instead of doing the full, instead of doing the full thing, we can either go whatever is easier for you. Yeah, we don't need to reach the next waypoint. Yeah, yes. Something like that. So we change a little bit our course in case we are able to see more things. Hemos cambiado un poco el curso a ver si encontramos algunas cosas interesantes, aunque acabamos de ver hace unos momentos otro cangrejo blanco. We just see this white crab laying around. Just we need a little bit of patience. We need to be patient, so un poco de paciencia. Pero al final esto es lo que es la exploración. No sabremos lo que buscamos hasta que lo encontremos. We won't know where is what we are looking for until we found it.
Also from astrobiological point of view, we are like searching the depth of ocean this part of ocean that have not been explored before. So it's also a point that how the life have been millions of years ago in our planet.
Mm-hmm.
So we can definitely see some ancient forms of life here. We can have some interrelationship drawing between how the life can have been in other planets millions of years ago.
That's it. Encontramos criaturas que son realmente antiguas, que tienen un origen muy antiguo y que nos pueden servir para entender mejor cómo podría ser la vida en otros planetas. O sea que este es el interés astrobiológico, estudiar esta zona que ha sido muy poco estudiada previamente. Ha sido, incluso algunas zonas no han podido verse con la tecnología que tenemos en el SuBastian. Antes comentaba Parna como no es tan distinto hacer este tipo de exploraciones aquí como las misiones que, por ejemplo, hemos llevado a Marte, donde van los rovers por la superficie del planeta, sin saber muy bien lo que vamos a encontrar hasta que no estamos allí. So can we get a closer look to this ray? How you call that on English? Do you know, Aparna?
I'm asking my colleague.
Yeah.
It's a skate.
Ok. Una raya en español, ¿verdad? Manta raya. O solo raya.
And there's a nice sea cucumber lying beside.
So the difference between this Martian or out of Earth, space exploration is that now we are able to do it in real time. That is quite convenient. Otherwise, people doing space exploration, you just need to. I think it's a fish. This one? You want to take a closer look? I think it's just an eel. Yeah. So when we work in another planets, we need to plan ahead all the things that we are doing now without knowing what we are going to find. So cuando estudiamos en otros planetas hay que hacerlo con ese delay que no permite poder hacerlo en tiempo real como aquí, sino hay que planear con tiempo suficiente sin saber lo que realmente vamos a encontrar. Aquí, gracias, que podemos hacer una exploración bastante rápida de un área bastante completa. Esto en una misión espacial podría tomar meses, si no años. Pero quizá en el futuro estaremos viendo los mares de Europa o de Encelado similar, como lo estamos viendo ahora. El fondo de las costas de Chile. In here it seems like the surface is a little bit more compact instead of the loose sediments that we just passed before. Parece que aquí el material es un poco más compacto que antes. Se ven algunas rocas, ¿verdad, Parma?
Yes.
But I still don't see any microbial mat.
No.
No parece que se pueda apreciar ningún tapete microbiano.
So we found different kind of rocks in the last day. So here the formation also is different. So
Yes.
I think it's less possible that we get some mats here because the sediment looks very loose.
So if anyone find a colorful stain, right, or something like that in the image, let us know. Si alguien ve una mancha de material con un color diferente en la imagen, pues hacérnoslo saber. Pueden ser verdes, ocres, oscuras, ¿no, Aparna?
Exactly. There should be something like some orange, yellowish, maroon, different colored stains that we can found probably with an indication of microbial mat formation.
Hay unos tubos de gusano que quisiera ver de cerca porque tengo una sospecha.
Avísame cuando haya y los...
So we are looking for maybe wormholes that may be interesting, even just to zoom in and to look them closer. Estamos también buscando quizá la posibilidad de que algunos de esos agujeros de gusano que pueden ser interesantes de observar de más cerca. Por supuesto que en el futuro vamos a poder utilizar un montón de diferentes técnicas, como puede ser la inteligencia artificial, para tratar todos estos datos. So AI will make a great tool in the future, probably, to recognizing or analyze large amounts of data in the future, both for space exploration but also underwater exploration. I mean, the analytical possibilities are huge. Y encontramos que este fondo marino es, quizá no tanto en esta parte, cuando encontremos afloramientos rocosos seguramente veamos que hay una riqueza de vida todavía más impresionante, porque las costas de Chile son especialmente ricas. ¿No es así, Aparna?
Sí, más que probablemente es el tercer expedición a Fosa de Atacama, pero esta zona que estamos buscando ahora, o estamos explorando ahora, esta zona no fue anteriormente explorado. Así que cualquier cosa que podemos ver es algo nuevo para nosotros y también para las gentes quien está mirando ahora en YouTube.
Sí, así tanto como en Facebook y Twitch, creo.
Yes, yes.
Nos está mirando directamente. Para él somos un alien, al final. So we are an alien for it.
It's a swimming sea cucumber again.
Yeah. But for him, we are aliens.
Yes.
Not one of their, colleagues around.
¿Qué? Es mi esposo.
So we just said the surface is getting a little bit more rocky, but still sediments, not any fracture or alignment to be seen close by. Let's keep looking. Seguimos viendo esta superficie un poco más rocosa que la que encontrábamos hace unos minutos, pero todavía nada que indique esos alineamientos que buscamos. Vamos despacio, atravesando este fondo marino. Actualmente estamos a un poco menos de 2,5 kilómetros, 2.498 metros, so we are a little bit less than 2.5 kilometers deep, 2,498 meters. You want to take a closer look? It's okay, we can keep going. Yeah.
I just want to take an opportunity to say hello to my husband also. He's seeing from Chile live. And I have 3 cats also. So hi from me
Hi.
to them.
Thank you for following this sitting drive with us. Can we get a closer look to the purple? I think we did a cancer, right? Can we get a closer look?
That one, yes.
To the rock.
To the rock.
Oh, to the rock? Which one? Can you
That rock. That rock. This one. Yes.
So we may. Vamos a mirar más de cerca esa roca, a ver cómo es su aspecto. We are getting a closer look to this rock, just to see its texture.
Do you want some geological observation also, Antonio?
Yes, please. Yes. No, I think it's okay. We can keep going. Yeah.
So it's a carbonate structure.
It looks like that, right? With all those holes. It's probably not very hard, but I think we have plenty of those.
Yes.
We just want to keep going. Sí, parece una estructura similar a los materiales que hemos encontrado antes. Hoy se trata de continuar a ver si encontramos algo característico de estas zonas hidrotermales.
There is some interesting fish.
Do you want to get a closer look to this? Parece una especie de anguila. It seems like an eel of some kind.
It's kind of eel, yes. It's an eel?
It's in shape.
Okay.
At least it has a similar shape to one.
Yes, like a eel shaped, yes.
What is the distance between the two? So sometimes you will see these lasers that provide us a 10 centimeter scale distance. So veis esos dos puntos verdes, esos nos permiten saber la distancia entre ellos. Hay 10 centímetros y podemos saber el tamaño de lo que vemos en el fondo. Again, more and more rocks as we keep going up this structure. Yeah. Vemos más y más rocas conforme subimos esta estructura tectónica. And more rocks means different kind of organisms, right?
You can see some worms and
Mhm.
again some pinkish sea cucumber,
Yeah,
echinoderms.
echinoderms, yeah, echinodermos. También, como siempre, vemos esas estrellas de mar. No son exactamente estrellas de mar, no, son otro tipo. ¿Cómo se llaman? ¿Tú lo sabes, Aparna?
Some are sea stars also.
Ok.
And there are some more sea cucumbers floating with a lot of echinoderms in the floor.
Okay, we can. So, do you think that we can come back to the white rocks that we just passed by? So some of the people working with us say that these white rocks could be interesting, so we are going back to see what's around it. Vamos a volver a estas rocas blancas que vimos a... I don't see them yet.
That.
Do you remember, Aparna, where they were?
Yes. This white spot.
Okay.
This.
Is here?
Guillermo is showing, but the.
We're here?
That white.
Oh, here.
The rock we were having more back. I think we have come.
So we are going back our track to see if we find again those white rocks. Estamos volviendo sobre nuestros pasos para ver si vemos esas rocas de nuevo. Do you have, Aparna, the observation?
Yes, I have.
the geological observation. So we may show to the pilots.
Yes, it is here.
Can I take this? So we can read the coordinates?
Yes.
Thank you.
So basically we are trying to go to the white rocks that we have seen few time back, so that if we can see some methane seeps over there, we want to have a look.
Yeah, the thing is, in these cases, I think they are pretty loose, so they may come from an upper part, so we may go, up, following those, so we find the outcrop. Parece que esas rocas estaban sueltas, así que igual podemos seguir ladera arriba a ver si encontramos de dónde afloran. Thank you, partner.
Okay.
Oh, there.
I think we have found it. Yes. Yes.
The idea to go around and see some of those.
Yes.
Maybe. Maybe over here, yeah. Over here. So maybe we can go all this way just to see if there are more of them. And if not, we can try to take example easier to take. Okay, so if you want, we can stop by this one, take example, and then keep going uphill.
Back of the coordinates.
That's exactly the one. It's not super hard, so we may try with the Geological to create a list. And if not, we have a Bruce list that is in the number. I think with the metal block, we will be able to take as many pieces as possible. No, it's not necessary to be yeah, that's it. Yeah, I think it could go either being on the sides on one coral river or coral or light, depending on the size. If we don't take a very large sample, we can go to coral river. Depending on how big it breaks.
So we'll have to use 2 arms. I don't know if there's a portable for us.
Alright, I mean, whatever is easier for you.
Okay. I'm gonna stick some.
So we were talking about how to get these rocks. We will try to take a piece from the large rock right down of us with the metal claw and put it in one of the containers. Estábamos hablando de cómo adquirir esa muestra geológica y seguramente lo que hagamos será utilizar la garra metálica en uno de los brazos y almacenarlo en uno de los containers.
Antonio, so we are going to have a sediment scoop?
No, I think it's going to be, depending on how consistent, how it breaks. But if it's large enough, we will going to put it either, depending on the size, on a...
Okay, so rock sample.
Yeah, it's a rock sample either way, yeah.
Do you think it's a biology sample, Antonio?
No, I think you can
Geology sample.
say that it's geologic, but if we find something interesting, some of the people working in biology can take a subsample.
Okay. Where are you planning to put it in rock box number?
Mm-hmm.
Which rock box number?
No, I think it's depending on the size.
Okay.
We'll be going. So coral keeper 4, right?
Okay. Okay, coral keeper 4.
Vamos a almacenarlo en este container. Denominan coral quiver. Y esperemos que podamos obtener un fragmento suficientemente pequeño como para que se pueda almacenar. So we're going to try to put the sample in that container, depending on the size. But I think it's very easy to break, so let's see how it breaks. Vamos a ver cómo rompe. Yeah.
You want to grab that small piece one, or you want to grab a bigger piece?
How large is that? I mean, it could be enough, depending on how big your arm will be. So after taking the piece, we are going to try to have a closer look, just in case we find some fossils. Bivalvi, it would be great, because it's one of those organisms that are related to what we are looking for. Benjamin and Taylor are looking the best way for taking it. It's a very delicate operation. Again, even if it's not an organism, the sediment is super loose, so it's easy to break it apart. It's very crumbly. Es un sedimento muy poco consistente y es fácil. So when we break the piece of sediment, it's also a good chance to
Maybe we can put it in a Bible.
Okay.
I was saying that it's a good opportunity to look at the part that we left exposed, to see the fresh rock under it. Es una buena oportunidad poder ver en detalle esa parte. Still trying to figure out how to retrieve the sample without crushing it. Estamos intentando ver cómo recoger la muestra sin que quede totalmente. Aquí podemos ver un zoom directo para también ver los detalles de la roca. Aparna, can you take a geological observation?
Yes.
I think it's a sponge.
I think it's carbonate only, or
I think it is, but
Guillermo, ¿es that a sponge? ¿Es una esponja?
I think brachiopod. I think it's
Look at this.
brachiopod.
Okay. Ketone. Ketone.
Ketone.
this is a very relaxed. So we may do the sample as much as we can retrieve in the bio.
In a piece of rock, we can see number of lives living together with their micro colonies. Very interesting.
It's an habitat
Yes.
very rich. Es un hábitat muy rico. podemos ver, como dice Aparna, un montón de organismos distintos asociados. Again, this is a very delicate maneuver. Es una maniobra bastante delicada.
Okay, so this kind of samplings are often very tricky because, as you can see, the carbonate rock structure is loosely bound. So the rover SuBastian has a lot of specification to catch this up, but you have to be very careful and precise when you are collecting the samples. So what we are now trying to do is catch it up with high precision.
Let's see.
It's
possible, it's okay, yeah. Still for a cleaner for So a partner, we will try to put that in the coral quiver number 4, just in case it fits.
Okay.
It's difficult to know the size. Yeah. So after this sampling, I will be leaving for today, and I will leave you all with Dr. Andrew Palmer, that will guide you all through the rest of the next 2 hours. And I think he has lots of nice, interesting things to tell about all this. So, thank you for staying with me. Gracias a todos por estar estas horas con nosotros. Ahora haremos un cambio de turno. Y os dejaré con el doctor Andrew Palmer, que seguro que tiene un montón de cosas interesantes que contar.
Thank you so much. Thank you so much, everyone. See you in some again opportunity.
So yes, I will give you Andrew. And what I was thinking is, as we were skipping the recording number 4, instead of going where we were going, we just go uphill to the next point. So I don't know how the topography is at this point, but just uphill.
Okay, so
I think
this is where I was heading,
Yeah.
and now you don't want to go here, you just want to go here.
No. I mean, here is fine, but just going, to the most upslope as possible on that direction.
Straight uphill.
Yeah.
Okay.
Does it work? Yeah. So thank you so much, Benjamin and Haley.
Roger.
See you later.
It was easy, it was easy. Thanks so much.
Thank you. Thank you.
What? How you doing?
I'm good. I'm getting adjusted here.
Yeah, no worries. Tyler, you've got this.
Oh.
What's the same?
They said, despite being handicapped by your phone, you do a good job.
What's that? The camera on the side is kind of
The battery,
How often?
this one is every minute. It's like every 15 minutes, and then all of a sudden, and that's the transition.
I'm sure. My coffee's done afternoon. For that's all the time.
So Andrew. are you
Yes.
gonna- So we're gonna head. You want to head uphill?
Yes, please. I'm basically moving towards Site 5 instead of Site 4. We're just going to bypass it and head for that outcropping. Well, what we believe will be an outcropping. So good afternoon, everybody, or good afternoon for here. I'm Drew Palmer. I'm a professor at the Florida Institute of Technology in Melbourne, Florida. We're about half an hour south of Kennedy Space Center, if that helps you get your bearings. And I'm taking over for Antonio on our methane seep search today that we're going for. We just watched a kind of great display of skill there, capturing a rather crumbly bit of rock of interest to our geology team. And we're now ascending this hill that we think where the outcropping from that might have been.
Andrew, just so you know, we're gonna use science camp for a minute.
Okay.
Bridge RV, we are on the move again.
Okay, thank you.
On the move again. Fantastic, thank you. I'll follow you there.
So if you joined us for yesterday's dive, you'll know we did quite a lot of exploration of the macrobiology in the area, and we collected quite a lot of samples, which were rather fun to look at last night. And then we're doing some more today, a little bit, but we really are focused today on the methane seeps which is another big part of our expedition here. So while we are slowly making the ascent, I am going to give a big shout out to the Schmidt engineers. We started this morning with two of our water pumps that we needed for some of our filtering not working anymore, which was a bit of a scare. And in less than a couple of hours, we had one working, and they're still working on the other one. So really just amazing work. I can't thank them enough.
Some fractures on the wall. See like this.
We have a question about whether we're using the same mapping that was used to find hydrothermal vents a couple of expeditions ago. I see a head shake in the no direction.
That was a very scary. Yeah, if the question is referring to the INSAS, no, there is no INSAS, the interferometric aperture sonar. We don't have that currently installed on this expedition. That was a very specific expedition testing that piece of equipment.
All right, thanks. So while we are continuing our ascent, I'm actually going to give a shout out to one of the members of my lab. Happy birthday, Tyler.
Happy birthday.
That is from Katie Hubrick, who is one of my grad students in the lab who's working with me here, and Ty's partner.
There's your fracture.
Yeah. Right. Good. I was just checking to make sure one of the geology team members is in the room here with me, because my WhatsApp is for our group chat's not working so great in here. So don't want to lose sight of anything. Do you think we should get up to 5 first and then we will? Okay. So like here or up to there? We're consulting with our geology participants in the team for some likely sites to do some good digging. Hello to Kirsten Kutchaw, who's in the chat, who is busy writing her dissertation. I know because I've been editing some of it on this trip. Yes. I could. Sure. Right. As Joanna sat down, I'm Drew Palmer. I'm a professor at the Florida Institute of Technology. I am a biochemist by training, but I currently work in areas of microbiology and plant biology. My typical area of research is actually not under the ocean, but more outside of the world. So I'm a space biologist by what I do most of the time, just trying to understand how biology on Earth is going to survive in space. Specifically, we try to work on understanding how to grow food to support astronauts, say on the International Space Station or on a future Mars or lunar settlement. But we have a lot of interest in the origins of life and weird life, as I call it, like extremophiles. And so this particular location and what we're looking for today with these methane seeps are of great interest to us. And so in particular, these methane seeps are of interest to me because the types of microorganisms that can survive in these types of environments, we do believe that at one time there were methanogens or methane-producing microbes on Mars. And so understanding more about how that life can survive in an environment like Mars when it might have had water and methane could be really interesting. Right, and we are going to try and touch down here, I think. Yes.
This kind of alignments that you can see clearly in those outcrops is the kind of factors that may be interesting to look at. Yeah, maybe closer looks or whatever.
Okay. So these outcrops are of particular interest to our geology group. Do you guys want to try and take a core? Do you guys want to try and take a core?
I don't know if that would be possible, but we may try, depending on how far it is.
Do you think we could try and sit down and take a core at one of these spots?
Sure. We just passed that there was like, all this deep, and at some point there was like a super straight linear, so maybe that would be nice.
Antonio, I think you do a much better job of explaining this. Do you want to hop on the mic real quick and relate that to everyone? That's really interesting.
Do you want me to talk?
Yeah.
Okay. So, in Spanish? Oh, in English. In English, it's okay. So, we just are looking into this linear, very linear, as you can see here, very linear structures. That is the kind of things that we are looking for. Sorry. It's just following this outcrop along and to see there are some specific things that may have still open and fluids bubbling up. But that's the difficult part.
So this is a good site for perusing for methane seeps.
Just underneath me now, there's nothing fun. I just saw this on Swag, so I went.
Okay.
So if you want to stay in this area, I'll have to back up a little bit more.
Okay, we can maybe take a good look to this and then keep going.
Okay, because there's nothing on fun.
Okay.
That was the last part.
So
I saw this on Swag, that's why I went this way.
As Benjamin just said, this is super located in this area, so we are going to look around a little bit in here and then keep going to the next waypoint.
Okay.
So the push core in this area?
No push core here?
Is it, I don't know if it's possible to take it, but we may try.
It's up to you guys, yeah.
You want to go for it?
I will look
Click
for,
here?
as you can see,
Yeah.
there are lots of lines in this way, and sometimes those lines have like a specific texture, so I don't know if, for example, over here or someplace similar to that.
Okay, so a place where it looks like there's kind of a transition.
Yeah, maybe it's the best place because we may found not just the rock, but the things that go through those tectonic, spaces. Yeah.
So if we could try and take a push core from this line, I guess this area, there, there. And for those of you just joining us, we're setting down a little bit to try and take a push core of the material in this area, which is of potential interest to our geology members of our expedition.
So we do want to push more on this?
Yes.
Yeah. Okay.
Yeah.
You want to
please.
do it here, or you want to explore?
He wants to try and you want to grab the push core first. Let's just grab the push core now, and then we can continue exploring.
Yeah.
Okay. Yeah. So we're gonna go ahead and grab push core. I guess along one of these transitions here. Yeah. If it's not.
Andrew, just in case, if we are able to get a very close look to those materials when we took the push core, just in
Okay.
case there are little bivalve fossils or this
Okay.
kind of things.
Okay.
Just in case. I mean,
Yeah.
I'm gonna park all of it like this, and then we'll go at the
Okay.
edge, right?
We're just working on the maneuvering.
ROV breeze.
Yeah, like we're going to pause here and grab a push core, but you are good to head to the waypoint.
Copy that. I'll
so
take that to the way.
you can see
Thank you.
these very, very straight lines that are
Yeah.
doesn't look very natural, but these are tectonic forces, stressing the surface.
Okay.
So you think that's push coreable, Antonio?
Mm-hmm. Yeah. I mean, depending on where we can, but anywhere.
That's not rock, is it? I
For
think it's
example,
rock.
in here, if we can get a closer look to this part, I think this is super nice because
Okay.
it looks like you have the front of the fault.
Okay.
Maybe get closer. You could point where you're on it.
Oh yeah. That's so. Andrew, thing is, we may be looking for seeps, but not active seeps, but some
Yeah.
that
Were formerly active, yeah. So in the methane seeps that we were looking for everyone who didn't hear that, we may not be able to, we may be looking for the former presence of seeps. Active seeps would be really great, but we're also trying to look at some of the geology in this area to determine when seeps might have been present.
Okay. So you want in between, right?
Yeah. So I think this area,
It depends on
how rocky it
easy
is.
to penetrate that.
You want as close to that as possible.
Yeah.
So,
If we're not able to take the push core, we may take just a piece of rock.
okay.
Your magic.
Neither. Can we move away from push core 1 or 2? We're trying to reserve those in case we find biofilms. Sorry. We should have push core 4 2s, yeah. Okay. They're all just as quiet as we are, waiting for the push core magic.
Push core suspense.
Da-da-da.
What kinds of things are you looking for in these push cores?
So it's going to depend. In some of them, we're looking for interesting geological features, microscopic fossils and other things that can tell us about the history of the Atacama Trench. Some of us are looking for interesting microbes that survive in this environment. That again, as many of us on this particular expedition are astrobiologists, we're kind of interested about how life on other planets might survive by using organisms that survive in some pretty crazy environments on our planet. And in fact, that's part of the interest in the Atacama Trench is the Atacama Desert, which is on land here in Chile, which is the highest, driest, and most Mars-like desert on the planet. And so, and so we think a lot of what we might find here may help us understand ways and strategies that life might have to survive on other planets, but also teach us about interesting geological and biological forces at work on Earth. My particular interest is in a lot of the microbes that we might find here and some of the interesting types of chemical processes or metabolism that they might have. So one of the really interesting things about this particular expedition is the diversity of the backgrounds of the scientists involved. So we have astrobiologists, chemists, geophysicists, geologists, marine biologists, a really interesting group. Did that answer your question?
That was literally perfect.
Awesome. This is that moment when everybody gets really quiet in the control room.
Cores are high pressure.
Yeah.
It's also high pressure because I'm switching from cam to cam all the time.
Also high pressure
It's high pressure. I'm switching from caffeine to decaf. Could some of this be information be useful for origins of life stage 2? Absolutely. So I'm particularly interested in certain types of prebiotic or pre-life chemistry processes that go on, and some of those processes may be at work under the sediment here. Materials that are known as coacervates. In fact, Amber, who's one of the grad students on this project, on this expedition, is going to be working on coacervates for her dissertation work, probably. Coacervates. A coacervate is, I'm going to work my brain on a simplified explanation. I immediately jumped to NASA pre-proposal. So the best way to think of it is coacervates are like collections of small types of molecules that we find in life, like the nucleic acids that make DNA or the individual amino acids that make up proteins. And when they aggregate together, they can make almost kind of like an oil droplet in liquid, so like a compartment inside of like a compartment of like the cells in your body. So in fact, we have coacervates in us. They're different parts of our cells that are these structures. And there's some theories out there that that's kind of how life began, was you kind of had a little mixture of different types of molecules before life actually got started, but it was all the right molecules that we find in life now, and they found complementary chemistries, so to speak.
Thank you. That's really cool. Really cool. I also think we have any word.
Okay.
I did a coacervate.
All right. So we have a geology request. Can you test how They want to know how strong the rocks are and if we can get a sample of the rock or a core, or do you?
If they're soft enough to get a core, but I don't think
Okay.
so. If they're not, then we can
So
try this.
if it's soft enough, they want to try and get a core from one of these rocks, and if it's not, they want to just see if they can get a hunk of rock.
Some scientific book.
This is a scientific term, a hunk of rock.
Yeah.
So,
So you wanna, he's gonna poke the ground first and see how far it'll go.
Okay.
And if it feels good, then we'll get a core and then sample.
otherwise, you will get a metric chunk of rock. It is a kilo chunks.
We're gonna move forward. All right.
Drew, Hannah's gonna join me in the MMT seat. If that's fine. It's gonna be both of us.
You're gonna get both of us talking to you, Drew.
Okay.
Should I introduce myself?
Yes, please. I was just read my mind.
Yep. Hi, everyone. My name is Hannah Nolan. I'm the Expedition and Community Outreach Specialist at the Schmidt Ocean Institute. So I'm part of our shoreside team, which means I don't often get to come out to the ship. But this is one of the rare opportunities that I get to be here, working with our fantastic crew and learning from our scientists. If you go on the Schmidt Ocean website and you do any reading on the expeditions, it's probably me plus our team that have written it. So I usually do the drafts, and then our fantastic science communication manager Jenny will edit those drafts. I work with the scientists before they come on the ship to kind of learn more about what it is they plan to do, and I write our outreach plans. So try to explain to everyone what it is that the scientists are planning, and those guide our video production, they guide our writing and our messaging. I write press releases again with a team. They usually draft the press releases for all the expeditions, and then I run our Ship to Shore program. So if you haven't heard of the Schmidt Ocean Institute Ship to Shore program, it's a virtual field trip that happens over Zoom. There's a registration form on our website to request a ship to shore, and so I set all of them up, and then Sophia runs them. She actually makes sure they happen. So basically, I'm on the organizational side of things, and then our wonderful multimedia correspondent on this expedition, it's Alex. They actually make sure it all happens. So
Okay.
that's what I
So it
do.
just broke off.
I'm here just to
You
kind
wanna
of
try and
learn
grab that
and
piece? Do you
see,
think we can get a core?
and it's really exciting.
Do you think it'll core, or do you think it's too?
Through the rock?
Yeah.
Thanks.
Also, it's just really fun to hang out with our crew and with the scientists.
Or Crumbly?
And this is my first Schmidt Ocean Institute expedition. So seeing the live
Do you wanna
stream
try
from the
further
other
up?
side is pretty special. Because usually I'm monitoring it from home.
Okay, so you wanna knock some rocks off and collect both samples?
Okay.
Oh, yeah.
So not a core. Break some rocks up and do
No,
this.
they don't think the core is going to work, so I
Okay, so
Or even this on the whip.
So for those of you who might just be tuning in right now, we are
Yeah.
currently looking for methane seeps.
uh,
These are areas of the seafloor where there's been a high concentration of methane buildup, usually due to decomposition over a very extended period of time, but then geologic activity has exposed these parts of the seafloor, resulting in methane seeps, and this chemical, this produces a lot of chemical energy, and there's usually quite a bit of life living around the methane seeps. We're at an exciting place right now because this looks like an old methane seep. We have this crumbly carbonate structure that is indicative of a methane seep in the past. We also have these burrowing kind of holes that you can see. The methane seeps around Chile that other past scientists have researched have these mussels that live on them. So this could be leftover mussel holes, potentially.
Can't confirm that, can't deny that. We haven't seen any shells, so we're not sure. But it's really exciting that this is kind of showing us that we are on the right track. And even if we can't find any active methane seeps, we can also learn a lot from these potential older, like past methane seeps. So again, we have to get all of these samples on board to confirm this this hypothesis, I suppose is the word. But for right now, yeah, it's pretty exciting. We're hoping we're getting close to what could be a methane seep. Drew, it looks like we have a question for you in the chat.
So this is from Kirsten, is one of my grad students who actually works on bacterial eukaryotic quorum sensing, which is something we work on in my lab. And so actually, yes, we do think that some of these biofilms might be quorum sensing related. And Aparna, who's one of the professors in the university system here in Chile, is a biofilm expert who's going to be working on those cores. We're actually going to be collecting a core ourselves here, and the geology team's going to collect a core in just a minute as well. So turns out we're going to be at the site for a little bit longer. And Aparna and I have already had a couple of conversations about quorum sensing. Nice, very nice.
See the close-up of this before it goes in the box?
Yeah, thanks.
Oh, poor Miguel. One of our scientists in there, Miguel. It's quite sad that we dropped his rock.
Yeah. Would it be easier to get this with a sediment scoop? Would you like to try
So another reason that this is indicative of a past methane seep is just how crumbly this structure is what we've been hearing. So while we are very sad that Miguel has lost his rock, we are excited that it shows the structure that we're looking for.
Okay, so we're going to try one more time to get a piece of intact rock. That little guy is very intimidated by the claw. So in answer, quorum sensing, I see this question in the chat. Quorum sensing is a process by which microorganisms, unicellular organisms, communicate with each other and can regulate behaviors like the formation of biofilms, stuff like that.
Do you want the top or like one in the middle? That part.
You want to try and break that rock?
Which one do you want? Yeah. This or the one in the bottom?
Okay, if you want, you can try this one around here so that this piece falls off.
Okay. We're going to try and keep a piece intact here by breaking that off all in one piece.
You want to pick it up or you want to allow each other? Might be hard to differentiate what we got, so let's try to try and cut it.
Mm-hmm.
worm too.
Yeah, there's a little worm inside there.
Oh.
Actually, I'm looking across the surface of this. There's quite a bit of living things in here.
Yeah.
So, oh. Oh, oh. No cheering yet. No cheering yet. Don't jinx it, guys.
Thank you, Andrew. Nope, don't turn around on top. Yeah.
Everyone hold your breath.
It's too big for the quiver. I'm going to slide in there.
But don't pass out, please.
Feel really quick. Stand it up. Yeah. That's why I love you, Dan.
All right.
You can do it.
Okay. We feel so gotta get too close. Too close here. Okay. Let's get close.
It's like nothing happened.
Nope.
I think if it breaks a little bit, guys, if it breaks a little bit going into the quiver, you're fine. Right?
Hi, Brent.
You see?
Yes. Shout out to my boyfriend, Brent, in the chat. Nice to see you, Brent. Thanks for joining us.
Switching back here, I can explain.
And returning.
Can I steal this?
Yeah, please steal this.
We have a question in the chat. Is that a new camera mounted on the manip? No, it's always there. We just don't frequently show it because it is in standard definition.
Yay! Crumbly rocking the tube.
All right. Awesome.
Good job, Tyler.
Yes, Tyler.
Do you want the other chunks in there as well, or do you want them in a different?
There's a whitish chunk down to the right,
Yeah.
which I would appreciate you getting rid of.
Okay.
Okay.
Do you want it in quiver 4, or do you want it in a fresh?
I'm gonna go for the whitish chunk down by the rock.
Miguel is unsatisfied with the rocks.
Miguel needs more.
He needs
rocks.
more. His appetite for rocks cannot be sated.
No, don't apologize. You're good.
Miguel, you want to hop on the mic and tell us why you need these rocks?
Yeah.
Except he's very busy thinking about the sampling protocol. So maybe in a bit we'll have Miguel hop on the mic and tell us about the rocks he's looking for.
That one can go in the same quiver.
Same quiver?
Yes.
Okay.
So if you're just joining in, hello, welcome to the dive stream with Schmidt Ocean Institute. We are currently off of the coast of northern Chile. My name is Hannah Nolan. Joining me is Sophia, our multimedia technician.
Hello.
On our main, on our chief science mic, we've got Drew. And
Hello.
A whole room full of people. So we're off of northern Chile in the kind of slightly above and in the Atacama Trench. We're not going to be able to get to the very bottom of the Atacama Trench because the Atacama Trench is 8,000 meters deep, and our remotely operated vehicle can only go to 4,500 meters deep, which is still quite deep, just not as deep as the Atacama Trench.
These chunks, Miguel?
We are looking for methane seeps.
These are the
And the
ones.
reason we're here is because
The
the
biggest one
Atacama
you got?
Desert, which is just right off, like, is right next to us, basically. If you go to the land, you will get to the Atacama Desert. It's the oldest and driest desert in the world. It hasn't really moved position over the past 150 million years. And so the microbial life in the Atacama Desert is really interesting. And so our scientists here, a lot of them are astrobiologists, and a lot of them specialize in microbes and microbial ecology. And they're looking to see if the microbial life in the trench is just as interesting, and also if the animals that live here can also provide some insights into how life developed on our planet. And maybe we can find some animals that we refer to as living fossils. These are animals that haven't really morphologically changed a whole lot since their common ancestor, or since their ancestors in the fossil record. They might have changed genetically, but physically they still look pretty similar. Where some of them haven't genetically, like they have changed genetically, but much slower than other animals on our planet. So we found something that's indicative of methane seeps, this structure that we're looking at right now. So that's why we're sampling heavily here, and then we'll continue on and hope to find some active methane seeps.
And yesterday's dive, we actually found two samples that we acquired that we think may actually be pretty good representations of living fossils also.
Yeah, do you want to tell us about those samples, Drew?
They look like living fossils. As a microbiologist, one of them was called a chitin, and it looked kind of like a big roly-poly, for lack of a better term. So, but our marine biology team was really, really excited by that. And I think, did that get into the coral quiver? No.
It's just on the edge of the coral quiver. Ooh. It's stressing us out, Tyler. Nothing.
But so yesterday was very exciting. In our first trip down, we caught a bunch of really interesting examples of known marine life, sea cucumbers, anemones, stuff like that. And we captured some of them, which looked like they may be species that hadn't been well characterized. But then we found at least one or two of these chitins and a few other things. And then today we've been all about the geology and microbiology and these methane seeps.
Were there any brachiopods that came up yesterday?
I actually do not know the answer to that question. Any brachiopods come up yesterday? I think in the excitement we've lost track.
Fair enough. We're going to have a pilot swap out right after Tyler puts this rock into the tube. And I think the rock has fallen between two of the coral quivers. Or in between the coral quivers.
So here we got a chunk of the stuff on top.
And Miguel's cool with this because we still want to collect two cores at this site, and we want to focus in on on a micro, what we think might be an organism. So we can.
All right, so go bids back out then.
Miguel looks sad. No, but has accepted his rocks.
Is there anything in there that we need to put this back on?
4 has a big chunk of rock in it.
But it's not
Pretty
even
Charlie.
about 5 meters.
Okay, well, we're having our pilot changeover. We have Zach on co-pilot and Adam piloting. I want to give a quick little shout out to my friend Laura, who's tuning in right now. She's getting ready to defend her PhD at the end of the summer. I'm wishing her the best of luck. She studies the waggle dance of honeybees. Cool. Yeah.
That's very cool.
Good luck. Yeah, so good luck, Laura. We're rooting for you.
I'm going to say hello to Mia, who also works in my lab. Mia's an environmental science student at Florida Tech. Okay. Katie says hi, Mia. She's in the back of the room, waiting for her turn on the pumps.
Okay, so what is our first task now?
All right, so we'd like to focus in, I think, Jess, was it right here? And this guy? Yes. I'm going to kind of get a zoom in on this little critter here.
Squat lobster?
Yeah.
Hi, buddy.
Okay.
It does look like Gonzo.
All right. Yeah. Okay, so that's cool. I think we saw what we wanted to see there. So we wanted to take 2 cores at this site, if possible.
Sure, and you just want them directly in front of us on the bottom or on top of this?
I think we want them here on the bottom of Perna. You should confirm you want yours here at the bottom? Yes, okay.
And, according to my thing here, we're on 5.
So can we take the first one? We'd reserved cores 1 and 2. So if we can use one of these for core 1, and then the other one can be core 5, please.
1 and 5.
Yeah. Core 5. That's going to be for. Sorry, that doesn't need to be out there. That's for my lab. Core 5 is for me.
What is that?
Then core 1 can be for Perna. So both Perna and I are interested in some interesting similar things in microbiology. So these former methane seeps might have some interesting biofilm type architectures and microbes there. And so we both would be interested in looking at the microbial populations at this site and what kind of metabolism they might do.
All right, how's that looking for you?
Good.
Happy?
Okay. We have a go.
All right.
And I'm going to confirm that's push core 1 to our data logger.
See, earlier we took a push core and we got a critter in there, Alan.
Yes.
That's pretty cool. Starting to push pretty good. Maybe hitting something. Give me a little zoom out. So, yeah.
Okay, do you want a partial or do you want to move? Do you want to save that as a partial or do you want to move to try and get a different core? Okay, so she's good with that.
Okay. You can push out a bit.
Male fauna. Male fauna. Animals made from male.
Yum.
Did you say animal?
They shouldn't be allowed to sit next to each other. No.
And so in many cases, these first couple of centimeters, 3 to 5 centimeters, what most of us are really interested in.
Oh no, are you switched?
And that's just kicked up a ton of sediment.
Yeah.
All right. And then one more. With short 5 is free.
What? Oh, it's an orchard.
Yeah.
Your mic's currently not on.
It's her chair, I think. No?
All right, you want this one a little further away from the cliff? See if you can get a little deeper maybe?
Actually, about the same distance would be fine. We're only interested in taking the first 5 centimeters too. So if we get about the same, we'll be just fine.
Okay. So I'm probably going to push you around a little bit. You ready? Won't get too crazy.
It has been like 2 minutes since I switched up the feed. Did you guys fix this? I can't.
Usually I found people like break through this layer if you just hold pressure.
Yeah,
It's like
just
you're getting off it.
you hold pressure and walking around a little.
Yeah.
That's what I'm trying to do, but it's just, you know.
I jinxed it. You totally jinxed.
That's
I'm gonna look at the first 5 minutes.
great.
Just hold there because it's going to be a lot of rocks in there. That's great.
Miguel, if we switched your mic on, would you want to explain why you wanted those rocks so badly? That's what is. Okay, so we're going to hear from Miguel, and he's going to tell us about the rocks that we just sampled and what he hopes to learn from them.
Okay, so the main thing about these rocks is that they appear to be, well, I don't think there's so much rocks as compacted sediment. And wait, I'm being assisted. Thank you. Okay. So the things that they appear to be heavily weathered, they do show not so much as different strata as different colors within them. I am mostly going to focus on the microbiology of the sediments, particularly focusing on their transcriptomics, what the microbes on the sediments are doing. So I took them mainly to see what are exactly the metabolisms that are taking place in these rocks and to which degree they may affect the weathering they appear to suffer.
On the geology side is that we're moving up if we can move along the outcroppings.
So, sorry, just to clarify, because we had a lot of activity just going on there. So you're looking at the potential metabolisms of the microbes in the rocks?
Yes, I will There are several of my colleagues are going to work on metagenomics, which will basically focus on identifying the microorganisms in these spaces, as well as understanding their complete genome. That will be all the things they are and they can potentially be. I will be focusing on their transcriptomes, which are the things they are currently doing, basically, which can align but do not always align. Because of this, I will mainly these rocks are not like amazing in the sense that they were a mass choose, but they seem interesting. The amount of water they had was interesting. They appear to have several pockmarks and little holes that were actually the habitat for fauna. We saw several crustaceans there, sea stars and other fauna. So that's the main purpose, to see to which degree are there microbial communities living within these rocks, and what exactly are they doing, what are they living off? Are they living off degrading and consuming the decayed organic matter that arrives here in huge amounts, or do they maybe show the metabolisms? That's mostly the purpose. In fact, we can just hear probably more attractive place than the one we just sampled with two
just a few meters ahead.
Yeah.
Was an even better.
Yeah, with two very clear strata, which might be very interesting.
Probably shrimp.
Yeah, this is like shrimp heaven, and we have come here to disturb it apparently. Bad luck. Anyways, that's the main purpose. I could do this as well with these rocks, and it might be even more interesting. Who knows? The great and bad thing about having access to such amazing technology as here in Falcon 2 and with ROV SuBastian is that the amount of samples and data you can acquire is so amazing that it can overwhelm you at times, and it can be quite complicated. So you have to choose. Choosing is part of the job.
Yes, it is indeed. Especially when you don't know what's going to be next in front of you.
Mm-hmm. Yeah, exactly.
How long did it take you to process all the samples from last time?
Yesterday, shorter than I expected. I think the ROV was on deck around 8, if I remember correctly. I went to sleep at 1:30. So, but we'll have to take into account that a couple hours were then waiting because I had a long incubation. In another hour and a half, I had to wait to get my sample. So, like actual constant lab work, less than 3 hours. So, not terrifying. I was able to sleep, which I think some other people in our group cannot say.
Okay, the biologists are asking if we can take a zoom in on our friend here. And while we're zooming in, if you had earlier, if you joined earlier, you might have heard me mention that the Schmidt engineers fixed one of our 2 broken pumps. We just now got the second pump back, which members of the lab, my lab, who are listening should know that was our pump that was broken, and it works better than before. So thank you again, everyone on the ship. You've been so amazing, and thank you so much.
From the technician using the pump, thank you to all the engineers, but thank you to Dan, who spent the last few hours fixing that for me.
Yeah, basically, if you have any.
Okay, so this is starboard?
Hi, Ali.
Are we at max zoom for the crab, or? Okay, now I wait to make the observation.
Are we interested in anything else here, or do we want to keep moving forward? We would love to move forward, please. Absolutely.
Yes, please.
Oh, look at that.
Ooh,
That is nice.
That's a nice crab. Crustacean.
Very cool. Wow. No. And like everything else in the Atacama, he's covered in dust. So, yeah. So for anyone who, the Atacama Desert You feel like you have to wash a pound of dust off after you've been out there for a day.
The average rainfall through the Atacama Desert?
Depending on where you are, it's measured, I think, in less than inches per year in some places.
There's some parts of the desert I read that haven't had been rained on in 300 years.
Yeah. So unfortunately, one of the things that Armando Azuabustos in our group, one of our project leads here, the PI, he has been doing research there for decades now, and actually said there's places that essentially due to climate change are now starting to see rain in some spots, and that actually might eliminate some of the biology that's in the Atacama. Oh!
What?
This is fantastic. And so again, I think one of the really interesting things is comparing the morphology and the geology we're seeing here in the trench with the morphology and geology that we see on the land in the desert there. And continuing up this line.
Geology, are there other indicators we're looking for methane seeps?
Sure. So we could be looking for, a common sign would be temperature abnormalities, temp fluctuations. We could be looking for bubbles, though that can sometimes be from large decaying biomass. So a bigger organism dies and begins to break down. Here could create fake false bubbles. So we're looking for bubbles is one of the telltale signs. But sometimes we might be able to see just high biological activity, especially in the forms of biofilms, biomats, things like that might be indicators that we might be near something. Which would be particularly useful and informative. So the methane seeps are a real interest for the astrobiologists in our group who study Mars, because there's strong belief that methanogenic or methane-producing microbes may have lived on Mars at one time. If we believe that Mars was there at some point, sometimes at some point supported life. We had a lot more water, and those methanogenic bacteria might have been responsible for essentially global cooling on that planet.
Oh, interesting. Are you looking at that?
Oh, hold on one second.
Oh, yeah.
All right. Can we zoom in on some of these, and then I think we might have a geology request. You just want to look.
This looks like it more material that might have fallen from upslope, potentially.
Was that a question for me? I took my headphones off.
Yeah, I think so. Or, because we're not seeing any of those fractures. Maybe a question for one of our geologists. Oh, sea spider. Picnigonid. Good?
Very nice.
Nice.
Yeah.
It's one of my favorite critters.
Why do you love them so much? They scare me.
Their guts are in their legs.
Their guts are in their legs?
So they have like almost no body.
I just learned something new. So if you're just joining us and you're seeing this creature, this is a picnigonid or sea spider. And I just learned that their guts are in their legs. Apparently.
forward?
Yeah.
Okay. Ready to move forward when you guys are.
Do you want to take that question from Allie? Or do you want to do that one?
By they, do we mean Pygmalion?
Can they see? Does the bright light bother them? I mean, when we shine the light on, they didn't run away, so.
True. But a lot of deep sea organisms have really modified
Right.
Sensing organs for light.
Yeah, so they may be not able, they may see in very different wavelengths of light, if at all.
Yeah. So usually no.
Yeah. I think some of the fish might get a little stunned sometimes from
Yeah.
the light.
Yeah.
And we're continuing our ascent here.
Allie clarified.
Yeah.
They being the ocean life. I think it depends on the organism if the
Yeah.
light bugs it or not.
Could we get a quick zoom in on that, please? Is that just like an anemone or?
Yeah, it's an anemone.
Oh, okay. I guess we don't
We
need
are
to. I
always-
guess
we can
it's a
always
good
see.
picture. Yeah, it's a nice picture. That's a really cool little hiding spot.
There's a swimming crinoid in the background too.
Nice.
Aw. So I'll say hi to my wife Heather. I'm assuming when she says we all say hi, she's probably there with my kids, Malcolm and Adele. It's good to see you guys on the chat. Hope you're enjoying watching this. It's super, super exciting.
Are we headed to site 4 or 5?
We're moving up this ridgeline towards 5.
So
We're getting there, but oh, look at that sea star. Yeah, we have a request from the biologist for a close up of the sea star. It's ready for Hollywood.
We had a fish from a couple expeditions ago go viral. It's a Tunicate. And something I just pops into my brain every once in a while that I can't stop thinking about is how that fish is still at the bottom of the ocean and it has no idea how big this is.
It might be showing his fish buddy, so look, that was on Letterman.
Yeah, that fish made it on Stephen Colbert.
Nice.
It's everyone's fish now.
Beautiful.
That is, wow, look at that texture.
Yeah, that was very spectacular.
Drew, do you
Yeah.
know how sea stars eat?
I would love to learn.
So, sea stars extrude their stomachs. So whenever they want to eat something, they just kind of crawl up to it, and then they vomit their stomach out of their mouth, basically. And they digest externally. So imagine if you were eating, you just plopped your stomach onto your dinner plate and digested outside of your body. That's how sea stars eat.
Cool.
Are we done?
Yeah, yeah, I think we're good.
Can we zoom back into the starfish, please?
Okay. The starfish?
Yes.
Okay. There's a request for us to return to the starfish.
I want to show you something.
Starfish
Oh.
part 2.
Is there
On the sides.
One of our biologists, Nick, wants to point something out.
So these are commensal zoanthids. These are Cnidaria. So it looks very interesting. I would suggest it's not my problem. It's on me.
Can we pause here for a collection? We haven't done a biology collection a bit today, so.
That's really cool. I don't think I've ever seen that.
Yeah, that is really interesting. We also have a question in the chat. Are the red dots on the arms the eyes? And yes, those are the sea stars version of the eye, right? Like they don't see the same way that we see, and their eyes are pretty different, but they can sense kind of motion. We call them eye spots, I think is the more technical term. It's been a while since I took invertebrate zoology, probably like 10 years now, but
I think one of my favorite facts just in biology in general is that eyes evolved many times in the animal kingdom.
Yes.
So that's why we see such a huge disparity in forms and functions of eyes if you look at various animals. And that's why sometimes you'll find spiders with many different eyes, or marine critters with many different eyes. And then if something's related and it has eyes, sometimes, you know, when that animal goes and lives in the deep sea, it'll lose that function too. So eyes have a crazy history in all animal forms. Yeah. Eyes evolved many times. So like, if you imagine like a tree with many branches, branching outward, like in terms of time, and you get species kind of forming, eyes appear on those branches at different times.
yeah. The term is convergent evolution.
Yeah, so it was very beneficial for eyes to happen many times.
Yes. Everything over time evolved. Not everything. Not
The
everything
Homo
ceased.
phase.
But at the same time, some organisms have not decided, because that's not, you don't decide to evolve, but evolution have selected for not having eyes, like certain species of cave fish, or even some deep sea fish, I believe, have lost their eyes.
Yeah, and that's because, you know, it takes a lot of energy and resources to quote unquote maintain eyes.
Yeah.
You know, it takes energy from the food you eat to keep those organs
So,
functioning.
and that was BioBox.
So at some point it was very beneficial to not be spending those resources to maintain those eyes.
We could just
And it all sort of randomly happened.
Okay. Can we zoom in here, please? We're going to zoom in on some potential shells, clam shells. So while we're zooming in, Guillermo's taking a look, or Oscar's taking a look. I can tell you that I know that the ability for fish to see in different wavelengths of light, like the rods and cones, there's very, very little mutations that are required for that to happen. So it's I've worked, when I was in grad school, I worked with a professor who did that kind of research and showed how easy it was for biology to adapt to different wavelengths of light in terms of their rods and cones and their eyes. You good? Yeah. All right. All right. So we're gonna push forward, and so we were wondering, we were ready. We're kind of done following the ridgeline. If we're ready, we can just make the ascension.
But not the true ascension. We're not going back up.
True. Sorry.
We are climbing.
Yeah. So we're still on the hunt. We've seen some good evidence today of nice remnants of methane seeps, but we're really on the hunt for active ones. So that's part of what our goal is for today. So we're going to make the next big climb. We've been kind of going along a ridgeline here for the last 2 hours or so. We collected a good amount of cores that will hopefully have some geology and microbiology. And then we've collected some nice macrobiology today as well. So as someone who works mostly with microbiology, when I say macrobiologist, I mean if I can see it without a magnifying glass, then that's too big. Though we do grow plants in my lab, but it's basically so that the microbes can, do stuff on the plants, like control their growth and make them grow better. So they're testing there are ways that we test bacteria using plants.
Drew,
Yes.
earlier you said that there's evidence of life having existed on Mars. What is some of that evidence, and then what are we still looking for potential, what would life of Mars been or could be?
Well, so there is still, a fairly decent camp that thinks that there's, strong possibility for finding biomarkers for life on Mars. So different types of nitrogen in different forms. The presence of water on Mars is really, and the evidence that there was once a larger amount of water on Mars is good evidence that there could be support for life. Right now, the big concern with going to Mars is potentially contaminating for any biomarkers, right? That could show that life was once there, right? So if we show up and we leave our biology behind, then it'd be harder to look for the life that was there. And so that's one of the big concerns in moving there. Some of the nitrogen cycles that we see in Mars suggests that there could be or could have been biology. So a lot of it has to do with nitrogen metabolism.
Oh, interesting.
Yeah.
How
And so
do you observe nitrogen cycling on Mars?
so there's a planetary nitrogen cycling, but nitrogen on Mars is actually very, very low. It's less than 2% of the atmosphere, which when you compare that to Earth, it's substantially less, or it's got a huge nitrogen-rich atmosphere. And in fact, that's going to be one of the biggest problems, for people like in my lab, where we think about how we're going to grow food on Mars. There's just such a low amount of nitrogen. We're going to have to bring a lot of nitrogen. And so that's a really important consideration. And in fact, we have several projects in my research lab where we try to figure out how to introduce nitrogen into material that we're going to grow using on the surface of Mars or on the moon. One of the things that's been really crucial to life on our planet has been the nitrogen cycle, right, which has both biological and environmental elements that work in concert.
There's a really interesting question in the chat. How plausible would it be that we are contaminating or introducing bacteria?
In the deep ocean, as we might in space exploration. So I was averting my eye.
I don't want to.
Yeah, so NASA has an entire division called Planetary Protection, which is all about trying to figure out how we're going to go to other planets and not potentially contaminate them if we want to look for biomarkers. We don't have quite the same idea here. I mean, so there's a lot of interest in invasive biology now, right? So we worry about invasive species, although there's sometimes what's described as invasive can be difficult to understand because of this phenomenon we have in ecology called shifting baselines. But this, in this environment, I'd say probably the chances that we're going to bring contaminations that are going to survive in this environment are fairly low.
Yeah, we've had microbiologists on the past who study the microbiology of the deep sea, and they say that a lot one of the hard things is keeping those organisms alive
Yeah.
on the surface.
Yeah. And vice versa. I'd say most things are not going to make it in this environment. But we find some outliers. There are definitely things that we would call mesophiles, or things that survive in basically the same kind of environment as we do, that can survive some pretty ridiculously tough environments. But it is an excellent question, when we think about invasive organisms and contamination of various environments. All right, we are continuing our climb up the ridge here.
This might be a really outside of left field question, but I'm gonna ask it. So if there had been life on Mars in, a very, very long time ago, is there any chance that there might have been some of that life might have come to Earth, or?
Yeah, so that's sort of the panspermia hypothesis, is that life might have begun somewhere else, like on Mars, and then transitioned to Earth. I think that's completely within the realm of possibility, and there's a lot of really interesting theoretical papers looking at how well some of the molecules that make up life could survive in that environment of outer space and the transit. And I think one of the things about that is a bit challenging. I think Lauren, one of the other scientists on the project, said earlier today, is it's sort of kicking the can down the road, right? It's saying, well, life started somewhere else. I don't know what happened there either. So it's possible, right? And I mean, one of the big questions that we have that NASA thinks a lot about when it's thinking about how to define life is, how do you define life that might follow the same rules but not be exactly the same as us, right? So we have DNA and proteins and fats and lipids and carbohydrates, and what if biology was organized in a different way up there, right? And so we have to think about the sort of the rules of the game, like how does biology work? So there's no necessarily reason that biology on Mars would have to look like our biology. In fact, it'd be really fascinating if life could arise in different forms in different places.
Just with entirely different molecular structures.
Yeah, but following some basic rules, like liquid water is that's the presence of liquid water is considered one of sort of the most important things for a planet to support life. Mostly because the way we understand the energetics of biology, of living systems, you really need liquid water. Okay. Ooh, so can we pause, look at that rock?
Yeah.
And take a moment here and look at a couple of rock samples. I'm going to zoom on this?
Yeah, please.
Okay. I'm going to zoom in on that guy. That looks like a rock we collected in the desert.
Yeah,
That's
similar,
amazing.
very
That's
similar.
amazing. That's crazy. So it's only a 7,000 meter difference.
Yeah, something volcanic maybe.
Yeah. Do you want it, or you just want to see it?
No, just to see it. Because
Okay.
It's one of the big ones. So okay, we can.
All right.
Keep following.
We can keep moving on, but that's really that looks exactly similar in composition.
We passed for an area that's with a lot of little rocks that the same.
I would want to drop.
Do you want to talk for a second?
Yeah, I took a look for that kind of rock because we are following Ah, you wanted the rock? Oh, okay.
We're going back. Miguel wants the rock.
Oh. Miguel and his rocks.
I want that rock. Okay, let's go.
Okay.
Miguel just wants all the rocks.
And it's not the geologist, so.
Hannah.
Yes.
Is it on?
It's on.
Okay. Okay, so I'm going to justify myself. Okay. The thing is that in my lab, not currently, but a few years back, we were studying a thing called rock varnishes, which are specific weathering that takes place in rocks upon exposure to different conditions. And from yesterday's samples, I already saw that there is a small 3 to 5 millimeter area, surface of weathering that we would like to study later in the lab to see exactly how, if it is microbial originated or if it is just purely chemically produced, basically. Yeah. Okay.
Easy collection.
That was Rocks Box 1, right? 1 or 2?
No, but it is remarkable, having spent a couple of days in the Atacama Desert before we came to the trench. So much of the terrain looks very similar, and we actually see a lot of the similarity in the rocks that we see. It's really, as a non-geologist, so the geologists seem not surprised by this at all. But as a biologist, I find it very fascinating. Right, we're nearing our next ledge here. Point number 5 on today's map. Seven. That's right. 7.
Head towards that.
For those of you joining us, we are currently, today's dive is focused around trying to detect methane seeps, or active methane seeps. So we're looking for things like bubbles, biomats, temperature anomalies. So that was the main goal for today's dive, and we've seen some really great examples of now extinct methane seeps that might that still provide useful geology, and we can see that some of that carbon and useful biomass is still there available. So they're pretty rich in different organisms living there, but we haven't seen active seeps yet today.
I like to say an ROV dive is going on a night hike with a flashlight. Actually, I think Randy Rogen, one of our past PIs, that's her analogy. It's just you're on a hike, going up a mountain. You don't imagine.
When you cut the corner here?
If you live near
So we want to
any
start heading
sort of
towards
forest or mountain
6,
or
which is
place where you like to hike.
which one's 6?
You can only see what's on the trail. You don't see the entire forest. So there are certain things we can do in ocean exploration to make it a little bit easier to find things that we're looking for. But overall,
Yeah.
it's been it's you know, it takes a while.
Right. So we're going to continue moving up. So one of the ideas, since what we've been looking at are ancient steeps, there's a possibility that newer seeps, may actually be present higher up on the ridge. So if we move towards site 6 here, we might be able to get some good evidence of that.
My friend Nama just said hi in the chat. Hi Nama, good to see you. Thanks for joining us. Yeah. That's what?
Give it. It's gonna be dangling over you. Sorry. Mission control life.
Control life.
It's being covered in wires.
Can you hear me yet? Yeah, I can hear you.
So for those of you following at home, we're moving at about 0.2 meters per second as we climb up this ridge line. We're hoping to find some evidence of active methane seeps. We've been looking at what we think are ancient ones for the past couple of hours, which has in itself provided some useful information and some sampling. But we're going to see if we can find something a little more active up above. And again, these methane seeps are active geological sites, which produce basically a high energy environment for biology as well, right? So. Cool. Yeah, the nice anemone, and a cucumber, and a couple sea cucumbers here and there.
So many sea cucumbers. Which makes sense for where we are. Yesterday, quite a few sea cucumbers were collected, and they looked really big on the screen, but when they finally came back up and I was able to take a look at them, I was like, oh, those are really small.
So I kind of have a question. I think it seems like the kind of the astrobiology collection that have you had a lot of astrobiologists on Schmidt expeditions before?
I think I can think of one. Yeah.
We have NASA and 2017 on the
Okay.
Looking for a piece of meteor.
Okay. Looking for meteors. Okay. So.
For those that didn't hear Adam, we had a past expedition. Was that 2017, Adam?
Somewhere around there.
Somewhere in there where we were looking for meteors. Long time ago. And I think we've had a couple expeditions where we'll have like one astrobiologist on, or a couple.
Yeah, we had Betty Pugel from NASA as well.
Yeah.
So yeah.
Now you can't look around without seeing an astrobiologist this expedition.
I think a lot of the microbiologists who've come on in the past, some of them will do have work that kind of is in tandem with astrobiology, just because there is so much we can learn from the ocean, and especially again if you're thinking about that origin of life work, a lot of that happens here in the ocean, and then what Lauren does, looking at different metabolisms.
Yep. Wow, that's
not uncommon.
Well, that's very cool. I know it's been interesting from the astrobiology perspective. We've been getting a crash course from those of us on the team who have more oceanography and marine biology experience on sampling, because it's a very different kind of environmental sampling. So my thanks to all the members on our expedition with that expertise who have helped us out a lot in the last couple of days. All right. I'm going to scan the geologists in the room for interest. Good?
Yeah,
Keep moving?
That's good.
I'm very curious about this mound at Site 7.
Yeah.
It's
Interesting enough, seeing it on sit, which means sit science or bones collapsed on that one fiber camera ball. So I suspect that's good. The Omicron is the only link, right?
And inside
Or
the
top
camera,
side.
inside the camera.
Yeah. So pop that open to those cameras. Hey, do you know those numberings on that come into that can on the grid? 1, 2, 3, 4.
Just visible.
They just kind of don't miss that.
We should be able to see what's going where, you know? The one that goes to the 2 channels that go to the end of the one for sit side. What do they say? One for side.
Was there 2 that I didn't see last time?
There's 2 CWMs and they're 4 channel.
One of the 3. Seems interesting.
It wasn't disconnect a bunch of stuff like that though.
Yeah, we were trying to test this.
And this is the second dive of this expedition. Yesterday we were looking at living fossils or creatures that were thought to be extinct but might be here alive in the Atacama Trench, and we had some success there. Now we're really focused today on our second expedition, second goal of the expedition, which is looking for active methane seeps.
Hey Jason, can we bump up 0.4, 0.5? Oh, is that Ollie? Sorry. It's the echo so bad I can't tell who it is.
No worries. It's always, yeah, we're just fine.
Thanks, Ollie.
Yeah, I was wondering too, is something moving around?
Well, I suppose if we were to stretch it way out, would our attenuation go up? And if it's just on the edge, attenuation goes up.
There's confidence in pretty good.
Yeah, when we're sitting still. Like where we are now, 120? 128?
So what if we rack in or out? Is that anything?
So if my family is still on, I wanted to let my daughter know that I named the stuffed axolotl that you gave me for the trip. Her name is Gia.
I might just, I'll make the measure that Omicron and just see what it is.
Yeah. There, just short, new one, I guess.
Short width anywhere and just see. And measure the whole path with the short width then.
Yeah, do it with the old one and then the new one.
Yeah. I mean, if it came down to, and the other thing is, if those SFPs are both on the edge, keep less, because we know they get hot, right? Maybe they're both on the edge. I was reading an article about how semiconductors start using charge to manufacture fluid.
Maybe what?
Like they start using a charge like the natural components. So you start getting component failure over time, and it's like basically particles happen. Very breaks. Very interesting. I'll send you the article. It's good read.
We're still moving towards this sixth
You know this
spot.
worked great last job, so. It's something simple.
It's what we're messing with.
It's not the flux capacitor. It was working great until we got to camp, really, right?
Maybe we gotta fill it up more with things like that.
Could be something simple as just reseeding the microbial.
Yeah.
Aw.
Yeah, 'cause we definitely keep it out of the can. Well, everything I did that night.
I miss all y'all too. This is a great experience, but I do miss you.
At first, I thought, when he was looking at it, they were so tight. I'm like, did he unseat them on the bulkhead and then pinch them in?
We are continuing to move up towards 6.
Why'd you want to get them off?
I was just looking at it. Yeah, when I was cold, I was thinking.
I thought about gluing them on, but I was like, no, we might have to change it someday.
Honestly, I think they shrunk when they went down to depth, so they're really pretty good, yeah.
There we are. Yes. Yeah, so it's actually, mine's very, very interesting. So I started off I didn't get into astrobiology, space biology research until about 6 years ago. So I was mostly doing plant bacterial associations was kind of what my lab was focused on. So how bacteria and plants work together, or how they sometimes are bacteria can work as pathogens and infect plants and kill them. And so we're doing some research in that area. And, as I said when I introduced, Florida Tech's only about 30 minutes away from Kennedy Space Center. And one of the physics professors, a space sciences professor, was like, hey, do you want to go talk to some people at NASA? And I went, well, yeah, of course I do, because I'm a nerd. And so they asked me about growing on Mars. And they introduced me to Martian regolith, which basically is just sort of like the substrate on the surface of Mars, and said, well, people want to know how to grow plants in this someday, and we want to know whether or not that's something you should try to do, or whether or not you should just grow everything hydroponically. And that kind of really got me motivated in that area. And then, in about a year or two, we realized that was about 60% of what my lab does now is space biology, astrobiology, and now we've moved into whether or not bacteria can survive in these environments. We're building a Mars chamber on Florida Tech campus where we can simulate the atmosphere of Mars. We've started working with a bunch of different bacteria from the International Space Station that they've cultivated and brought back down, and we have a little pipeline where we screen them for bacteria that could help plants grow. It's been a really interesting process, and now we're kind of moving into origins of life work with Armando and the rest of the team here.
Try that. All right, I'll try that. Yes.
Yeah, it's been a very interesting thing over the last 6-7 years to watch how the slight career shift, but it's still, a lot of the fundamental principles of what we do are still the same.
Yeah, it blew my mind when I first met you all that there are actually people thinking all the time about how to regeneratively grow plants on Mars.
Yeah.
It's really, really cool.
Well, the really, I mean, as always, where the benefits of science are not solely in the discipline you're looking at. A lot of what we think about in terms of things like the sort of hyper sustainability that we think about for these sort of, we call them closed loop ecosystems, right? Like living on a settlement on Mars or on the moon. It's not like living on Earth. That kind of hyper sustainability could really provide insight to, reducing carbon footprints on Earth.
Absolutely.
Right? And so there's a lot to be sort of gleaned from this research that goes beyond just, helping support people that, will be on Mars in 50 years. It's more important than that. It's about teaching us how to use resources wisely. And so, it's sort of a natural marriage between what we're trying to do, thinking about the future, and the challenges we face now in terms of agriculture and, feeding our population.
So what I'm hearing you say is that astrobiology and sustainability have a really huge overlap, though?
They do. And
cool.
In fact, there's a whole area. What we do falls under the category of bioregenerative life support, and that is all about sustainability.
It's very cool. Thank you
Yeah.
so much for today.
Yeah, it's a lot of fun to get into, and I've had to learn a lot about sustainability also.
Just some folks popping in the chat. Is this machine diving underwater? Well, you're watching ROV SuBastian. ROV stands for remotely operated vehicle. So this is a robot that's tethered to our ship, and you're watching a live camera feed from it. And we are conducting science operations with our ROV. And we thank you for joining us in our live today.
So that's how hot was it?
Not that hot, actually.
So it was cool enough.
Yeah.
There was something that was up there
Said
for a
something,
second. I
yeah.
wanted to see if you were interested.
Okay.
It's, there it is. Yeah.
Oh, yeah.
So close.
We're having some issues with our side cam here, so
Can we take a closer look at that structure,
Still?
please?
so we really appreciate your patience as we're getting a bit more of the sit cam love today. Oh, you've got fans in the chat.
Oh, thank you.
Consider this in, see?
Yep.
Combo number 2.
Here you go. Anything you're interested in? Or?
Sorry. Oh yeah. If we
Yeah. Do you want to try and break a piece off or core around it? What's
yeah, it's. I think first a close look, assuming.
Okay. Yeah. What's with the?
This looks like an old floor,
Okay.
an old sea floor, for the extraction over the surface, but also have a lot of bioturbation. As you saw, all that channels, this
Mhm.
look like all were holes or
Okay.
caves, and also you can recognize lineaments that maybe are all ripples. That is look like the movement of the waves is created these structures.
Okay.
So it's very interesting. Yeah.
Right. Do you want to try and get a piece of it? You want to try and grab a piece of it?
Yeah, I think it's a good idea to take a piece of that rock.
We think we can break off a piece.
Oh yeah, that's pretty muddy.
Nice.
It's one pilot show right now while we have our other pilot trying to troubleshoot our science camera.
You have permission. Sorry, there's important football things afoot in here. For those of you from the United States, I mean soccer. But
Yeah. So, Andrew, you remember when Armando takes a few years ago before take the seep, Terra Roja? It's very similar. One kind of rock there in this place, in that place, is very similar at the aspect of this rock. So maybe here, this rock was part of the ancient island in front of Chile, a very long time ago,
Nice.
millions of years ago.
That's fantastic.
Do we want to put it in the rock box screen for us?
It's a bit crumbly. Can I go into a coral quiver? Or maybe even an empty bio box, maybe. I think we have maybe one.
Is 1D free?
I think it's
1D is you got the
there's a sea star.
yeah, the sea star in there, but it was on top.
But I think 1C is empty. So
1C is empty. Yep.
If 1C is empty, let's put it in something by itself. Start.
Es blandita, tenemos que guardar.
And
With that original one, it seemed to look at home. So it's got this.
Solo comentar que acaba de empezar la final de la Champions, así que por cada gol del Madrid recogeremos una muestra y por cada gol del Dortmund tiraremos una.
Do you want to get anything else from this side, or you want to keep moving up?
Yeah, keep moving up.
All right. Cool. You're ready to continue forward. So a lot of those acorn worms.
Yeah.
Really prevalent down there.
There's like
It's
occasionally
the timing.
sometimes we just delay.
It's a delay because we're getting live feed on this one. And we're getting
But it was also
This TV reacts slower than the other screens do. So this one will freeze first. And those are freezing. And these will usually come back and close.
doesn't have
Cuando quieras te pones y se lo dices.
I'm with this camera.
Yeah. Right. For those of you who just popped in or need a summary, we just stopped briefly to take a rock sample. That we took a similar rock sample a few days ago, well, a little over a week ago in the Atacama Desert. It's about 6,000 meter difference in elevation.
But theoretically, you should be able to use the 12G. Maybe 12G.
And we're now climbing this ridge line. Our hope is that as we move up the mound, we will find new active methane seeps. That's what we're on the search for today. So again, yesterday was living fossils. Today is methane seeps. Trying to balance our macrobiology, microbiology, and geology goals. It's interesting thinking about the relationship between microbiology and geology because they're very closely related because microbes acting on Earth's mineralogy plays a big role in geology. But in terms of time, right, microbiology measures things in generations that are often, hours and maybe days, while meanwhile in geology you're thinking in terms of centuries and longer.
Bridge RV, new waypoint.
I mean, I'm definitely noticing a difference in the terrain here relative to where we were down in 5. So it's definitely changing. I feel like this is different than what we were looking at down in 5. So as we go up, the geology is definitely changing. Yeah. So that's positive. Thanks, Adam. We'll see you. No, that Adam.
Oh.
Sorry, yes.
You're welcome.
I'm here as well. Oh. Yesterday's headless chicken monster. What's the total distance that we traveled today? Is there a way for us to know like the total distance we traveled today?
Yeah, we have to zoom out and we can
That's
just
yeah. Oh, okay.
Yeah.
That's fine. I was just wondering.
Is the sit direct to that? It should be. Just lose that.
Just check the input on this. I feel like we just switched inputs or something.
You got a remote for that or something?
Some of these that put us in 4K sound.
Alright, came back. I didn't get it, it just came back.
But you had it on yours, eh, though? Like you didn't lose your feet up there.
Oh, you're taking over. Yeah. Oh,
So
I
don't
thought,
worry
I thought Armando was taking over.
No. It's my stuff.
All right. So at this time, I am going to be bowing out. So sorry, I'm gonna get a little closer to the microphone here. So I'm gonna change over dive lead and take my break. It's been great talking to all of y'all. Hopefully, I'll see you on one of our later dives on this expedition. We still have several days to look for methane seeps and living fossils. So it's been great, and I will hopefully see some of you again maybe tomorrow. All right. I'm going to pass this over now to Oscar, who's one of the geology leads on our project. Yeah, I thought Armando was taking over. Yeah.
Going till 7?
Hello everyone, here Oscar from Spain. With me is Miguel that is still here in the control room. This transmission we will do it mainly in Spanish, that is our mother language, so you are welcome if you are for any country that speaks Spanish or you trying to learn our language. So maybe if you have questions or any comment that you do it in English, also Miguel or I, we can answer, so don't worry about it. So, hola, soy Oscar Ercilla, del Centro de Astrobiología. Conmigo está también Miguel Arribas, del mismo centro. Hola, Miguel. Hola. Yo soy geólogo como parte de la expedición a la fosa de Atacama que estáis viendo ahora mismo. Ahí tenemos una botella, ¿puede ser?
Yeah.
That's a can there. Can you get back?
El permiso que tenemos que nos ha concedido el gobierno chileno estipula que en cualquier tránsito en el que estemos bajo el mar, si encontramos basura o cualquier material antropogénico, debemos recogerlo y subirlo al cielo. Básicamente, aparte de equipo de investigación y exploración, somos equipo de limpieza.
Si nos lleváis viendo algún tiempo en la transmisión, habéis visto que hemos estado recorriendo una zona de la fosa de Atacama, más en la zona de la placa de Sudamérica, ascendiendo un pequeño monte subterráneo que se localiza frente a las costas de Antofagasta. Hemos visto una transición desde unos materiales en la zona más baja de esas montañas hacia unas zonas donde vemos los fragmentos de roca que veis esos en la pantalla negros, se parecen a rocas volcánicas. Hemos visto cómo progresivamente ha ido aumentando el tamaño de las rocas. Drew ha explicado durante su transmisión que el aspecto del terreno es muy similar al del desierto de Atacama y no le falta ninguna razón. Si tenéis la oportunidad de venir por aquí, o bien si sois chilenos y conocéis el área, habréis visto que recorrer la zona de las regiones de Atacama, de Antofagasta, de Copiapó, Arica, Parinacota e Iquique, el área en la que se extiende el desierto es muy parecido a lo que tenemos aquí en la zona de la fosa, pero sin los 2,5 kilómetros de agua que tenemos ahora mismo encima del submarino. Si estáis más interesados en lo que es la geología local, Chile es un país tectónicamente muy activo. Desde hace 250 millones de años ha evolucionado hasta su formación actual, donde podemos ver la cordillera de los Andes, que forma una frontera física con sus países fronterizos, Argentina y Bolivia, y al norte, bueno, con Perú, pero ya por la parte del desierto. Que encontremos esta cordillera aquí no es más que el proceso de evolución geológica de la subducción de la placa del Pacífico sobre la placa sudamericana a lo largo de aproximadamente los últimos 250 millones de años. Okay, we can store here and take a rock sample.
The rock field there?
Okay. So maybe we can zoom here. Aparna, que es una de nuestras compañeras, nos ha pedido el poder recoger una muestra de roca en la que posiblemente haya bastante microorganismos viviendo sobre ella.
¿Dónde van a colocar la muestra?
Where we put the sample?
The rock box?
We have space in box 3 A and B, or we can just put it in the rock.
Rock box?
Okay, in that case, let me take a good look so I don't confuse it with my rock. Okay, yeah, that's good. Rock box should work.
Okay, we can see in the screen all the little organisms that are living over the surface of that rock. For me, as geologists, it's very surprised how the life we can find here in that depth. The multitudes of microorganisms or macroorganisms we can see. Or college biologists are very excited about what are we looking for here and what we find.
What do we got, 57 now?
Just on the cusp and whatever we can find it. No, because it'd be a bit. Maybe not, I guess.
parece si lo visto un rato?
Venga.
Bueno, ya os ha contado no sé muy bien, pero el objetivo principal de la inversión de hoy ha sido hacer una serie de transectos a lo largo de una serie de puntos de interés que no sé si podréis ver en el mapa, con el objetivo de encontrar surgimientos de metano, lo que se llama methane seeps o coal seeps en inglés, que son simplemente aperturas en paquetes de metano subterráneo por donde sale y en torno a los cuales se genera un ambiente tanto químico, geológico, biológico muy interesante. Hemos encontrado varias zonas que probablemente sean antiguos restos de estas zonas fosilizados, pero ya no activos, pero lo que son activos no hemos sido capaces de encontrar todavía. Por eso estamos siguiendo nuestro recorrido. Hemos empezado en una zona más baja, unos 2.600 metros de profundidad. Llevamos estratos subiendo de forma relativamente lenta y progresiva hasta los 2.400 metros de profundidad en los que estamos ahora. Estamos en una zona, como bien ha dicho Oscar, con una cantidad de rocas mucho mayor. Antes era mucho más abundante en sedimentos y podemos ver también que hay una cantidad bastante importante de seres vivos. De hecho, aunque la expedición de hoy se va a alargar hasta las 7 de la tarde de Chile, es decir, unas 3 horas y media más. Si no hemos encontrado nada para las 6, es decir, dentro de unas 2 horas y media, muestra interés en volver a esta zona para que todos los biólogos marinos puedan dedicar esa última hora a muestrear todo lo que les apetezca. Ahora parece que estamos centrados en esta zona, podemos ver un crustáceo tipo balón y demás cosas.
We can zoom there.
Y en eso estamos.
We can zoom there.
Nos piden que hagamos zoom para ver ese fragmento blanco, que puede ser un bivalvo. Es un poco difícil de distinguir, a ver qué dicen mis compañeros.
Can you zoom more?
Something at a certain point rotates.
This one. This one. We can take a sample of this area. We take care of the tortillas. Yeah. Drew en la anterior, durante la anterior presentación de Drew, bueno, ha explicado un poco en inglés parte de los objetivos de la misión, que está dirigida por Armando Azúa, también del Centro de Astrobiología. Uno de los objetivos, aparte de buscar las methane seeps, o las fuentes de metano, como ha explicado Miguel, es la búsqueda de fósiles vivos, es decir, organismos que durante los últimos millones de años no han variado prácticamente nada en la evolución, como por ejemplo, para algunos podría ser conocido el árbol, el ginkgo biloba, que durante millones de años no ha sufrido ningún tipo de transformación. Podemos ver fósiles en el registro geológico que son exactamente iguales a árboles que podemos ver en la actualidad. Lo que hacemos en esta expedición también es buscar esos organismos que se localizan en el registro fósil y geológico de Chile, pero que posiblemente todavía sobrevivan dentro de las costas chilenas en estas profundidades.
Una curiosidad de poquito que vamos a intentar adquirir ahora.
For this organism here, and if possible, The white one.
Es un molusco. Así desde
Sorry.
afuera. Pues parecer al ser
Yeah,
un organismo
this-
segmentado, puede
yeah.
parecer un artrópodo, una cochinilla, pero es un molusco.
Lo que vamos a hacer ahora es intentar, lo que veis aquí es lo que parece ser un bivalvo, esa parte blanca de ahí. Y bueno, lo que se estaba comentando de los fósiles vivientes, lo que aparecen un par de organismos al lado de lo que en español, o por lo menos en España, se conocen como cochinillas, pero bueno, aquí lleva otro nombre científico que desconozco. So you are looking at in the screen a cell that is a bivalve. Right now we going to try to recover that shell, and also two microorganisms that are next to the cell that probably are living fossils. We will try to recover and know if we are in the right way.
Bueno, para los que vengáis aquí por primera vez, esto que vamos a ver ahora es probablemente una de las técnicas de muestreo más divertidas de las que dispone el ROV SuBastian, que viene a ser una aspiradora. Entonces, para organismos, en este caso vamos a usar para un brachiopodus sesi, pero para organismos que se muevan o que tengan mayor facilidad para huir, no había nada dentro. Qué rabia. Bueno, para organismos que tengan capacidad de escaparse o cierta movilidad es lo más útil, porque con pinzas no lo vas a capturar, jamás. Bueno, si lo capturas, se te da un premio.
Yeah, we're just
Okay, thank you. Sí, sí, sí. No lo sé, o sea, estamos viendo a ver si no. Pero no sé, se ha movido todo cuando ha movido la. A lo mejor un poco más.
This is what you want here, sir?
Yeah. So the cell is not so important right now. Maybe we check the other two organisms next to the cell. That is the most important thing. So it's good for us, right, Amanda? Okay.
Okay. That's enough?
Sounds good. Yeah.
Okay.
El SuBastian, que es como se llama el rover con el que estamos trabajando, puede recoger 4 tipos de muestras diferentes, tanto biológicas como geológicas. Miguel, como os ha explicado un poco, ese elemento que ahora mismo están moviendo es una aspiradora que es capaz de absorber sedimentos y organismos que vayan pegados a ese sustrato y que recogemos en unas cajas que están insertas en el propio vehículo. También podéis ver ahí unos tubos de PVC blanco que nos sirven también para recoger sedimentos, muestras también del sustrato rocoso y algunos organismos. Y un poquito en la imagen abajo, por ejemplo, veis un número 8. Esos son push cores o con los que podemos recoger muestras de testigos del sedimento y del sustrato rocoso y poder recuperarlos hasta la superficie. Esos testigos, por ejemplo, ayer mismo se recogieron tanto por interés geológico como por interés del tipo de microorganismos que puedan existir y de restos biológicos que pueda haber en ese sustrato y que puedan vivir. También, si habéis seguido este stream durante un tiempo, es posible recoger algunos de los organismos de mayor tamaño vivos y traerlos aquí al Falcor, que es el barco en el que estamos actualmente, para que los biólogos se lo pasen muy bien. Ya. Miguel os ha estado hablando un poquito de los methane seep, los methane seep o los flujos de metano. Son muy importantes a nivel biológico en la Tierra porque son fuentes de alimento de organismos que viven en el fondo oceánico, pero también lo es a nivel astrobiológico. La astrobiología es parte de mi trabajo, como en el Centro de Astrobiología, que por eso lleva el nombre. Lo que hacemos en este centro es tratar de explicar un poco el origen y evolución de la vida en nuestro planeta, pero también en nuestro sistema solar, incluso en el universo. Es una ciencia multidisciplinar en la que trabajan desde astrofísicos, biólogos, geólogos, matemáticos, y que permite tener un conocimiento muy amplio de lo que es la vida, no solo a nivel biológico, sino para entender las condiciones para que esa vida se produzca. Y porque, por ejemplo, la Tierra es un lugar donde ha florecido tan bien tanta vida y tan diversa, y intentar encontrarla en otros lugares como puede ser Marte, que quizás sea el más conocido para la audiencia, pero también otros lugares como son las lunas heladas de Europa en Júpiter, que orbita alrededor de Júpiter, o de Encélado alrededor de Saturno. Como os he comentado, estos methane seep tienen esa gran importancia aquí en la Tierra porque posiblemente sean lugares donde la vida pudo florecer en nuestro planeta, pero también es otra posibilidad que exista en estas lunas heladas. Por eso los astrobiólogos consideran que Europa y Encélado podrían ser dos buenos candidatos para buscar vida en nuestro sistema solar, aparte de la que ya conocemos aquí. Uno de los aspectos importantes también de esta expedición es, a nivel geológico, es el ver un poco las condiciones de esas methane seeps para intentar extrapolar esos resultados a lo que podríamos encontrar en el fondo oceánico de Europa. Para a lo mejor los que no estén tan familiarizados con lo que es la geología fuera de nuestro planeta, tanto Europa como Encélado son satélites que orbitan los grandes planetas del sistema solar. Su superficie, en lugar de ser rocosa, está compuesta principalmente de hielo de agua, pero con sales. Las misiones que han ido a estos cuerpos o que han podido estudiarlo con cierto detalle, lo que han podido ver es que esa corteza oceánica con sales lo que nos indica es que posiblemente el interior tenga un océano de agua líquida. Hay otras pruebas que podrían ayudarnos a demostrar la existencia de estos océanos de agua líquida, que serían similares a los océanos que tenemos aquí, llenos de sales, mediante datos gravimétricos, magnetométricos, que si alguien lo desea en el chat se puede explicar en mayor detalle, y que aquí en la fosa de Atacama podríamos tener un posible análogo a lo que podríamos encontrar en el fondo de Europa o incluso en Encélado. So for the English language people, all the objectives of this expedition is to follow the similarities that we have here in comparison with Europa, the moon around Jupiter, and Enceladus, the moon around Saturn. As you know, these two moons are icy moons, moons where its surface or its crust is formed by icy water with salts in its composition. Under this ice crust, we suppose there exists an ocean of brines, water with different kind of salts, and under that sea floor of this ocean is probably that exists methane ships or another hydrothermal sources of where the life could be exist or even created, as we think that is created in the earth billions of years ago.
Can you see that clearly?
Sort of. Make it right down to the wire if you could. Perfect. And then just do one on the other side too, and we'll say in between is your best.
Other side here?
Yeah. Perfect.
Is that fairly obvious?
Not super obvious, but
But you can tell.
yeah.
I can go get a paint marker if you need.
I think you might have to, because all I see is the reflection of it, like its shadow.
Yeah, I'll get one.
Pictures.
And this is the main purpose of this specific transects. Today for those of you who may be interested, as I know live streams like this tend to hold people for a limited amount of times. The main purpose of today was to dive down to the deepest depth, close to a small seamount, and then slowly climb it up around 200 to 300 meters until we get to the top in the search for methane seeps. Sadly, we have not been successful, at least today, in finding these seeps, although we have found what do appear to be remnants of them. Right now we're closing in to checkpoint number 6, which should be close to the summit of this seamount, and then we will start descending again along the edge of the seamount. You can see a nice fish here, which has nothing to do with the main purpose of our expedition, but it's nice nonetheless.
En los vídeos también se han ido mostrando lo que sería la topografía de la superficie. Es una topografía que se ha conseguido a lo largo de la noche por batimetría. Es información original de la expedición. Esto nos ayuda a comprender un poco el aspecto del fondo oceánico y tratar de explicar, o tratar de buscar los posibles estructuras donde pudieran existir estos methane seeps o fuentes de metano.
También, tanto ahora como una vez termine la retransmisión, os recomiendo que volváis al inicio del vídeo y no hace falta que lo veáis entero, pero pasando 10 minutos, 10 minutos podréis ir viendo cómo ha ido cambiando la topografía del terreno, como bien ha dicho Oscar, a lo largo de este tiempo, pasando de zonas con sedimentos bastante consolidados y poca presencia de rocas a lo que nos encontramos ahora. También ha ido cambiando hasta cierto punto las especies animales presentes y similar. La temperatura varió muy ligeramente, 1/10 de grado. Desde abajo está 1/10 de grado más caliente aquí arriba. En estas zonas abisales la temperatura es extremadamente estable, ya que no hay muchos aportes energéticos ni geológicos que sean capaces de cambiarla. Ayer, de hecho, que nos mantuvimos en una temperatura mucho más constante, estuvimos a 1,82 grados durante 9 horas, de forma totalmente constante, para que tengáis una idea del grado de estabilidad que tienen estos ambientes.
Sí, bueno, como ha dicho Miguel, esta mañana el descenso inicial del ROV ha llegado hasta los 2.500 metros de profundidad y actualmente ya estamos sobre los 2.388, unos 120 metros de diferencia que se deben únicamente a ir ascendiendo una de estas lomas de una pequeña cordillera subterránea que tenemos aquí frente a las costas de Chile, a unos 50 kilómetros aproximadamente. Vamos a dirigirnos a esa plataforma más clara que veis un poco a la derecha, que posiblemente sea muy similar a una que hemos visto anteriormente.
¿Y qué consideras que podría ser eso, Óscar?
Bueno, la que hemos visto hace unos minutos, lo que parecía la superficie era una antigua zona de suelo marino, no al que tenemos actualmente, sino quizá mucho más antiguo, y que tenía restos de bioturbaciones producidas por los organismos y también estructuras que recordaban a los movimientos de las mareas. Y bueno, en el caso de los organismos que viven aquí, como podéis ver, sirven también como refugio de algunos de ellos.
El cangrejo. Entiendo que lo chupará, es que puede ser la suction chamber. Vamos ahora a tratar de succionar / adquirir el crustáceo que tenemos ahí. We're now going to try to suction and obtain the small crustacean that can be seen out of that small crevice in that rock formation.
Keep going.
Sorry?
We keep going.
Oh, you don't want it?
No.
Okay.
Al parecer, bueno, han hecho observaciones de lo que han visto y vamos a seguir adelante buscando los metaseeps, o por lo menos las estructuras o algún indicio de su existencia en el área.
Este crustáceo va a tener la suerte de ver otro día.
Sí, ayer, si podéis ver el streaming del día de ayer, fue bastante interesante para los que no somos conocedores tanto de la materia ni de los organismos que viven en estas áreas del océano, los sorprendentes que son, y para los astrobiólogos casi extraterrestres de alguna manera. ¿Verdad, Miguel?
¿Verdad?
So it may take some time to figure out what's happening. It's not right on the edge, but it's okay. It's still good. It's inside of the trench.
A ver qué son esas estructuras de ahí, Oscar.
Sí, vamos a ver un poco más de cerca qué es lo que tenemos ese afloramiento de ahí. Sorprendentemente, respecto al área de ayer, hoy tenemos un área más interesante quizá a nivel geológico. Lo que vemos ahora mismo en pantalla es muy similar a otras áreas que hemos visto previamente, con una pequeña zona superficial más oscura que vamos a ver un poco más en detalle. Ayer el recorrido, aunque también sufrimos variaciones en la profundidad, okay, thank you. Sí que la mayor parte del área que cubrimos estaba cubierta de un sedimento muy similar por todas las zonas. Hasta que alcanzamos una zona baja, donde empezaba el comienzo de una elevación en la que se podían ver algunos tipos de afloramientos de roca.
Okay, so Oscar, if you can tell them to get a piece of rock of this for me, and then if Aparna wants this as well, we can share.
Yeah, we can share the rock. This one, yeah.
Where do you want it?
Where?
If it's not too crumbly, it can go in the rock box.
Oh yeah.
Maybe the coral could come off in the rock.
Oh, that's true. Maybe to one of the bigger bio boxes. Maybe 3A, maybe.
Sí, Miguel lo que quiere es tomar una de las muestras de esa superficie de color más oscuro que tenemos ahí enfrente, junto al organismo blanco que vemos que sobresale por ahí. Es posible que su superficie esté cubierta de microorganismos muy interesantes.
Okay, that's good. En este caso vamos a tratar de tomar la roca. Tiene un coral. Vamos a tratar de tomar el sustrato rocoso que podemos ver ahí. Tiene pegado un coral que, aunque no hemos preguntado a nuestros compañeros biólogos, seguro que les es de utilidad. 2 por 1.
Sí. Hay que aprovechar todas las oportunidades que tengamos aquí abajo. El color de esa parte del material que vemos ahí, bueno, cuando llegue la muestra aquí al barco, a los geólogos que estamos aquí, que no estoy solo yo, quizá lo podamos ver un poquito en detalle, ya sea visualmente para intentar comprender si hay alteraciones de los minerales o de la roca, o si se debe a precipitación de algún tipo de mineral o incluso de microorganismos que estén alterando la roca.
Una pregunta, Oscar, como nuestro geólogo residente.
Dime.
¿Todas estas formaciones son sedimentos compactados?
Sí, la mayor parte de lo que vemos aquí y lo que hemos estado viendo a lo largo de esta inmersión son sedimentos que proceden de la zona continental, que está situada, como os he dicho hace unos minutos, a unos 50 kilómetros de aquí. Aunque estemos cerca del desierto de Atacama, aún así llegan sedimentos, no tanto como por ejemplo podría ocurrir en el sur de Chile, donde las precipitaciones son mayores. Y aún así, a lo largo de miles de años y millones de años, pues se producen estas acumulaciones que, en este caso, mediante la acción de la subducción y la deformación tectónica, ha producido la formación de la cordillera que hemos localizado en este área, aunque ya era conocida anteriormente, pero bueno, hemos podido hacer una batimetría de mayor detalle para conocerlo un poquito mejor. La tectónica chilena es bastante interesante, es un país que para los geólogos es como un paraíso a nivel de volcánico, tectónico, estructural en muchas partes y también geomorfológico. Nuestro compañero Javier, que está aquí en esta expedición con nosotros, cuando hemos realizado unos pequeños viajes antes de embarcarnos en el Falcor, bueno, iba alucinando bastante con todas las formaciones que se veían muy claramente y que para nosotros eran solo cuestión que veíamos en los libros y bien dibujadas. Aquí se pueden diferenciar perfectamente. Así que bueno, a todos los que sean geólogos o os apasionen la geología, venir a este país merece la pena. Ahí más adelante veis también otro de los afloramientos que, como Miguel ha comentado antes, son diferentes capas de sedimento que se han ido depositando en el área. Aunque la evolución geológica nos haga pensar que ha habido un empuje continuo de la placa pacífica que subduce bajo la placa continental de Sudamérica, en realidad son procesos de compresión y de extensión. Es decir, aunque el material se deforma hacia el continente, hay etapas en las que se produce extensión, que es decir, la placa continental va hacia el mar y hace que parte del material quede por debajo, vaya subiendo por debajo, vaya subiendo en diferentes ciclos producidos por los terremotos que se producen en el país.
Where are you going? As close to that as we can.
Yeah.
Maybe not on it, but next to it.
Just below it or?
Yeah, just below. Sure.
There's like a nice sedimentary spot there. It's pretty rocky, but do you want to do it like we saw Senator go out there?
Yeah. You could also take a piece of that rock. Yeah, well, we could keep going. If this doesn't look good. I agree, it's not as carbonated as it
Can we shoot here?
Vamos a tomar ahora un pushcore en esta zona,
Oh, I thought you meant the one that we just
en
passed.
concreto para el equipo de Loren y mío. Y luego posiblemente tomemos también una muestra de la roca que estamos enfocando en este momento.
And, yeah, let's take a picture. Actually,
Bueno, ahora mismo Loren está eligiendo un lugar donde
I'm gonna wait until we get a little bit closer to something that looks a little bit more carbonated.
Okay.
So I'll hold off on taking a core here.
Okay.
Unless you really want me to. Alright.
So
So we'll wait until we see this one. Yeah, I think you can move it along. Geologist wanna say something?
I do like that rock. No, I think we can go on.
You go? Yeah. For me now. Yeah, we can take a sample from here.
Pero pero Oscar, entonces yo también.
So Miguel and I we will share a sample for the formation that we saw near this cute animal here.
Este sustrato rocoso es maravilloso y por bastante de los más coloridos que hemos visto de momento. A la Biobox 3A también si quieres. I think it could go in there as well. If it fits. 3A?
3A. Yeah.
It should work.
That's the Evers. And then this one will drop out. You can see it is still up there. So.
Yeah.
So I'll let Zach tell about it. There's the ones in the niche. Which are both direct to me. Coming up on 3. Carbonates and bubbles if you see them.
So keep going. We can follow this structure to show what is, a little bit to know. Lo que tenemos enfrente, esa formación rocosa que tenemos enfrente, podría ser algún tipo de fractura. Vamos a seguirla durante un rato para ver qué es lo que qué sorpresa nos puede llevar. Vamos a acercarnos un poco más al afloramiento rocoso que vemos ahí adelante, con esos fragmentos blancos, para echarle un vistazo un poquito más de cerca para ver si podemos reconocer algo a nivel geológico. No sé si Miguel nos puede explicar algo a nivel biológico para no aburriros tanto con mi tema favorito de conversación.
A priori, sin llevar a cabo ningún tipo de estudio previo, es difícil afirmar exactamente el momento que me estoy extrayendo con el bicho ese que hay arriba. Hacer una observación. No sé qué es eso.
Bueno, arriba en la pantalla estamos viendo algo.
Sin llevar a cabo ningún estudio previo es complejo saber nada de microbiología, y creo que ahora lo que quiere que sea esto es bastante más interesante. Sí, sí.
Bueno, como veis hay animales, organismos fascinantes, sorprendentes. Ya, ya, asumen. Vemos un pequeño cangrejo ahí arriba y algunas cosas interesantes superficiales del afloramiento rocoso. Ya. Vamos a seguir un poco más hacia adelante. Aparna, una de nuestras compañeras, nos ha pedido recoger una de los fragmentos blancos que vemos ahí. Aparna, one of our colleagues, will request one of the white pieces that we can show in the screen right now. So a quick description, only what we view here. We can show a upper layer of maybe sand or something very similar.
Do you want the rocks on the ground?
Yeah, that's one.
So the ones right on the sea.
It is possible. I don't know.
They're pretty smooshable.
Como veis, el material es bastante suelto, parecido a arcillas, arenas, quizá una mezcla de ambos tipos de rocas.
Can we try a coral quiver for this?
Coral quiver. Is there?
I'm not sure we have one open.
Oh, okay.
Yeah.
Thing is, we don't have too much.
Have to go in the box.
Okay, you can put it in BB3B if you want to, but I don't know if it will disgregate.
BB3B. Alright. Let's see if the torch is gonna switch this. If not, I gotta back that up.
Aunque ahora mismo estáis viendo lo que estamos trabajando con el SuBastian, que quizás sea lo más visualmente interesante, también desde el barco podemos hacer una recogida de aguas a diferentes profundidades. Es muy interesante la recogida de estos volúmenes de agua para ser estudiados, principalmente filtrados para determinar el contenido de pequeños organismos, rocas o incluso elementos muy diversos que se pueden localizar dentro de las aguas. Nuestros compañeros trabajan bastante duro filtrando esas aguas para obtener información que puede ser muy interesante a nivel científico.
Mira, una gotina colorada.
We return to go to the point 6. Yeah, 6. Bueno, a Miguel antes ha aportado una información muy valiosa, que es el tema de la temperatura. No es un tema baladizo a dejar de tener importancia, ya que cambios en la temperatura también nos puede indicar la presencia de fuentes hidrotermales, por ejemplo. Y en este caso hemos aumentado a unos 0,20 grados centígrados la temperatura en aproximadamente 120 metros de diferencia de cota. Desde esta mañana me refiero. También, si os habéis estado fijando, bueno, como también ha comentado Miguel, ayer estuvimos durante prácticamente las 8 horas que duró la inmersión con una temperatura bastante constante. En este caso, en los últimos minutos, la temperatura ha aumentado prácticamente una décima de grado, que en estos entornos y a diferencias de cota muy pequeñitas, pues resulta muy alta.
También llamativo que, aunque a ojos de personas que quizá no estén muy acostumbradas a transitar o a ver lugares como estos, entre los que yo creo que nos incluimos buena parte de la tripulación científica, puede parecer extremadamente desolado este ambiente, pero como ya habréis visto, los que lleváis aquí un rato, es extremadamente rico en seres vivos. Dime tú en qué paseo por el campo en cualquier sitio del mundo ves la densidad de organismos que vemos aquí, que cada movimiento de cámara nos desvela un pepino de mar, un crustáceo y todo tipo de criaturas. Entender también que una parte del ecosistema de aquí depende también del aporte de lo que se llama la anidre marina, la caída de materia orgánica en suspensión que va cayendo de las capas más productivas del océano.
En los mapas que podréis ver en pantalla estaréis viendo que estamos casi llegando a la cota máxima de esta loma subterránea. Miguel, como biólogo, ¿qué es lo que más te sorprende? ¿La cantidad de vida que hay en este fondo oceánico o la desolación que hay en el desierto de Atacama?
Como bioquímico...
Vale, como bioquímico, ¿qué te sorprende más?
Me sorprenden ambas, porque no somos muy dados a la macrobiología. Entonces, esto de estar aquí viendo organismos de 20 centímetros, de 30 centímetros, es algo totalmente inusual para mí. ¿Qué me sorprende más? Probablemente me sorprenda más la extrema aridez no me sorprenda, pero me inquiete, me interese más la extrema aridez del desierto de Atacama. Porque esta zona tan extremadamente árida las zonas áridas son abundantes en la Tierra, pero no son tampoco omnipresentes. Pues tienes el desierto de Sahara, tienes la península arábiga, el Gobi, el Namib, tienes desierto de Atacama, pero buena parte de la tierra está cubierta de vegetación. Entonces encontrarte de golpe, y sabemos bien los que hemos estado unos días por aquí explorando, encontrarte de golpe con la extrema aridez del desierto de Atacama, en el que pues, puedes conducir 200 kilómetros y no ver ni una sola planta, ni una brizna de hierba, nada, es abrumador y incluso terrorífico imaginarse qué pasaría si todo el mundo fuera así. Mientras que este fondo marino, aunque es cierto que es extremadamente estable, sí quizá puede ser más extrapolable a lo que se puede encontrar en otras partes del mundo, a nivel genérico al menos.
Y a nivel bioquímico, la extrema aridez del desierto de Atacama, ¿qué desafíos trae para tu campo de experiencia y sobre todo para los organismos?
Depende de en qué ambiente. Si tu campo es la microbiología, el desafío obvio es que no hay casi nada. Vamos a tomar ahora un core aquí,
sí,
de Loren, que tiene interés en esta zona, en la zona ahí con sedimentos más negruzcos elevados que podemos ver hacia adelante. Esa zona parece ser de interés.
Sí, me recuerda un poquito a
la milla que compartimos muestras.
Sí, nos recuerda un poquito a una zona que hemos visto en anterioridad, que hemos seguido durante unos minutos.
No, I want this white crust.
Vamos a intentar tomar un testigo de la zona que ahora mismo vemos en la parte inferior de la pantalla principal del SuBastian.
Oscar.
¿Sí?
El cobre, sí, 6, ¿no? 6, ok.
Good job. We're going to take a sample here. So, awesome. Great.
Okay. Can we use 6, 7 o 8 core?
6.
6.
Good job, you on there. Starboard.
Por si os hacéis la pregunta, nosotros no somos los que controlamos el rover. Hay un... El ROV. Hay gente del Smithsonian Institute, que es quien es el dueño y quien ha facilitado que estemos nosotros aquí haciendo esta expedición y este streaming, que manejan a la perfección este monstruo cuando lo ves en la superficie, que es increíble poder estar cerca de una creación como esta, que es capaz de llegar hasta los 4.500 metros de profundidad.
Los pilotos tampoco están manejando el ROV de forma continuada. Hay no sé si 4 o 5 y se van rotando a intervalos de, si he visto bien, unas 2 o 3 horas.
Sí. Bueno, hay un comentario sobre la columna de agua y no soy quizá el más apropiado para hablar de ellas porque no es mi campo de experiencia, pero sí es cierto que hemos ido encontrando cosas bastante interesantes a nivel de análisis. Un poco de interés de cómo se hacen esta recogida de muestras. Primero se baja una sonda en la que van cargados los tanques donde se van a recoger el agua. Esa sonda va cargada de una serie de sensores que van recolectando información sobre la conductividad eléctrica, la salinidad, la temperatura, y a partir de esa información la gente que está interesada en la recogida de esas aguas decide los puntos donde quiere recoger esas muestras. Los intereses pueden ser muy diversos, desde el querer filtrar para determinar la presencia de microorganismos como pueden ser el plancton o incluso pequeños foraminíferos, pero también otros a nivel geológico como puede ser la determinación de la turbidez y ver qué elementos forman parte de esa turbidez. Una de nuestras compañeras en el Centro de Astrobiología está interesada también en la posible presencia de virus dentro de estas aguas.
Así es, vamos a hacer un shout-out a Mara Laguna,
sí.
Que va a presentar la tesis dentro de muy poquito, dentro de menos de un mes, si no me equivoco.
Sí.
Y buena parte de los estudiantes del CAP vamos a ir ahí a apoyarle y a darlo todo, que lo va a hacer súper bien, seguro.
Estamos convencidos.
Sí. Y estas muestras, te seguro que va a conseguir un montón de virus submarinos, serán para ya su postdoc o lo que ella considere.
Exacto. Miguel, ¿quieres que hablemos un poco de por qué tenemos interés en los virus?
No.
¿No? ¿Por qué?
Sí.
Vale.
Vale. El interés viene en parte de que cuando Armando propuso esta expedición intentó buscar un equipo multidisciplinar y Mara es no la única bióloga, pero es una de las 3 biólogas que tiene el CAP en el mismo grupo y tiene el conocimiento necesario para poder encontrar virus en estas condiciones. Entonces, lo que pretendemos es encontrar tanto, hay una gran algarabía porque estamos consiguiendo adquirir el core de forma bastante efectiva. Pero bueno, si esperamos que tanto los microorganismos como los organismos de estas zonas sean antiguos, es también de esperar que el mismo caso se dé para los virus, que puede ser también una vía exploratoria bastante relevante. Oscar,
¿sí?
Drew va a querer otro core aquí.
Okay. So we can check a second core here in the same area when you finish with this. Core 7?
Yes. For Drew.
Oh,
For
okay.
Drew. Drew wants another core of this area.
Está teniendo lugar ahora un efecto llamada de cores y todo el mundo quiere uno aquí.
Como veis en el testigo que están subiendo, algo que también nos ocurrió ayer y que hemos conseguido recoger algunos de los organismos que viven en la superficie del sustrato. Si our colleague Nick maybe can answer better the question of what organism is that, but I think that is foraminifera or something like that. Miguel, you know what is that organism?
No. Yes.
Well, Miguel is a little busy. Uno de los efectos que tiene el levantar o el recoger uno de los cores es lo que veis en la pantalla, que se levantan una cantidad enorme de sedimentos que empiezan a flotar. Gran parte de ellos es también materia orgánica, que nos impide un poco ver mejor lo que tenemos enfrente del SuBastian.
for Drew, and the following one for Daniel.
Okay, we need the next one in the same side. This is the who scored 7.
Voy a hacer un pequeño resumen de para qué usar cada persona cada core, ¿te parece?
Perfecto, Miguel.
Este primer core es para Loren y para mí. Con estos muestras vamos a hacer un análisis conjunto, ella va a hacer la metagenómica, yo la metatranscriptómica, ya lo he explicado antes, pero el objetivo de la metagenómica va a ser conocer los genomas, es decir, toda la información genética de todos los microorganismos que hay aquí, mientras que la metatranscriptómica yo me voy a centrar en conocer cuáles son las proteínas y los distintos mecanismos y metabolismos que se están expresando en un momento concreto. Y juntando las dos aproximaciones nos permite tener una idea muy buena de las comunidades que tienen estos sedimentos. El siguiente sedimento es lo que llama Andrew Palmer de la Universidad de Florida Tech, cuyo objetivo es buscar nuevos metabolitos en estos sedimentos para todo tipo de aplicaciones. Y el último es para Daniel Carrizo, también del Centro de Astrobiología, que se va a centrar en la búsqueda de biomarcadores lipídicos en estos sedimentos, entendiendo estos sedimentos como análogos de lunas y similares, ya que Daniel se centra en el estudio de biomarcadores lipídicos en distintos tipos de análogos.
Ahora la actividad aquí en la sala de control es bastante excitante, cuando hay que recoger muestras. En ocasiones vienen los propios investigadores para seleccionar los lugares donde quieren elegir una muestra y donde recogerla. Aunque penséis que estamos solamente la gente que opera el SuBastian aquí y Miguel y yo, en realidad hay bastante más gente que está entrando, saliendo o bien permanece durante un tiempo con nosotros, viendo todo lo que sucede aquí en la sala de control para, hacer una observación o hacer una recogida rápida de algún área de interés. El barco está lleno de salas, laboratorios, camarotes, como el comedor, por ejemplo. En todas ellas, en todo momento, cuando el SuBastian está haciendo una de sus inmersiones, está en la pantalla puesta lo que estáis viendo ahora mismo vosotros. Y en el caso de que alguien necesite hacer una recogida, puede bajar inmediatamente a la sala de control o comunicarse con nosotros para que recojamos alguna muestra que puede ser de interés.
Oscar, hago un resumencillo en inglés.
Okay.
Okay, so I'm going to quickly summarize what we've been saying for the past few minutes in English for those of you who are English speakers. We've arrived at a really interesting outcrop, and many scientists want to get postcores here. The first one was dedicated to Loren Seiler and me, and we're going to do a joint metagenomical and metatranscriptomical study of the sediments to basically understand with very good quality how the microbial communities in these sediments work, basically. The next sediment, the ones that we should be taking in a few minutes, is going to be directed to Dr. Andrew Palmer. Is going to be focused on the search and discovery of novel metabolites. And then the next one, which should be score 8, it will be centered around Daniel Carrizo, another scientist at the Center for Astrobiology in Madrid, Spain, which will be used for his main line of work, which is focused on detection of lipid biomarkers for space exploration.
Also to this course, when it's here in the seep, from that time to in maybe 4 or 3 hours. Also, we do a geological description of that, because the information that the geology of these sites is very important also for me as geologist, but also for all the researchers that are on the seep, for connect geology and its fields of expertise. Cuando los testigos lleguen también al barco, se les realizará una descripción petrográfica lo mejor que se pueda, porque bueno, como comprenderéis, no son testigos que lleguen de una forma firme en todos los sentidos, ya que llegarán con una cantidad de líquido importante y en algunos casos incluso muy como si fuera una especie de gelatina o de un líquido muy poco denso. Esta información es relevante tanto a nivel geológico, pero también para el resto de investigadores que están en el barco, porque podría representar la conexión de la geología con alguno de los campos de conocimiento que tienen ellos y que desarrollan en sus carreras profesionales. El SuBastian como carga con 17 push cores actualmente, que pueden recoger hasta 35-40 centímetros de testigo al presionar sobre la superficie, según el material sobre el que pretendamos atravesar. El que queden dentro retenidos de los tubos de metacrilato que tenéis ahora mismo en la pantalla se debe a un proceso de presión inversa, lo que hace que no se salga el material, aunque pueda parecer contraintuitivo. Cuando regresa al ROV se le pone un tapón de goma que está dentro de los contenedores, y al llegar de la superficie podemos tener el testigo completo sin ningún problema. Esta exploración no se desarrolla solo por el CAP al que pertenezco, sino por diferentes instituciones y centros, tanto en Estados Unidos como en Chile y en España. En ella hay diferentes investigadores de diferentes campos de investigación: geología, biología, geoquímica como Miguel, como Javier, o bioquímica como Miguel. Y bueno, como podréis comprender, todos tenemos diferentes intereses, pero que en algunos casos se enlazan muy bien para obtener objetivos bastante interesantes. También el llegar a estas zonas, explorar estas zonas, tiene un pequeño índice de incertidumbre sobre lo que te puedes encontrar aquí abajo. La mayor parte de las veces en zonas tan desconocidas para la mayor parte de la comunidad científica, las sorpresas son en el lado positivo.
Just gotta settle here again,
Yeah.
then.
That one of them is for Drew. So that's not as deep as you want, right? Right. Are you okay with that spot? If we just take it? Yeah, it's a little more than 5 centimeters. Right, so she could try again. Okay.
Yeah, I can work with that. Yeah, good.
Okay, cool. It's the same area. Yeah, and then there's still extra frame.
Bueno, como habéis visto en este pushcore, a veces, aunque el terreno pueda parecer muy suelto, no siempre cumple con las expectativas.
That's his favorite part, so I'm really fast today.
Miguel, ¿te recuerda algo esto a la misión InSight en Marte? La incapacidad que tuvieron para penetrar bajo la superficie marciana.
Me puede recordar, pero honestamente preferiría que no me recordara.
Vale. Por si estáis interesados, el Centro de Astrobiología, que está situado en Madrid. Un momento, por favor.
Y luego te toman el Daniel.
Excuse me. Can you take a zoom here, this area, first?
Take a what?
Zoom in here.
Zoom in?
Yeah.
Alright, hold on 1 second.
Nos han pedido hacer un pequeño zoom en esa zona de ahí. Armando. ¿Se ve? ¿Algo interesante? No. Keep going with the course.
Course.
Yeah.
Course. Alright, man.
Ok.
Number 8.
Habían pedido un pequeño aumento de esa área por si era algo que pudiera ser de interés. Bueno, como comentaba, el Centro de Astrobiología, que está situado en Madrid, al que pertenecemos varios de los investigadores que estamos aquí, tiene un alto componente de investigación espacial. Uno de los investigadores al que habéis podido oír durante la mañana, que es Antonio Molina, participa en alguna de las misiones espaciales de NASA, como por ejemplo Curiosity y Perseverance, y el propio Centro de Astrobiología en la misión InSight y también en las dos que he mencionado, Curiosity y Perseverance, participa y ha participado en InSight, ya que es una misión que ya no está en funcionamiento en Marte, proporcionando las estaciones meteorológicas de estos exploradores del planeta rojo.
Este core número 8 se va a tomar a la vez que Madrid ha marcado gol, así que será en su honor.
This one a little bit closer to us, I think. This is gonna be deeper.
How's this one?
Yeah, it's a good.
Ready? Alright. I'm ready.
Como habréis comprobado, Miguel es multitasking, puede estar tanto haciendo ciencia como comprobando el partido de la Champions.
Y eso que no nos gusta demasiado. Bueno, ahora estamos tomando un pushcore en esta zona, ya que todo el mundo quiere uno. Este en principio, depende de la longitud, irá Daniel Carrizo, pues será uno repetido para Loren Seiler.
That's okay.
You want this? You want the duck in? Save the core.
I'll let save it. Because there's another short one that I can use instead.
So you want this?
No, you can dump that one. I can use it.
No, it's close on the side.
Yeah, yeah.
Cool.
Drew wants one, but I don't know how much does Drew need?
Drew has one already.
Oh, he already has one?
Yeah,
Okay.
we need Daniel's in yours.
Oh, Dan's, yeah,
And that's
okay.
it. Daniel could probably have used that, but he would appreciate a bit more.
Okay.
So like 5-6 centimeters should be enough.
Como nos recuerda Guillermo, las estructuras que veis ahí en la roca parece ser que pertenecen a animales excavadores. Una de las posibles salidas del metano que pueda encontrarse bajo la superficie son rocas porosas o rocas bien muy fracturadas, como la que tenéis ahí enfrente. Nos hemos parado en este punto porque es posible que estemos ante lo que era un antiguo coal seep, por lo que el interés biológico está al máximo. La otra posibilidad es que es parecido a lo que tenemos aquí en este área, es la formación de un anticlinal. En un anticlinal es una estructura geológica con forma de U inversa, en la que el material, en este caso los gases, podrían quedar retenidos y que podrían escapar mediante también fracturas que podrían incluso ser provocadas por la propia presión del volumen del metano o de cualquier gas que se encuentre retenido. Un análogo muy fácil de comprender sería las zonas donde se produce la acumulación de petróleo, que sigue el mismo tipo de estructuras geológicas. También la formación de anticlinales, donde el petróleo más o menos líquido y los gases quedan retenidos por rocas menos porosas.
Yeah, sure. Too much longer.
It's still dropped, but it's holding a lot better.
I don't want that. Thank you.
Este quizás es uno de los momentos más excitantes del día, ya que hace aproximadamente 8 horas llevamos trabajando en esta inmersión y estamos ante la posibilidad de haber encontrado lo que pudo ser un methane seep. Ahí vemos que el core parece que está entrando muchísimo mejor que en los anteriores puntos. Y bueno, hasta que no lleguen a nuestras manos no sabremos exactamente con toda seguridad lo que hemos encontrado.
I'll take this one and you can give 6 to Dan. This one's deeper. Because Dan doesn't need that much material, right? Yeah, take this one. And then a piece of the rocky substrate there.
Keep moving. Just move down west. Just gonna hold it right here.
That's fine. That's good.
Thrust.
parece que el pus core sale relativamente bien. Aunque veáis esos brazos que parecen duros y con una fuerza increíble, están tratados con una delicadeza que os sorprendería.
Yay! The ones in future king.
Yeah. Alright, force going up. Right underneath the
Thank you very much. No, that's fine.
Lauren, do you think that is a methane ship?
I don't think it's a methane seep. I think it might have been at one point, but it is not anymore.
No, actual, but maybe in the past.
Maybe in the past.
Yeah, we are talking about with Lauren, that is a co-IP of this expedition about this site. It's probably the actually no is a methane ship, but maybe in the past was.
Still have not found bubbles. But we found what might have been seeps previously.
Yeah.
We're not sure.
Otro de los aspectos de hacer la batimetría es la búsqueda de las fuentes de metano a través de las columnas de burbujas que fluyen de estos lugares. Una analogía sería como si tratáramos de buscar desde la superficie del océano la presencia de un humo que sale desde la parte inferior, desde el suelo del océano. Como si veis una chimenea o una factoría, veis que aunque el humo sale de un punto inicial, se va extendiendo hacia arriba y ampliando su volumen. Si intentáramos detectar el punto desde el que parte ese metano, sería imposible por radar y batimetría. Por lo que se busca es la presencia de esos volúmenes de metano en esa columna de, llamámoslo, humo de metano dentro de la columna de agua. Esas acumulaciones de metano tiene una respuesta mediante radar muy diferente a la que tendría el agua o lo que tendría la parte inferior de los sedimentos, porque podría ser detectada en buenas condiciones.
This is where you want Lauren, right?
Yeah, just a check of that, please.
Any of it, right?
Yeah. No, thank you.
If you look at your SIP camera, you're still using it.
So why don't we come and do one, is we can go through that, but no out of chop, and then we take the pieces.
Sounds good, yeah.
Start coming out.
Do it up.
I'm not ramming out. Do you want to get that photo? Five is open still.
You want that sample, Lorraine?
I'll get you that.
Yeah.
Should be good.
I'm just curious.
Maybe geologists can take a sample for the outcrop
Yeah.
for everyone.
Sure, yeah. I don't need a big piece. I don't know how big the challenge is.
If the rock is soft, I don't know if the push core can penetrate it under the rock.
Yeah.
It's not enough. Okay.
Yeah. Just get a good amount of pieces Geobiology
Yeah.
I'm allowed to sing songs I made up, right?
Oh,
Is that okay?
Yeah, all right, I'm gonna swing you over a little bit here, man.
the carbonates.
Mm-hmm.
That's maybe when we have the sample with Crinoid, with Crinoid acid, we can do a quick.
Cool. Yeah. I was thinking maybe I could grind it up, extract some DNA from it.
Okay.
I don't know if there's anything in it.
I only piece, so we explorate to notice these carbonates.
Excellent. Thank you.
Okay, we'll do the scan too. Okay, let me know if it's
It's
you can
very
see
similar
red,
to
the red one.
carbonates.
Not too
A
bad.
very
A little bit more. Come down a
carbonate.
little bit. Okay. Push in a little bit.
El afloramiento que estemos
Nice and easy.
viendo ahora mismo podría ser algún tipo de carbonato.
Alright, looks like you might have it.
En él habéis visto que,
You definitely got it now.
que viven los organismos de este fondo oceánico. Wow.
It's alright, you just need to
Como veis, no es algo fácil recuperar muestras del sustrato rocoso y de afloramientos como este.
keep pushing in.
Maybe they could try the scoop instead of the claw.
Yeah, maybe better.
Maybe that would be better.
Maybe scratch and later take it.
Or they could just
More?
use the
A little
Yeah.
more? A little more? Looks like you got it there. And it comes straight back so you don't rush. Looks like it's dangler. Alright, let me get you set for you so like that. It's good for you, Lauren?
Yeah, that's great. Thank you.
Got it, okay, looking there, it's working.
It's a nice hunk of rock you got there.
A veces para los geólogos cuando estamos en el campo nos es difícil diferenciar algunos minerales o rocas o diferentes elementos o la presencia de algún tipo de mineral. En el caso de la muestra que estamos recogiendo, podríamos tener la duda de si es una arena o una arcilla o bien un carbonato, por ejemplo. El carbonato, una manera rápida que los geólogos tenemos de determinarlo en el terreno o bien en el laboratorio, si no tenemos otras herramientas a mano, es poniendo una pequeña gota de ácido clorhídrico que al reaccionar con el carbonato produce un burbujeo. Por el proceso de degradación del mineral.
Anything else at this site? We'll continue on.
Continue more for here.
Straight ahead?
Yeah. Also, no, maybe there to know what is follow the structure.
Okay.
Okay.
Are you good with the scan?
Arriba. ¿Cuál dices? Okay. Todas las roquitas negras.
Follow this fissure. This break right here.
Yeah, follow that. And later, how long we take to return to one of the sites that have a lot of rocks?
Rocks?
Yeah, I don't know. We passed, I don't know how many times.
15 continue on here, or are we heading back the way?
Okay.
No, which would you like to keep on?
Keep going
Okay.
here, solve something of the structures, and maybe return to the one of the side. Yeah, we pass. We pass it.
We pass?
We pass it. We return to the same route,
Oh.
just to looking for one area that Armando is interested. But right now we follow this side and return.
So we'll just go on this for a while. And then
Yeah,
we'll
for a
tell us
while
to
and
go back.
later
Okay.
return.
Oh,
Yeah.
okay.
Sorry.
No, it's all right.
Okay.
Dead ahead, there's some feature.
Yeah.
Well, yeah. Something to track on.
AC.
Yeah. Dark blue.
Vamos a continuar por esta estructura que parece un anticlinar para ver un poquito más por dónde sigue y posiblemente en unos minutos regresemos.
Once we reach 6.1, we're going to follow along this bridge here.
We follow the anticlinar
Yeah.
to try to find maybe another site
Yeah.
of interest.
Yeah.
Even we could take any sample with any
Yeah.
curve or self.
Yeah. We're going to go another hour, and then the last hour of the dive, we'll
Okay.
just take what they need to take.
Okay.
And maybe I'll take another core.
Yeah, so Amanda was about to return for one of the sites that is a lot of rocks and maybe have a... ¿Por qué Armando? Biodiversidad o algo? To return to the field, the rock field is, or something in special? Yeah. Sí. Ah, es para los micros, luego. Y la gente de la zona de lo que hemos pasado. Okay. Okay, so first we take time here to looking for the structure and later to the 6 o'clock.
6 o'clock,
Return
yes.
to one area that is to interest by the biologists.
6 o'clock, you can go wherever you want.
Okay, maybe 5.5 we
No.
can start to return?
No.
No?
This next hour belongs to me.
Okay.
So 6 to 7 for biology.
Got it. Torch coming in. Let's see if we got it. There we go. Alright. Just let it dangle down. Okay. Yeah.
Stay here, this area. Maybe check out the score here for Geology. Whose score? What number? ¿Qué número de tu score? ¿Qué número de tu score? In this area, it could be possible.
Let's see. Might be a little rocky, but we can try.
And right here, a little bit separated of the outcrop. Yeah.
So
maybe here. It's okay. Here, around here. Okay.
see what we can do.
The number 9. It's number 9. Okay.
Right.
La parte de geología de esta expedición vamos a tomar un testigo en este área, un poquito separada de las anteriores, pero que posiblemente tenga interés.
Might be rocky, but we can try.
Yeah.
Okay. I'm gonna do a poke first. You can do a scientific poke? Yeah. Alright. Your second favorite thing. You can use port. Nope. Yeah,
It's not like this deep.
yeah, it's only about
Not even.
this
No,
deep.
it's deep. Okay.
Yeah.
See it? Ah, okay. So keep falling a little bit more.
Yeah, sure. Sure. Alright, coming up.
Al final no es posible coger un buscador de ahí porque el sustrato rocoso que veis oscuro ahí está a unos pocos centímetros bajo la superficie del sedimento.
Let's do it.
So we go to continue to explore a little bit more this structure, following an active methane ship, or also the keys to find it. Here. We can try here at the score.
I don't know. We can try.
We can try.
Yeah.
This. We can try at two other push core in this area if possible. Again to Geologists. Also we have the hard rocks near this place, but
That's very shallow too. Yeah, this looks like it's also going to be rocky. I can poke. You can poke though?
yeah.
You want to poke? Oh.
Yeah.
Here.
We want to check it as the site before is rock near the surface, so rocky too.
It's pushing so it's pretty hard.
Can you scratch the surface a little bit?
You want to scratch it like
Yeah, only to show how it's under the surface a little bit.
dig a hole.
Okay. Thank you. Oh my.
Yeah, it's really hard.
Oh, yeah. Yes, wait there. Can you zoom it? Oh, the material on the arm? Yeah.
Yep.
Okay. geology feature, but yeah. Okay. We can continue.
Continue?
Yeah.
You want me to throw this?
Yeah. It's good to know. We take a sample for the surface to know how it is, the color and maybe granularity or something that we can recognize, because the color of the surface always is altered, so we need to check what is under that thin layer over there. So the aspect of the sample is very similar as we'll look in one of the cores that we checked before.
We're just trying to fix this. It was dragging.
To check another?
We just, the hose is loose, so we don't want it to get caught under anything or break.
Okay.
So real quick. Just screw it right up. Gotcha. Right on the plunge of that blade. Get in. Beautiful stuff.
Why didn't you tell me? Because I didn't know. Just don't.
You ready? Alright.
Is it okay?
Yeah.
Okay.
Thank you for the pause.
No, don't worry.
Just don't want to damage it. Alright, here we go.
Okay. Here in this area, you can see the diversity of organics are less than areas that we passed before in this dive. Here the rock layer is very close to the surface that the other zones that we pass in this day. The layer of sediments in other zones is more white than here. Here we have all the rocks of the surface because we are also in the top of the mount that we is today or field of work.
Can't
anything good on the sonar.
Are we heading across in the mapa?
Yeah, we can follow a little bit more. Keep going a little bit more to find some place that I don't know, to take another sample, but I think this could be very difficult. Maybe a rock sample or another kind, because I thought that the push core will be very difficult.
Yeah, it's pretty
Rocky.
thin sediment.
Yeah, yeah.
You can see the map. It will stop here, and this is it, and then we're going back.
Okay.
Do you even want to go here, or you can go back already heading Palo Norte? See?
Okay, maybe the idea is good to return to the main objective and go there for a while to know if there are something else, and maybe return because the biologists need to recover samples in points before. In maybe 40 minutes, but we had time to explore this area and maybe find something.
So you want to go here?
Yeah, in that direction, please.
Okay.
Thank you very much.
So that is about 350 meters away. So
How much time take it to you?
Oh, what's it say? I can't read it.
it's about 300, yeah.
300 meters.
Yeah.
It's too long?
No, we'll go there as fast as we want.
But then to come all the way back past the ship is going to take a lot of time.
How many?
You can go really fast, but you're not gonna. You might miss
Okay.
so many things.
So maybe the best idea is return to 0.6, no 0.61, 0.6, and later return to the previous sites.
Okay. And do you guys know about where the previous sites are?
The biologists could tell you better than me, because it's where the biodiversity are better.
Okay.
I'm gonna start moving the ship towards 6.
Okay.
Speed up a little bit.
Yeah, because it will take time to get back.
Okay.
There, so we might run out of time.
So maybe we can go around here and return?
Yeah.
Okay. So right now we need to reconfigure our routes of work to try to recover more information, more samples, and return to one point that for the biologists are very interested for the biodiversity of the area.
Bridge updated waypoint.
Here we prepare the day before. We prepare the actions for the next day. We prepare a route, a point where we'll come for, and sometimes the plans go right and sometimes the plans go wrong. But also the information that we recover in one of these dives always will be interesting.
do it.
We passed for this area before?
Yes.
Okay.
Yeah, so up here.
Yeah. This is the two cells that we see before. Okay.
It's pretty flat here. So probably dominoes. Alright. Loogie Ben? It's Loogie. That's funny. Loogie. It's pretty flat, huh? It's only got about 30 wall. Range it out any further?
Could we step here and take a score maybe here in this area?
Yep.
Oh, okay.
Oh, we can
Near
look
here.
at that. Yeah.
Here and try to recover the cell at the same time.
We can definitely cover that. The score might be tricky. It's pretty rocky, but we can.
I know. Try at before if there are enough sediment or
You want to recover that? See,
the cell. Yeah.
the white one?
The one.
Where do you want to put that?
It's score 9. Push score 9. It's push score 9. Yeah.
Oh, but no, the white one you don't want. That one. You guys want the shell too, correct?
Oh, this is not cell. Okay.
Or what? You want that?
No, it was a cell, but it's not. No. Only a core from here. It's the sedimentation is enough
Gotcha.
to take us.
Gotcha.
Okay. I thought that was a cell. So in this point.
We're going to do a push score here. If you wouldn't mind taking a pause.
So in this point, will we try to recover a push score for geology? I say all the two sites before we will check where is the rock and if it's possible to recover a core. Free and roll.
It's rocky. I can see it.
Impossible.
It's pretty rocky.
Rocky.
Yeah, let's try it here.
Okay.
He's gonna
So.
get another scientific look. Yeah, that's even. That's even worse. You think? Push it through? Yeah, let's give me some. Your sample's gonna be too steep.
How many? 3-4 centimeters? No, it's not like that.
You want to keep moving?
Yeah, keep moving with the cycle.
Keep going.
Keep going. So again, it's not possible to recover a good sample of core in this site. So we keep going, explore this area.
Very darky. We're moving on. On the move. You want to try here? This looks pretty flat. Can you look at it? See it?
Here?
Yeah, maybe.
Maybe here is softer?
Yeah.
Yeah.
Here?
We can set first and see if that's possible. Near this one white stuff.
Bridge, we're gonna try and get a push cord.
Understood, coming to stop.
Will we take a look at this?
Hey, you wanna
Up there?
torch?
You say
Yeah, buddy.
right here?
Torch kinda.
It's the point, this is the direction. So from here to point 6.1 is 300 meters. But we'll take a look. So you need to go here? Yes. You will? Okay. So maybe let's go here and maybe take a sample here and a score here.
Let's go here and then continue on the
and
sides.
go next to the premises.
No problem.
So we'll try to recover a boost score here for Geology. Maybe cross your fingers to have a good sample.
You're going to do 9, right? Correct. 9.
Yeah, I put score 9 and so here we go.
You ready? Are your fingers crossed?
Finger crossed, please.
Alright.
Everyone at the same time.
Everyone in the universe. I'm ready, Ben. You ready?
Go, 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 go.
A little bit of down thrust, a little bit of back thrust.
Wow. Keep going, keep going. Keep going a little bit more. Yeah, 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 yeah.
Keep it straight, you might get it. A little bit to the right.
Thank you, Lisa, for cross your fingers.
More to the right. Just gonna match it. Nice and easy. Nice and easy. Nice and easy.
And the surface of this side is a little bit sandy. A little fragment of rocks that probably are volcanic rocks for its black color.
And max down, so you are picking up that, maybe as far as we can get. Again, it's as far as we can get.
Ooh, that's interesting.
Yep, that's it.
We can see at the top of the Puskor a white layer. Very similar that all the outcrops that we see before.
Alright, that's as much as we can get.
Okay, it's okay.
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 Alright, whenever you're ready, bro. Yeah, bring it up. Alright, ready to off. Just like that. Ready to off.
Oh, ooh, yeah. It's a good sample.
It's good?
Yeah, it's good. It's really good. Yeah. Thank you very much, guys.
Yeah, yeah, of course. Alright, what's coming up in that?
Yeah, it's very.
I don't have a single core yet. Now that we're here,
You want
like
one here,
dedicated.
but it's not too soft this area. We only can recover maybe 6-7 centimeters, even less.
only 5.
Yeah.
So
Maybe in the area we go to follow to 6.6 that maybe there are more sediments.
take one there.
Take one here.
Take one wherever you say. Yeah.
Okay, okay.
Yeah, for me, I will never know what I have until I look at the microscope. So anywhere that, you know. So
Okay, okay. So
when
yeah.
everything is right, take it.
Okay, one for you. Okay. So here was Nick. Nick's a good guy, and the field of research is to try to recover every microorganisms that are in the sediments. For example, this pushcar that's maybe only 6-7 centimeters is enough for its research.
So no more push bars?
No more, and go to 0.6. The things that we saw in the course in the geology fiction is the color that is important, because the color gives you a lot of information about the aspects of the chemistry of the course and the material, and also the mineralogy, the composition, the relationship between the minerals, the side of the grains. That is very important.
ROVs back on the moon.
And also we take samples to describe under the microscope. Sorry. The information that we can take under the view of geologists could be very important to do a cartography of this area, or maybe the relationship with fossils or other organisms that we can show under the microscope.
Oh, right. I forgot about that. Yeah. No, you need to put it back. That's fine. That's okay. I no longer push. Yeah. Okay. I forgot about that.
So we go right now to another point that we in our schedule to find the methane seeps, or any proof about it existing. Here we pass for another outcrop of we can take a core here.
Want to do it here?
With only 5 centimeters is enough for us. So here we go to take another pushcar, or we try to recover a pushcar for Nick, the guy I explained you before.
What are they gonna be, about 70 out from us? Yeah. Nice. Alright, what's coming out?
Here in control room, there is no Miguel again. It's here Victoria with me, that is collecting different events that the researchers are looking for. We do a recovery information over every dive, with information about the coordinates, depth, and also a lot of data for example, salinity, temperature. For every site, we need to take any point, any sample, any observation.
Go just a little like this. That's right. Really push.
Yeah.
Debbie?
Yeah, it's okay there. So here you can show the push score is better than mine. Maybe geologists don't have luck today.
Where are we at?
So we also get the 0.6.
Fantastic. This is a core for geology.
No, Nick.
Oh, for Nick.
Yeah.
So close to that. Just working it. Get past the question. You got it. It's a little bit more, huh? That's it.
Spot them out. Okay.
And ease him up.
All right. Sometimes let's start right off the bottom. Make that visual now. Oh, that's me off the toaster.
So, is there anything?
Yeah, a lot of. So we have here a good sample for Nick. I hope that Nick is happy. Yeah, great.
All right.
One thing that you can see in this sample is the porosity or open spaces in the sediment. Sometimes the rocks and the sediments are not too hard or consolidated to be impermeability to water. Rocks like carbonates, for example, the water can easily pass through it, but other like granite is one rock where the water can pass through.
Smush it. Alright, push them in. Yep. Alright.
Continue on.
Keep following. Keep going.
Let's do it.
To 0.6, please.
Warp speed.
Rich, ROV's back on the move.
What?
Updated position.
Alright, so we're about at 0.6. Wait, 0.6.
Point, wait, 0.6.
Okay. We can go to the right, this area.
Okay.
Thank you.
No problem. Uniform pulse. Alright.
Do you want to carry on to 6.1?
We can go here to take a look.
Yeah.
Different animals. Are you?
Maybe a burrow of some kind.
So we need to walk around. I like the camera for that.
Thank you.
Yeah.
See if we get there though. Yeah. Star.
Yeah. So we go fall into the. Yeah.
Cool.
Blast off.
Is that
Warp speed? That's what
Warp
we'll see.
speed.
Warp speed. It's 6.1, right? All the way to the end.
Yeah.
Yeah, please.
alright? Where's the main?
Just want to get up over this hill. I think the biologists are going to come in and take over.
Rich, updated position.
And they said I just want to go back to
Here first
wanna
and,
go to a particular
go later. Oh no, go first here.
Yeah.
It's the same that this side, so it's okay. Go to that. Go there.
set down.
Yeah, please.
It's over there.
There are something. There are something
There's a
here.
crab there.
Let's scooch over there so we can look at all this.
Wow,
It's a crab.
that's a big one.
Unbutt crab.
Wow.
It's all the vinyl. The vinyl crab.
Alright, coming down.
I've seen these really interesting, like, crusts.
Wow. I just saw that the animals prefer rocks than sediments to live.
Yeah. Something solid.
Yeah.
crab looks like he's spoiling for a fight.
Yeah.
Wow.
Little critters.
Got it.
Keep going.
I don't know, do you want to take another hunk of rock?
For me, it's good because this area will be difficult as before.
Yeah.
Because we had the rocks and under the sediment rocks will be 2-3 centimeters under the ground.
Yeah. It's neat to look at though.
Sure is.
What are those little white things on there? Some barnacles or something? In the sponge.
Are you wrapped out? Yeah, I'm wrapped out now.
I'm good.
Okay.
Yeah.
All right. Continue on.
Yes, please.
Cool. So I don't know if the biologists put waypoints down, but if you guys see anything, you want us to pop a waypoint on the map. That way we can find it really easy coming back.
Sure, yeah.
So it's really easy for us to do. So Vámonos.
One fact is probably all the big animal that we are looking right now are all blinds, because as you know their darkness is permanent in this area. So they have other elements to know what is around. Maybe we can check here if that is possible, another push core. Yeah.
Who's this for?
For geology.
Okay.
Wow.
Alright, let's see if we can find, looks like that might be a little bit better.
Because this is very similar. That here right now has maybe take one sample here. It is possible.
Yeah. We'll give it a whirl. Alright, so some bottom. What's coming out? Number 11, right?
stuff at anybody. Like usual. Okay.
So geologists will have luck this time.
I'm ready.
Let's check it out. Oh, no.
No. We're optimistic though.
Victoria is laughing.
This guy's nice easy to us now.
No luck today.
A little bit more. How does that feel? No mas?
It's coming in slow.
Yeah. Here you go. Okay. I got pulled down, but I'm just
The animals, as I said before, are blind, probably blind, so all then. The form that try to go out or to find something to feed is have an incredible force that even we can imagine.
That's it.
Sometimes chemical, sometimes with antennas, sonar. I don't know. We check one. That's okay. Oh,
Oscar, you may want to say to the audience about the acorn worm that is just further away. It's the second common species of acorn worms we have in these areas, okay?
Okay.
So it is an acorn worm, as people write in the chat.
the specimen that was so over there. Okay, you saw Nick, I think so. So there are almost transparent worm next to the point that we check the push core. Why quiet? So another one.
I saw in the chat people were arguing if it is an acorn worm or not. It
Okay.
is.
It is, Nick?
It is.
Okay.
taken off.
So go to 6.1.
And there's our worm trap right there.
Yeah. Here you have a.
Oh boy.
Wow.
Oh boy.
A zoom to the worm.
Eats and defecates.
Yeah, you can see the.
Oh yeah, you can show the process of the worm is processing the sediment, is filtrated the sediment, Nix.
This is
Yeah, but it's filtrated the sun, right?
It eats the sun and filtrates the detritus
Yeah.
or other ingredients of the sun, correct?
Yeah, that structure, that tubes, we in different sites we can show it, and it's something that in the geological record is very common, even to Eisenstein, like the Cambrican, or Cambrian are 400 million years ago. As we say a couple of hours ago about the living fossils, is that the record of these animals or plants or other organisms are the same that fossils. The living fossils are animals that don't change for millions of years even. And it's one of the objectives of this expedition. And even the worm tooth that we saw before is something that we can show in the geological record, and other structures that are fossils also, that is not that also that is not our organism or a bone or other structure that we can recognize as organic. For example, the older record of the a living one is a cave. I think that they have 3,500 million years ago in Greenland.
Yes, I think it's SD3 is red cooking, but SD1 brown. So I just turned it on.
So goodbye everyone. I take my position with Armando. That is the IP of the expedition. So thank you all to watching us, and see you soon.
Armando, are you happy for us to make the turn and start heading back up the line there? Are you happy for us to make the turn and start heading back up there? Yeah, as fast as we can.
Hello everyone, I'm Armando. I am along Lauren, the principal investigator of this expedition. We have come here just to remind the audience here in order to look what we suspect there may be in the area around very ancient ecosystems that is based on the evidence I have taken for the last 15 years in the microbial life of the Atacama Desert, which is just in front of the continental slope we are now investigating. that tell me that maybe in this area, based on lucky findings, so to speak, on other sampling in this area, as I said, in very ancient depositions here. So when I say ancient, let's remind that the whole South American plate has been more or less in the same area here for the last 150 million years. So we are now, in this moment, looking for what we suspect may be one of the sources of energy for this ecosystem, which are methane seeps. Methane, if you haven't already not told this about it, in this dive, today's dive, I mean, is a molecule composed of a central carbon surrounded by 4 hydrogen atoms. And why we're looking for methane seeps, because it seems that the methane seeps is methane is being originated by all the organics that are being brought by the plate that is just in front of the South American plate, which is the Nazca plate. The Nazca plate is going down the South American plate, and as it's going down, and all the organic that have in that plate get down the South American plate, and thanks to pressure and heat, they are transformed by methane.